Good morning, everyone. We're going to try to get started in just a minute, so I'd like to ask you all to head to your seats, please. Oh, this group got very quiet very quickly. All right, well, good morning. As I said, welcome to today's conference on enhancing the application of real-world evidence in regulatory decision-making. I'm Mark McClellan. I'm the director of the Duke-Margolis Center for Health Policy, and we appreciate all of you making the effort to be here in person. I know many people are also joining us online. We thank you all for joining us as well. The topic that we're dealing with today is a critical one. It's about advancing the nation's health information infrastructure in a way that supports better evidence in a wide range of clinical and health policy decision issues. Today, focusing on the use of real-world evidence in regulatory decision-making. For some time, there has been growing interest and growing capabilities for using the electronic data that are increasingly part of our healthcare delivery system, as well as data that are increasingly generated by patients and others touching on our healthcare system to improve our understanding of what works and what doesn't work in healthcare delivery. And in terms of medical technologies. Evidence generated from these data, often called real-world evidence, is helping to bridge the knowledge gap between clinical research and clinical practice to provide a much richer basis, potentially a much richer evidence base for healthcare decision-making. And a range of groups have developed a growing experience with applying real-world evidence in the post-market setting for a number of different uses, such as payment for clinical care and quality improvement activities. But the application of real-world evidence in the development of drugs and devices and in regulatory review is an area that hasn't been as much of a focus and where there appears to be a considerable opportunity for innovation. Real-world evidence has been used by medical product regulators at FDA, mainly in the context of safety surveillance activities, but more efforts are needed to explore how real-world evidence could be incorporated into the regulatory framework and become a stronger source of evidence for regulatory decisions. The application of existing and emerging approaches to real-world evidence generation consequently may offer new pathways to address regulatory requirements and as well, important means of meeting the needs of post-market regulatory decision makers through better evidence. The conference that we're going to hold today and tomorrow to discuss these issues builds on an expert workshop that we convened earlier this year focused specifically on how real-world evidence can be used to approve a new indication for an already marketed drug. This is certainly not the only regulatory application of real-world evidence. Many activities are underway as I mentioned earlier, in supporting safety surveillance and potentially other insights about drug labels. And there are a lot of activities underway at the Center for Devices and Radiologic Health as well. But this is a good focus area for illustrating some of the challenges and the opportunities to build on existing systems, knowledge, and opportunities to make real-world evidence more relevant to regulatory decision-making. During today's discussion and in follow-on meetings, we intend to cover not only this example, but other examples where real-world evidence could be applied in regulatory decision-making to promote the safe and effective use of medical products. We expect that coming out of this effort will be additional papers, further development of case studies for applying real-world evidence in regulatory decision-making, and other activities as well. So we appreciate your joining us this morning for this important step on what we think will be a very productive and extensive path towards better use of real-world evidence within FDA's regulatory framework and the exploration of policy options to enable further progress. There is a lot going on in this space, as I mentioned, a lot of potential to improve the lives of patients, and having you all with us today is going to be an important step in making all that happen. Before we get started, I'd like to briefly go over our agenda and then a few housekeeping items. We're going to have 4 sessions today. Session 1 is going to provide a broad overview of the current landscape of real-world evidence and identify some potential applications in regulatory decision-making. It's going to consist of a couple of presentations and then some time for discussion with a range of expert perspectives and all of you. We're going to use that opportunity to explore the different views that stakeholders have on real-world evidence, as well as areas of common understanding around terminology, what we mean by real-world evidence, the evidentiary gaps that real-world evidence can help address, and ways that it can be used more routinely to support evidence development in the regulatory context. Then, in session 2, we'll introduce a use case, a regulatory use case for approving a new indication of an already marketed therapeutic, along with some other potential use cases, all focusing on when randomized designs, as opposed to other methodologic approaches, should be employed to generate real-world evidence for regulatory requirements. Session 3 is going to build on Session 2 with a focus on the data, study design, and other methodologic considerations for research protocol development and implementation. And again, Session 2 is going to look at the question of when randomized approaches can be used with real-world evidence. Session 3 will focus more on how to carry out these studies. Then in the last session of today, Session 4, we'll identify some areas of drug development and regulatory science that could potentially be enhanced through better, more routine application of non-randomized real-world data. Altogether, the goal of these sessions is to highlight key opportunities and practical challenges for policy development, and that's going to be the focus of tomorrow's discussion on policies related to real-world evidence use. Before we start as well, a few housekeeping items. This is a public meeting. The event is being broadcast online right now, so everything that we discuss here today is part of a public record. As you'll note in the agenda, there are a number of panelists who are going to help us lead each of the sessions with some opening comments. We are going to set aside time for broader discussion with these participants in each session, so for those of you who are here in attendance, there will be a couple of microphones set up, I think, towards the front. Actually, I guess we've got microphones on the table, so be sure to use those when you have a comment to make sure that everybody in the room and online can hear your comment. For those of you who are up here speaking, one of our staff members, Pranav Aurora, Pranav, let me say hi, is going to help us keep track of time during the presentations. And I think that covers all of our logistical issues to get started. So now I'd like to actually get going and introduce my colleague Greg Daniel, who's going to start session 1 with an overview presentation on what real-world evidence is, some definitional issues, and opportunities for applying this kind of evidence in regulatory decision-making. Thanks, Greg.
Thank you, Mark, and let me echo Mark's sentiment on welcoming all of you here to what will be an important discussion over the next 2 days. I'd like to kick things off with some, just basically, what is real-world evidence, and what do we mean by that term, as it will inform a lot of the discussion that we'll be having today. So first of all, why are we talking about it now? The concept of utilizing observational research, data from across the continuum of healthcare to inform decision making is not new, but recently there's been a growing importance of using real-world evidence and growing opportunities to do so. So what tends to be driving this is that we have the availability of digital health data across the healthcare setting, from claims to electronic medical records, data coming from patients themselves, are increasingly growing. In parallel to that, we're also struggling with costs across our healthcare system. So drug discovery and development is longer and more costly. which is resulting in a growing public attention on the cost of drug development, but also on prices as well. Providers and payers are struggling with figuring out how to best design the healthcare system from a payment and care perspective on moving towards value and outcomes and quality versus volume of care. And then patients themselves are becoming increasingly more sophisticated. They're becoming more educated about their own conditions, and they're wanting more information. And so all of these sort of things are driving the need for more evidence, better evidence that can support decision making. And so learning from the real world experiences of patients as they pass through the healthcare system can help begin to fill some of these gaps that oftentimes are left after clinical trials are done. And there are questions about how products perform in terms of quality and safety in a range of patient populations. So real world evidence has value for stakeholders, and opportunities to obtain more precise information on which products perform better in which patients. Real-world evidence can also help bring forth important evidence on the costs, value, and safety across the healthcare system. And generally, developing real-world evidence can come at a lower cost, potentially, but I think you'll hear some discussion around that today. And each stakeholder group has an opportunity to benefit from some of these, from real-world evidence. For example, on the industry side, we'll hear some of that today, but generally learning new information about how available therapies that are already on the market might perform, and might benefit other patient populations, as well as identifying gaps where perhaps innovation is needed. Real-world evidence can get a great assessment of how current available therapies are addressing the needs out there and where there are opportunities for new innovation. Payers are also leveraging already this data to help inform coverage and reimbursement decisions, to help guide programs that are aimed at quality improvement and support for increased value, and payers and providers as we move more towards payment models that align payment with value. Data in the real-world setting can be critically important to help understand how these products are delivering on value. And then patients themselves and the interaction between patients and their providers can be informed with better evidence on what patients can expect from available therapies. And then regulators, we'll hear a lot today about the opportunities for real-world evidence to benefit FDA and the potential uses of that. But FDA is already using real-world evidence as well. Safety surveillance through the Sentinel initiative is leveraging large amounts of data from across the healthcare system to more rapidly identify safety issues. And increasingly, there's interest in looking at how these data can inform other kinds of decisions that FDA engages in. So, there's generally consensus about, what does real-world evidence mean? Groups like ISPOR and NEHI and others have put forth definitions, have begun to define terms like real-world data versus real-world evidence. And the baseline definition essentially is evidence generated from data collected outside of conventional randomized controlled trials through appropriate real-world study designs and methodologies. And generally, most agree that that's a reasonable definition for real-world evidence. But the challenge is that this definition is broad and limited and doesn't necessarily capture all of the complexity of evidence development, the users of the data, and the methodologic considerations that go into developing robust, reliable, real-world evidence. So that's where I'd like to start to perhaps unpack what we mean by real-world evidence and go through with a cleverly developed figure here on how best to look at this. So real-world evidence is not data in themselves. You need data from the real-world setting to begin developing evidence. And so where's this data coming from? In this figure, we can see, we sort of have outpatient clinics, health plans, hospitals, and increasingly patient networks. So these exist across the United States in various pockets, but these data are routinely generated and are made available for developing evidence. So the users of these data include individual stakeholder activities. So health plans themselves, patient networks themselves, and hospitals themselves can use their own data to develop evidence, and they're increasingly doing so. What we're also seeing are large networks or systems that are beginning to, and have been for some time, linking data from multiple health plans to hospitals, bringing in patient data. In the examples that I have here, NIH Collaboratory, PCORNet, Sentinel, and also on the far right, IMEDS, are using these data to develop very specific applications for generating evidence and developing an infrastructure with common data models and an ability to more routinely tap into these data systems and generate valuable evidence. So one opportunity for real-world data themselves to inform evidence development is actually by making traditional clinical trials more efficient. And so what I depict here on the bottom is the spectrum of evidence that many of us have seen before, but ranging from traditional adaptive clinical trials all the way through to retrospective database analyses and case reports. And all of these studies lie on a spectrum of evidence that these data can be useful in supporting. And so as I've mentioned, these data can help traditional randomized controlled trials by becoming more efficient, but new applications of these data using large simple trials, pragmatic clinical trials, randomization in the clinical setting, prospective observational studies, retrospective database studies are being generated by these large networks. And so on Sentinel, and don't get hung up on these cones, they're my attempt at least illustrating right now where these applications, or the general types of study designs that these applications are utilizing. So Sentinel routinely leverages databases, doing retrospective analyses on safety concerns. But even Sentinel, as you look at sequential analyses over time, we're getting more into sort of prospective looks at these data. So you can see I'm sort of trying to overlap here a little bit in PCORNet and NIH Collaboratory. While they can also do retrospective database analyses, my point here is just that the focus is on doing more prospective observational studies as well as randomized designs. But over on the far right, I want to emphasize that you don't need these large networks in order to do these kinds of studies and to bring valuable evidence forth. But the growing interest in leveraging multiple data systems and developing this quote-unquote learning healthcare system that can have multiple users and multiple uses to develop evidence is growing. And so I think you can perhaps see, okay, so data, users, methods, all of these encompass what we mean by real-world evidence. And I'm not sure if this is shown on the screen. Actually, the most important part of this figure can't be seen on the screen, but I can see it because it's way at the bottom. And so what it shows is that what's real-world evidence and what's not, okay? And that's the critical important piece here. And so what we have is that generally, what we mean by real-world evidence is inclusive of, so it's basically a bar that goes under everything from the large simple trials and pragmatic clinical trials all the way through retrospective database analyses and case reports. Real-world evidence does not generally include traditional and adaptive randomized controlled trials. Even if real-world data elements can support those trials themselves generally are still tightly controlled, have more narrow populations, and don't necessarily reflect what happens in the real-world setting, whereas pragmatic trials, observational studies, and retrospective database analyses and case reports generally do a better job of reflecting what's really happening across the healthcare system in the real-world setting. So as I mentioned, even within traditional randomized controlled trials, real-world data elements can provide perhaps more targeted, more efficient recruitment of participants in the study, potentially reduce burden on data collection depending on what the data are being collected and if they're valid and reliable methods for collecting data that would be sufficient for randomized controlled trials, and potentially reducing the time because of those efficiencies, time and cost of trials. But data can also be used to generate new evidence on products, as I depicted in the previous figure. So as I leave this presentation, I want to touch briefly on, so when we talk about the FDA or regulators having increased opportunities to leverage real-world data, we're not necessarily just saying that FDA can approve a brand new drug on the market utilizing real-world data. There are a range of decisions that regulators make that are decisions on products that are even already on the market. So, for example, a product that's already available on the market and has evidence supporting its use might have new populations, and so a new indication might be one kind of decision that the FDA makes. Changes to the label, even including more information or removing information, a range of post-market commitments and phase 4 confirmatory evidence, and also safety surveillance. So there's lots of opportunities that FDA can look at where, if, and when real-world evidence might support those decisions. But it's important to go through, and we'll do that today and tomorrow, on what are the considerations with regard to the data themselves, the validity and reliability of those data and how they're collected, the outcomes or endpoints that are being sought, if they're endpoints that can be measured with these data sources, or perhaps they can't. And so those kinds of considerations need to be addressed when thinking about potential opportunities. And also randomization versus not. When is that important? When should we do it? Do we always have to do it? Those are very important considerations and worthy of certainly the discussion that we'll have today. But I also want to foreshadow into tomorrow that even as we discuss opportunities for FDA, they're not the only stakeholder utilizing these data, potentially utilizing, as I've mentioned, and making sure that we're building a sustainable infrastructure to support its use will be important. And so I have 4 points here that came from a recent paper by Rich Platt and Jeff Brown that talk through, what are the fundamental components of an ideal infrastructure. It needs to be trusted and valued by all stakeholders. It does need an appropriate governance and sustainable business model for its use, and it also needs to be adaptive and evolving so that it can be responsive to the needs of the decision makers. And also capable of engendering what they've called a virtuous cycle of health improvement. So it's one thing to generate the evidence, and even my figure kind of stops at the evidence. But the most important thing is, how do we use that evidence? How do we use that evidence to inform decisions and improve care? So as we go through that, there are lots of questions that need to be tackled just on that. What are the appropriate business models? How can we improve the data systems to bring more value back to the stakeholders that are participating? And are there specific policy levers or incentives that can encourage improved and increased development of such an infrastructure? Thank you.
I want to thank Greg for helping us set the context and provide some definitions. For those of you who could not see the whole slides earlier, I think we've got the slides resized now on the screens, and the ones that Greg presented will be available as part of the resources for this meeting afterwards. As Greg noted, it is possible through so-called real-world evidence, which he thoughtfully defined, to obtain data, real-world data on a broader range of patients and care settings and outcomes, including long-term outcomes that might be possible in traditional tightly controlled trial settings. That leads to, though, a spectrum of design issues and the opportunities for learning about issues that cannot be addressed as easily in tightly controlled trial settings where those are not feasible. But there are some very important methodologic considerations, and that's what we're going to focus on today, is how to develop and use this evidence as effectively as possible. Right now, I'd like to turn to Janet Woodcock, the Director of the Center for Drug Evaluation and Research at the FDA, to provide some contextual context-setting remarks as well. Thanks, Janet.
Thank you, Mark, and good morning, everyone. I'm going to talk about FDA's use of real-world evidence, both sort of current and then future. And Greg gave you some nice definitions, the definition that was put up there is more or less the classic definition, where real-world evidence is gathered from that data generated in healthcare. But I think we have to realize we're in an evolving situation with regard to data gathered from healthcare. And data gathered from healthcare has always had one characteristic: it's not been very good, okay? It's been confounded because of it's used in billing. It's had all sorts of other problems. It's had a lot of different problems with the data standards, or what people are calling things, so forth. But we're moving away with that with the implementation of electronic health records and so forth. We actually have an opportunity to sort of redefine real-world evidence and make it more inclusive as these other types of data become available. Now, real-world evidence, as classically defined, has long been used by the FDA in regulatory decisions, of course. And the most obvious one is adverse event reports and case reports and things that were coming out or reported out of healthcare that happened in healthcare. But also, we've used natural history data for a long time, including as controls, and that may have come from registries or just from data gathered out of the charts and experiences of patients who had a rare disease, for example. We've used observational studies, both retrospective types and prospective types of observational studies, where those data are collected, deliberately collected, but from health records. And we've used registries extensively, especially the Device Center. So we are very familiar with these data, both the, warts and all, shall we say. More recently, we had the Sentinel system, which is an attempt to institutionalize and use the digital data that exists, primarily the claims data, but with some electronic health records put in there. And we're closing in on about up to 200 million lives of that kind of data, all right, the claims data. And that is both retrospective type of analysis and prospective analyses, and we're trying to more institutionalize the prospective analyses out of that. But now FDA is trying to think about, okay, well, we built this infrastructure with Sentinel, Common Data Model. PCORI's built some as well. We have the collaboratory. Can we use the infrastructure built by all this to answer or investigate at least other problems, maybe answer other questions? So we are going to step into that as well. That won't be Sentinel, but it will be utilizing the infrastructure that we have that links these data, the Common Data Model and so forth, to experiment on how this type of evidence performs in answering questions other than safety questions. And we also, because we'd like to extend this more to the electronic health data and what's in there, we're investigating, can we link that with PCORnet and with other information that's around. And devices, of course, is really exploring linking it to registries, other sorts of data. There's also death records. There's all sorts of things that aren't captured particularly in the healthcare system always, but we'd like to know as outcomes that could be linked. So an important new area that we're just starting to explore, I think, and that has been explored with the collaboratory more than anywhere, is really, and this is the hybrid, it's on the far right or left, depending on how you're looking at it, of Greg's diagram. And okay, it's not classic randomized controlled trials that have their own case records over here, their case report forms and their own protocols, and they pull the people out of the healthcare system, basically. They have investigators doing these things to them and so forth. This is, can we randomize people within the healthcare system and do a trial inside the healthcare system, utilizing the data collection methods of the healthcare system, and then put that information together, randomized information, where we have a better way to link the causal inference because we've randomized the people and learn that way. And so that's what we're really interested in. I think we're also interested in talking about here today. Because, you know, it's wonderful. It's terrible if drugs have really terrible side effects, but it's easy to find usually because they're very dramatic and increase. And it's wonderful if drugs have really huge treatment effects where the people are cured. And that really isn't the problem with most drugs in determining whether they work. Most of them only have an incremental effect. Not everyone who's exposed gets the benefit of them. And so that's where you need randomization, because you don't just have something that hits you in the face. And so it's really clear, though, when drugs are developed, and other medical products as well, and then once they're approved and get on the market, despite the huge investment pre-market in these trials and learning as much as possible, there's a huge number of questions that are still not answered about how to use the products. This is very disappointing to all of us. And of course, the answer can't be, well, we'll answer all of this pre-market, because the healthcare system is changing, and new drugs are coming on, other new interventions, and that's the system that you put the new drug into. So you really need to be able to answer questions in a robust way after drugs on the market. How to use a drug better, its place in the armamentarium, and that's who it works in, who should start on a different drug, what combination should be used. Often we don't know all that, any of that information. And then, particularly for this conference, additional uses. So what additional uses can it be put to? Typically, we prove a drug out there, people start using it for all sort of related, but not exactly the people who are in the trial kind of conditions, right? And not indicated on the label. Now, how can we quickly and efficiently find out, is it really worthwhile for those people to take that drug? Or should they be doing something else? And that's where we think that perhaps randomized trials within the healthcare system, if we can get that up and working, could efficiently answer such questions without costing, $500 million and taking a very long time, and actually give us the answers that we want, instead of having a huge number of people sort of empirically exposed to a drug before we ever find out after 10 years or so whether the juice is worth the squeeze. So that's different, really, than what, quote, real-world evidence has been used for before. But here we're talking about a new kind of real-world evidence that we haven't been talking about before, where we perform randomization or other activities that allow us to trace back the drug effect and clearly isolate it. And but it doesn't have all the apparatus and trimmings of a conventional randomized clinical trial. But it also has enough rigor that we know the data actually has some validity. I'll talk about that a little bit more. So currently, for drugs, have we ever done this for an indication? Yes. But it's very rare, usually for rare diseases. But we may get convincing reports out of healthcare about a response that are well enough documented that we would, say, add an indication, something like that. Devices happens more often. There's a lot more, for some devices, there's a lot more face validity to the effect, right? And devices also have a lot of rapid turnaround in their life cycle, and they're evolving, and so this is very pertinent for medical devices. So in the future, maybe the second and third, et cetera, indication, if it's related or it's smaller or it's something that's generated out of healthcare that the drug might be used in, you could do randomized controlled trial, but do it in really utilizing the tools of the healthcare system. That's the thought. And what has to happen in order for that to be acceptable for all of us, acceptable evidence? Also, for some things, as a collaboratory has shown, cluster randomization is a helpful tool where you're doing a comparative study and you want to know, should we use this intervention in these people or should we use that intervention in these people? Both of them are out there. They're both considered acceptable. As I said at the beginning, often we just don't know which one might work better. So different types of cluster randomization often work very well in this comparing two different standards of care. Although there's always controversy about this. When one turns out better than the other, people then say, well, they all should have been on the better one. But of course you didn't know that at the start. That's the precondition for equipoise, that you would actually do this study. So it's also I think, possible in some settings where the natural history is pretty clear to do prospective open label studies for new indications. That's where you don't need that control necessarily. You can use what you know about the natural history. So, and these are typically done in the same settings pre-market. So we have, for oncology, we have pre-market uncontrolled trials, and it's perfectly feasible that you could do the same for new indications post-market. The only caveat there is, as I said at the very beginning of this, the problem with real-world evidence has not been very reliable. And in cancer, for example, Laura Esserman told me that, who's a cancer doctor who's working in this area, that often in the charts of breast cancer patients, in their medical records, there's the wrong stage of disease. All right? And so that's a really, really important thing for whether you're going to get this treatment or that treatment. But it's wrong in the record. And so she's trying to pioneer methods where you get it right the first time, where you document and agree upon, and there's maybe another party agreement before you enter that diagnosis in the chart. And I think that's a really good idea because, number one, it'll improve quality in health care, right? And number two, it'll make that data reliable, that we can, and valid, that we can use it as we do this kind of study work. So that's critically important, that, that those data be, and all key data points that are used in use of real-world evidence be valid and reliable. And, it hasn't been so much of it's been a terrible problem with claims data because they're used for another purpose primarily. And so, the relationship to the patient may be more tenuous than, and that's why I've used it mainly for safety issues, right? Because the safety problem is, may be described in there, and there's been a huge amount of methodologic work to kind of work around this problem of claims data. But that's going to have to happen. We're going to have good documentation of the key data points and really focus on that. And also, I think more use of registries that are embedded in healthcare rather than having to be pulled out somewhere and do a separate registry and go to those registry visits and all that may enable a lot more us to understand the natural history of disease better. These would be like virtual registries where we pull the information out. But again, if the measures are not somewhat standardized and carefully documented in the records, whatever the records are, we're still going to have a lot of noise in the signals that we're trying to discern. And that, I think, is one of the fundamental problems. So data reliability, number one, I would say. Informed consent. I think we can lick this one on, whether you're going to be talking about it next 2 days, but, people do need to consent, and there's been a lot of uproar about consent. For example, they randomized, there was a recent study published on randomizing residents' hours to see whether, because people were having, due to the hour restrictions on residents, they were going to walk out in the middle of surgery and hand it over to another resident. Okay, they weren't the primary person doing the surgery, they were the person learning the surgery, but they'd have to leave because their time was up. So they randomized and were flexible. Well, there was a big uproar over that about whether the patient should have been consented, that the residents might be in one schedule of hours versus another schedule of hours. So, yeah, so we can't underestimate this challenge Methodologies, traditional methodologies can be applied if we can get these other things right, and also additional methodologies are being developed. And then I think the final problem that we face is linking all these different types of data collection sites and systems together. For example, whether somebody died or not, or and that's kept here, and then the registries are kept there, and the claims data is kept here, and as Greg said, the clinics keep their records, and these things are not interoperable, nor are they linked right now. Now, this is a more straightforward methodologic problem, although it does raise privacy issues that are going to have to be dealt with as we work through this. So because we're all really motivated to do this, because we still don't know what we need to know about medical products and also all healthcare interventions than we should know in order to use these things. Now, the good news is I think we know more about the use of medical products than we do about many other interventions that are done in healthcare, right? So it's not like medical products are following along behind. But, the goal of many of us is to try to improve, as people call it, learning or knowledge accumulation in healthcare. And this effort will probably lead the way. Because it's ripe. There's a lot of motivated people coming together. We can figure out how to do this, and this could lead to greater understanding, so that when you or I or anyone else goes in the hospital or goes to the doctor's office, there's a better body of evidence there about how to treat us and what not to treat us with. So thanks very much, and I look forward to the conference.
As I mentioned, all of our sessions are going to have a good deal of discussion in them, and I'd like to go into that discussion format today with Cliff Goodman, Bill Chin, and Greg, I think, joining us on stage as well as Janet. So if you all could make your way back up. You've already been introduced this morning to Greg Daniel and Janet Woodcock. I'd like to just briefly introduce two other distinguished leaders that we have with us today who've been thinking about these issues of real-world evidence extensively. Cliff Goodman is Senior Vice President and Director of the Center for Comparative Effectiveness Research at the Lewin Group, and Bill Chin is the Chief Medical Officer and Executive Vice President at the Pharmaceutical Research and Manufacturers of America. So what we want to do now is spend some more time addressing the key real-world evidence concepts and some of the issues that Janet and Greg have already pointed out this morning in terms of how to identify and use effectively real-world evidence in addressing patient needs. And again, keeping in mind that we're going to have a lot of focus today on potential for using randomization in the real-world evidence context for developing evidence for regulatory decision-making. I'll just start out with some perhaps some initial thoughts from those of you who haven't spoken yet. So Cliff, maybe I can turn to you first.
Thanks, Mark. Glad to start. Real-world evidence is a kind of a wake-up call, and I want to tell you in just a minute about the wake-up that occurred. First, real-world evidence does support the basic concept of effectiveness. How well does something work in the real world as opposed to the idealized world, which is efficacy? And the best story about this that I've heard was one told by Hans-Georg Eichler, who was the #2 at the European Medicines Agency, and about which he wrote a beautiful article in Nature Reviews in 2010. And regulators, scientists, and so forth, and he realized that the EMA was allowing things to go on market. No, it's not.
Can make it even better, Cliff.
Guess so. Oh, gosh. That's fine, man. Good. He was realizing that as an official of the European Medicines Agency, they were basically approving for market all these swell new drugs and biologics and so forth. And then after a while, he was there, he starts looking around and says, it was a wait a minute moment for him. He said, we're approving all this stuff, but it's not being taken up in the market. What's going on? Well, in Europe, it was health technology assessment agencies and national payment authorities who are saying, hold on, glad that this thing got market approval. That tells us that it provides efficacy in those ideal conditions. We got a little bit of data on short-term, more common adverse events, but not rare, longer-term ones and so forth. So his wait a minute was, what's going on here? And what he started to realize is that there's no longer this border, this boundary between pre-market data collection and post-market data collection. And he started to learn about efficacy, effectiveness, and then relative efficacy also versus relative effectiveness, those four concepts. So that realization occurred because the market was changing and the low side, not just one, the low side decision making, those were changing as well. So an important thing that RWE does, and this is, I think, my essential point, is that it rebalances the relative importance of evidence sources in support of decision making. Think about it. If you're a pharma bio company, you designed and you ran and analyzed those RCTs for a few hundred, maybe a couple thousand patients, and that comprised the main body of evidence upon which decisions were made thereafter. Well, that's not the case anymore. That may be necessary, but it's not a sufficient source of evidence. Now, what's tipping the scales? What's tipping the scales is all this stuff from these other sources and saying, well, what are decision makers using now to make decisions about a guideline, a pathway, a coverage decision, a purchasing decision? They don't have to go back to the trials done in a dossier for regulatory agency approval. They're looking at other sources. Furthermore, think about how size of data sources matters. Janet's absolutely right, and a lot of methodologists are right about, well, these are larger data sources, but they're kind of weak. Well, if you talk now to somebody who's, let's say, a chief medical officer of a large PBM, well, I guess they're all large now. There are fewer of them and they're getting bigger. Or you talk to the chief medical officer of a major payer, they're saying, well, yeah, we used to depend on the regulatory submission data. That was very important. But now I got my own data. And I'm not talking about tens of patients or hundreds of patients or maybe thousands of patients. I got data on millions of patients. And if you run a PBM and you're watching scripts, you got billions of prescriptions in refill data. So I know that there's a problem with the quality of data and so forth, but when you start getting to very large numbers representing highly heterogeneous patients, there's a lot of power in the data. Now ask yourself, where's the scale now? Where's the main body of evidence that matters to a decision maker? That's a big change. Okay, a few more points about these sources. Yes, pragmatic or practical clinical trials are a source. Payment claims, pharmacy prescriptions and fills, registries, EHRs, EMRs, laboratory test results, radiographic images, biobanks, specimens, tissues, and so forth, molecular, genomic data, vital statistics data, and quite important lately, and I know there's some folks in the room that are experts on this, patient-generated data, patient source data, patient streaming data, and whether it's from personal phones, apps, and all those other things that I don't understand. It's not just that, though. It's credit card purchases. It's how far do you live from the Whole Foods or whatever grocery store you happen to like that might have fresh stuff. Who lives with you? Do you own a pet? Do you take a walk? So we've got ways to now be collecting those kinds of data to augment not just the claims in the EHRs, but all those others. We have better ways of linking those data and telling much better stories. Now, it's not just those data sources, the RWE sources, it's what you can do with them. We've talked about linkages and so forth, and high-power computing can do wonders, but it's also kind of the analytic tools. And so it's machine learning, it's natural language processing. With machine learning, if you think about those data sources I just rattled off, including how far you are from a grocery store and some other things, machine learning can start finding patient clusters that may be non-traditional sets of patients that aren't the kinds that you'd identify in a traditional RCT up front with certain baseline characteristics. Yeah, it's somebody with diabetes, but it's somebody with diabetes who exercises a couple times a week, does live near a grocery store, is living with a caregiver or a family member who is ambulatory, and with whom he or she goes to the movies once a week. You know what I'm saying? So patient clusters using machine learning can identify groups of patients that will behave much differently, respond much differently than other people with diabetes or congestive heart failure or rheumatoid arthritis. That's an additional bit of power there. So I just want to close insofar as some of the kinds of conversations that we have with pharma bio companies, and if we get into their C-suites, which is enlightened on their part, a nice admission, we say stuff like this. When you're thinking about what products to put in your pipeline, are you thinking ahead already about who the gatekeepers are? And the key question is, who is going to want what evidence when? Who's going to want what evidence when? And those decision points, those locks, those gateways are going to determine the development, uptake, and diffusion, payment, and so forth of all those swell molecules that you think that you're developing, okay? And if you're not thinking about that now, you're going to be behind. Thinking about that now necessarily embraces data sources far more than the ones that are required for regulatory approval. Far, far more. Therein lies that balance I told you about before. If they're just focusing, if they're traditional R&D people who might have developed a swell beta blocker 20 years ago and got wealthy and famous doing that, if they're just thinking that way, they're missing this market entirely. Last point, patient role in this. I talked about patient-generated, patient-sourced data. Think about the role of much more active, informed, and engaged patients in all this stuff. Research and development agenda setting, unmet medical needs. What trials to conduct. Trial design. What are the patient groups? What are the interventions? What are the comparators? What are the outcome measures? Over what period of time? How will data be collected? Okay? Patient, even recruitment into clinical trials. All right? Then what is done with the information that comes out of clinical trials? Post-market data collection. Patients are much more engaged in this. That is another key development here. So if you kind of look back over the 7.1 minutes I've just had, you say, how does that decision-making and data generation environment that I just described, and we're about there now, differ from when the laws that Janet carries out so effectively in those regulations were initially laid down? It's much, much different. Don't forget the thing, a rebalancing of the evidence that matters.
Cliff, thanks very much. 7.2 minutes, a lot of ground to cover. I'd like to turn to Bill next.
Thank you very much, Mark, and also Greg for inviting me. So you've heard already that these are indeed exciting times. Cliff has articulated that. Particularly exciting times for patients. That's your last point. I think the definition of real-world evidence in a broad way, that is evidence that's beyond the evidence that's derived from randomized controlled trials, is still a very useful one. It does, of course, however, put sort of the perspective that we're dealing with RCT as a solar system, and then the rest of the universe is really what RWE represents. So there's a great deal of opportunity in that, but there's also, I'm sure, a fair amount of anxiety in terms of ultimately where we're going to go with it. So I think with this discussion today and tomorrow, there's no question that intuition tells us that it's not a matter of whether we're going to use this. It's really a question of what, when, and ultimately how. And obviously all the sessions will deal with that. We agree that a broad definition is probably very useful because of all the things that we still don't know about RWE. And so as a result, we don't think we should limit our current discussions only to specific types of evidence. Well, clearly, we now and continue to use, and these are both sponsors and FDA, RWE for safety, and we heard certainly about that. But we also understand that it's useful for understanding treatment patterns, how to define target populations, how to benchmark adverse events observed in clinical trials, and monitoring safety. It's also clear that we already have some examples, and so maybe the focus has been on safety, but we have some examples where the agency has utilized RWE for efficacy reads. And very briefly, there's the example of blinatumomab. It's easy enough for me to say, focused on relapsed and refractory ALL, acute lymphoblastic leukemia. And that whole process went through breakthrough, Janet, really excellently, very fast. Of course, there are a lot of good things. There was a good effect size, a really terrible disease. I think there was a very good clinical trial design. But most importantly, utilize RWE as part of the control in this single-arm study. The second deals very quickly with recombinant human factor 7A, where, in fact, for this particular disease, Glanzmann's thrombocythemia, the use of registry data was helpful in adding on to the use here. And then finally, tocilizumab, another mouthful, which is something used in the refractory rheumatoid arthritis patient who is refractory to DMARDs. And this was also, the indication was expanded based on efficacy and safety information collected, from post-market studies. So there are at least 3. I'm sure there are many other examples, or at least some other examples, right, Bob? But the point is that I guess FDA regulations provide flexibility in the requirement for determining or demonstrating effectiveness. And I think the FDA has used its flexibility in this. So a couple of points. One, I think we as an industry applaud the agency and Duke Margolis for having this session, because I think it's about multi-stakeholder discussions like these that allow us to translate our intuition of the use of RWE into reality, into usefulness. Collaboration is key, and clearly having everybody here is also important. But there's great opportunity here as well, and that is how can we think about RWE in benefit risk assessment, and how can we think about it in a broader way and focusing on context of use, something that, Janet, you've talked a lot about with respect to biomarkers, taxonomy as what you've talked about, biomarkers. So the point is that RWE covers a lot. How can we be much more deliberate and focused in terms of understanding what that context is, then asking what are the kinds of data, the quality of the data, the methods that can be applied to each context. One thing that gives us great hope is that with the current PDUFA 6 technical negotiations, which are heading toward completion, we do have an element of it, of enhancing the use of RWE in regulatory decision-making. We'll hear more about that. So this whole idea of combinations, so context of use, really, so the way to think about it is, and my colleague Mike Levy has brought this up, Mark, in previous sessions, is really thinking about the types of regulatory decisions you're talking about, the characteristics of the medicine or the disease, okay? And we could even add another element, which is types of RWE, as Greg has articulated. So how can we sort of develop, if you only have 2 dimensions, a Latin square of these efforts, and try to then identify specific context? So just to give you an example. And Janet, you talked about it, and Greg, you listed a few of these as well. So for instance, on regulatory decision-making, approvals based on historical controlled data and a single-arm pragmatic clinical trial. Those based on RWE derived from medically accepted alternate use of medicine, RWE from another country with quality data, on promising early clinical trial results with monitoring of safety and efficacy of the drug through the use of RWE, subpopulation analyses, so approval based on that. And then finally, fulfillment of post-market commitments. You already talked about replacing phase 4 trials with analyses based on RWE. So these are just meant as illustration, and obviously not to be complete, but on the drug or medicine disease characteristic, and a few of these could be the availability of other therapeutic options, the urgency of the disease being addressed, the size of the patient population, and the drug effect size. So in the end of the day, we really want a better definition and clarity on regulatory framework. And we should build and learn from the examples that we already have, but also think about pilots. And we talked about prospective pilots, and we're going to hear about a case or cases. But I also have thought that we should be thinking about how we can use retrospective data in a sort of learning way. Okay, we're not going to learn new information, but how can we learn from retrospective studies how to perhaps do this work and where to apply it better. So I'm going to conclude by just noting a couple of things. So we believe that RWE represents a valuable source of information. We said that, okay? Two, that it enables sponsors to submit applications based on appropriate and adequate data gathered through the real-world use of a product by medical professionals could actually help and will help patients and society. So the benefits include reducing time necessary to reach approval, accelerating the availability of new approved therapies to patients, driving sponsors to improve the healthcare system's ability to draw from and integrate disparate clinical data, and improving the amount and quality of information available to all physicians regarding therapeutic benefits and risks. Furthermore, FDA-approved labeling, we talked about possibility of labeling could be affected by these data, and they can often then ultimately affect patients' ability to obtain reimbursement for therapies and access to potentially benefit. And lastly, it really, again, back to the patient, okay, just where we began. Patients' interest for all of us to think really carefully about how we can utilize RWE in the benefit risk assessment. And so we encourage, again, coming back to the theme of bringing stakeholders together in collaboration. How do we work together to perhaps not solve the whole big problem of what RWE can do, but how could we solve specific ones so we can learn from those and then grow from there? Thank you very much.
Great, thank you. I really appreciate all the context setting that's gone on this morning. Clearly, real-world data presents a potentially much richer source of information that can help improve patient decisions, as Bill was just emphasizing. But it is more complex, and it does raise some issues in terms of non-randomly missing data and in terms of potential confounding that has been highlighted in one way or another in all of your remarks. I want to go back and start this discussion with similar comments that both Cliff and Bill made. Cliff, I think you said the key issue is who wants what evidence when, and determining how to use real-world data to develop that evidence. And Bill, I think you highlighted the context of use, similar point. And Cliff especially highlighted how real-world evidence seems to be used much more widely now in decisions by payers. I remember that Nature study about the increasing disconnect between regulatory approvals in Europe and covers decisions. That may be because of the different nature of the context of use, where, as you pointed out, the questions really are for payment increasingly about comparative effectiveness and about evidence relevant to specific subgroups of patients. To bring it back to the regulatory context, though, I'd like to push on what you all see as some of the best unexplored or potential uses of real-world evidence in the regulatory context that haven't been used as extensively. If the FDA decisions, or some of the most important decisions, are about the safety and effectiveness of a new treatment, well, obviously it's going to be hard to get real-world evidence on a treatment that hasn't been approved yet. We've highlighted this morning a focus potentially on label extensions. Does that need the same level of evidence? You all have highlighted some other uses. Could you talk a little bit more about where you think the best opportunities are to expand out the regulatory uses of real-world evidence?
Yeah, real-world evidence can be thought of as well as feeding back to the thinking about what trials you run and how you design them. Real-world evidence can tell you that we're seeing outcomes in the real world that had not been built into earlier trials, and therefore when we do our next set of trials, whether it's within class or different class, we might want to consider other outcomes that matter to patients and that affect their quality of life, mortality, morbidity, and so forth. So it's a feedback to the outcomes. It's also feedback to the selection of patient groups and baseline characteristics, as well as those subgroups that you may want to follow longitudinally through the trial and after the trial. So all the way through phase 4. So it's feedback to trial design for which patients, what outcomes, over what period of time, and then also, Mark, in the context of what those other evidence seekers are going to be seeking, a smart pharma bio company and an ongoing enlightened Food and Drug Administration is going to be thinking, you know, we also want efficiency in the system. So while we're doing our traditional RCTs to get regulatory approval, can we be smart about being more efficient about the kinds of data we might want to start collecting now, so when the thing does hit the market and its first few years after it hits the market, we'll have more data sooner to help the other decision makers in the realm.
Janet?
Yeah, well, to your last point, Cliff, certainly the EMA is experimenting with getting the payers, the patients, and everybody together before development program goes forward, because we know the tech assessors mainly are looking at, what they're looking at differently, I have to differ with you, okay? They want quality of life information to do the qualities, because that's how they do their payment, whether it's worth paying for or not, okay? And typically, like, that information isn't collected. And typically, we don't have instruments to collect it, or if we do, they're more like health status instruments. So I think the first thing you were describing was actually research, which is what we have to recognize, I think, is that most health records don't have any patient-reported outcomes in them either. All right, they filling out a form on a computer. Okay, that's what they're doing, and it's not an instrument to figure out how the patient feels. It's how maybe the doctor thinks the patient feels. Okay, so there is research needed in the real world. I agree on. If you want to know what outcomes matter to patients, you have to ask them, and then you have to go through a structured process to figure out how to capture that data in a way that's valid, so that you can then, but that's really what the European tech assessors want to look at, is how does this, if it doesn't impact survival, okay, does it impact quality of life? Those are the two things they care about, right? So typically those are not strictly the endpoints for a lot of trials, even for symptomatic diseases. They might have a scale, they might have a Likert scale or a pain or something like that, but we try to put in quality of life. So that goes back to what Bill was talking about with the, the endpoints of the patient and everything. So we might have to do, you could do research in real world settings to figure out what patients want. But I think that's my understanding. I've talked to Hans Georg and all the folks over there, and they are doing pilots where they're getting together with the tech assessors and patients before the end of the program, before the efficacy trials are initiated, and trying to figure out what outcomes should be structured in there that would meet the needs of all the different parties.
Yeah, I'm just going to say, not all the tech assessors in Europe or Australia or elsewhere use cost per quality adjusted life years, the threshold criterion, but they all care about some aspect of quality adjusted life, whether it's a Euroqual 5D or other measures and so forth. So there is an international interest in better measures, and you are right, of quality adjusted life and similar things. And again, the patient advocacy groups and the patients and researcher groups are developing these and validating them further. I will say though, insofar as producing data that's of use to decision makers, you're now seeing groups in the United States, ICER, ACC, AHA, Memorial Sloan Kettering with its advocates and so forth. You're seeing different groups trying to quantify value in multiple dimensions that use some of those measures and kick in the economic component. We're hearing about cost per quality adjusted life here now, not from our government, but from our nonprofit private sector groups that are trying to provide information to inform these very same decision makers.
Mark, just additional comment to your question, which really, again, focuses on the regulatory framework. We, of course, think, and I think Janet would agree, that maybe the early applications of real-world evidence, beyond the few examples that I've provided, really will be in that post-approval space, thinking about how potentially new indications can be supported by these data. You know, this could be very helpful in situations like breakthrough, where you do have fairly good effect size and evidence that you're helping good benefit risk assessment, but that you're still not 100% sure, right? And so you could use post-approval to continue that assessment, not only of safety, but of benefit. I would maybe editorialize by saying that no one is 100% sure about benefit risk, really, at the time of decision making. And so ultimately, can RWE be useful for all medicines? And then finally, the last frontier, Bob, will be whether RWE, I'm looking at you, Bob. Last frontier will be whether RWE can be useful in some aspects of pre-approval decisions. I think it's certainly. I think you've told me once that not in my lifetime will we ever have RWE and RCT and mixed RCTs pre-approval, but so I don't know how long.
I haven't I'd like to raise.
But the point is that I think we need to continuously learn how you can maybe add RWE to RCTs. I don't think we'll get rid of RCTs as gold standards.
Well, we'll hear from Bob Temple soon. Sorry, we
Right.
probably should have had you on this panel as well, but Janet?
Well, this is somewhat tangential to this discussion, sort of. But, you know, we're seeing with precision medicine, okay, we're seeing we're shifting away from, to some extent, the traditional definition of disease. And we're moving into, you know, pathways and where there are underlying pathophysiologic defects that actually underlie presentations of sometimes rather disparate diseases. And what I would foresee is that what typically would happen in the past is, okay, we'd approve it for some small subset of this, okay, where the pathway, where there were enough people and the pathway was known, and you do a targeted precision medicine program with a randomized trial, and you get the drug on the market, and then people would use it off-label for all the different other defects where you could identify the defect. And that situation would go on for a long time because that's how it's always been, okay? But I could foresee that you could, with good data collection, if there was enough natural history understanding, you could add more subgroups then of people with that underlying defect to the label based on you know, experience in treating them. And so treating them off-label wouldn't be wasted anymore. You'd actually collect that information. Eventually you might understand, and we could rapidly decide whether there was an appropriate treatment for these other groups in other diseases. So that's one example where I think the follow-on, and we'll see that in cancer, of course, but it's actually happening in a lot of other diseases too.
And Greg, you had a comment too.
I just want to go back to a key concept that Cliff brought up about this sort of rebalancing of the evidence, and I think I'm quoting you, Mark, or maybe paraphrasing, but it used to be where, you know, we get as much evidence as we can during clinical trials, and then at the point of approval, we just sort of hope for the best. And it's clear among this group and what we've heard so far that there is lots of value in continuing to learn the experiences of what happens once these products are on the market, and the opportunity to sort of rebalance pre-market data collection with post-market data collection is something that our colleagues at CDRH are heavily involved in now, and are putting a lot of emphasis on building a new national medical device evaluation system that leverages the experience that patients have that are brought forth with registries, and potentially linking that to sources like Sentinel and like PCORNet, And I realize, this meeting is focused on drugs, and there are lots of differences between drugs and devices by which, perhaps that utilizing real-world evidence and new indications, like like we've seen in devices. One particular registry, the TAVR, the transcatheter aortic valve replacement therapy, has built there's a registry on that was built primarily for a payment. CMS coverage with evidence development program, where the data and evidence that were being produced were primarily supposed to be used for coverage decisions, to understand how this is working within the population. Was actually very valuable information that was collected, but was useful for a new indication that FDA used that evidence. And registries are difficult. There's a lot of burden on providers, but linking in and figuring out how to make better use of real-world data outside of registries can be an important aspect.
I want to open this up to comments from the rest of you who are here in just a minute, but picking up on this notion of learning from off-label uses. I think, Janet, you said that potential evidence wouldn't be wasted anymore.
Yeah.
This notion that we definitely seem to be moving farther and farther away, if we ever really were there, of just approving drugs and then hoping for the best. Payers certainly aren't satisfied with that and getting value out of the system, and patients are increasingly dissatisfied with that in terms of being able to get information that's relevant to them and to the outcomes that they care most about. Greg talked earlier about an infrastructure for real-world evidence. Several of you have referred to real-world evidence systems. I have to say that seems more aspirational now in many ways than well-established. We're going to hear today from some of the leading efforts around building a system like FDA Sentinel, and efforts by PCORI, efforts by payers, including CMS, to support registries. But it still seems like there are a lot of gaps. And I'd like to keep this focused on kind of FDA and regulatory decisions. How can the efforts that we're talking about today in terms of trying to use more real-world evidence more effectively in regulatory decisions, how can that contribute to a better infrastructure, better systems of real-world evidence?
I was just wondering whether Janet could comment on the Guardian system. I've heard a lot about the system that will maybe be a kind of a mate to Sentinel to look at benefits. The sort of other side comment would be, you mentioned the CoreNet. Janet, you and I sit on a steering group. Obviously, our great colleagues are sitting back there. We'll talk about the CoreNet. But what is the great potential of a CoreNet to be able to allow us prospectively to be able to utilize those networks that are being developed to be able to affect the use of RWE in a better way?
Including in regulatory decisions.
Including regulatory,
Oh, absolutely. Well, what I think has to be done next, or what we have concluded internally at FDA needs to be done next, is we need to do some demonstration projects, experiments. We need to try this out, see how far we can get, and what are the gaps in the infrastructure. Running Sentinel for a while has helped us to learn, what are missing and what was the power of that in actual use cases where we try to answer safety signal, and either we can or we can't, and we learn what is missing. So what we would like to do is, first of all, PCORNet has the electronic health record data, as I said, and so that's one piece of, that's one kind of information. Unfortunately, that information often isn't interoperable among systems and missing different pieces. The Sentinel has the claims data, right? And then there are other pieces out there that we're also trying to bring in, and there's our federal partners in Sentinel, such as, we can't directly analyze into the Medicare databases, but we can use them, we can access them, we can't just merge them. So the question is, can we do some demonstration projects where we're asking different questions other than safety questions and see how well it works? And I think what you suggested, Bill, is a good one. Can we, do a retrospective prospective experiment where we already know the answer from randomized data, go back and see how I like to do stuff like that, just like OMOP, like actually see how the methodology performs. Does it answer questions you already know the answer to? Because it doesn't make a lot of sense to me to ask questions you don't know the answer to when you don't know how your methodology is going to perform. So we would like to do that and do some projects, maybe merging across PCORnet and Sentinel as much as we can, see how we can link. So we'll learn a lot about that and also try to investigate some questions. I know in Sentinel we did a safety one where there's an ongoing randomized trial, and then we did it also in the Sentinel. Okay, so doing those things, to get your point, Mark, we'll start to get real on-the-ground understanding of what can we and can we not do, what is feasible, what has to be fixed, what bridging do we have to do amongst these and so forth. But that's what I would think. And Guardian, we have to have a different name, okay? Because Sentinel is, our safety system is statutorily mandated, some of this stuff we do. We can use the same infrastructure from Sentinel for other things. You know, we've been urging to do that for almost a decade. Let's have one infrastructure for all this stuff. So, but this other activity is not like a safety type of activity.
Right.
So that's what we're planning to do, and I think that will, you know, maybe in a year or two we can come with a whole bunch of things that have to be fixed, so to speak, or we have to do this project and this project and this project in order to be able to rapidly utilize data from existing networks.
One infrastructure, multiple uses, and more clarity and opportunities coming on these additional uses to strengthen the infrastructure.
That's
we have
right.
time for a couple of questions or comments, if there are any, from the participants here in the room. Bob, I think you do deserve a comment.
I think a lot of this has been said already. It seems critical to me to be very specific about the uses we're talking about. I mean, these things in a way are historical controls. That's what they really all are. You're not randomizing, you're collecting data that exists already. That's what historical controls are. Well, those are plausible, as we've written about repeatedly, when the effect sizes are large, like Janet's oncology examples, and when you think they're actually collecting the data. But as also Janet said, there's places one might look. Just as a candidate, there's been a recent study called Pegasus showing that long-term use of ticagrelor after a year or two is better than not using it. And these antiplatelet drugs, we don't know how long to use them. There's mixed data, it's uncertain. So now somebody did a study, showed a 20% reduction in major events in a randomized study with 20,000 people. Well, there were three groups. It really would have been 18,000 people. It would be really interesting to look within these databases at outcomes like heart attack and death and see whether longer-term use of ticagrelor in these settings corresponded to an improved example. Now, it's a small effect. 20% reduction is a big deal, but that's at the limits of historical controls. But it would be very interesting to see. And I'd be interested in the examples that Cliff was giving of where people have discovered all kinds of stuff by looking at these things. Did they publish it? I mean, did they change their payment? Do we know the payment wasn't changed because they wanted something cheaper? You know, it's really worth looking at examples because this is uncharted territory.
Well, I would add something to what Bob is saying. You know, you have to have a large absolute number of events too. If the 20% reduction was off 6 people or something, or or 10 people, then that's not going to be the kind of thing you're going to see with so much noise in an observational. So you have to have fairly large background rate of events and then a large proportional reduction to find, I imagine. Or we ought to start with that. We ought to start with something that is viewed as relatively easy, not a 0.1% reduction in mortality in a 40,000 person trial.
Okay, we're going to come back to those issues in the next session.
But you also can pick your patients. You can try to pick a patient population just like the highly enriched population they included.
Right.
Yeah.
So, I mean, it'd be interesting to see, is 20% below the limit, forget about it? Or does it lean? Or
20% or what?
Hold the thought of which are the best examples to
Yeah.
start with. What would be the key places to start in terms of these regulatory applications? We're going to come back to that in the next sessions. Time for one more comment, if there is one. Yes, please go ahead. If you can manage the logistics with the mic. Thank you.
Thank you. Grace Lieberman. Question. So I agree that sort of we say that, not our lifetime we'll use real-world data for first approval. I see it too, one that we talked about, it's the infrastructure, it's the data, but also one of it is looking at very different new endpoints. And it seems to me we almost may have a great opportunity of creating endpoints that meet what the patients are really looking and needing, what the physicians really are looking for, what the payers are looking, and sort of like thinking, what's the process? How can we get there? And also make sure that it meets the regulatory expectations. So would like to start that discussion. I know it's something that might take years.
Just to make that a little bit more pointed, I mean, are there reasons why, if it's what the patients want, what the doctors want, and what the payers want, at least for value, or at least the outcome side of value, why would that not align with what's needed for regulatory decisions?
Point well taken. The healthcare system, new drug development healthcare system is not done for the purpose of satisfying a statute. We are really glad the statute is in place because it does protect us from unsafe things and it helps establish some evidence about whether or not something works. But satisfying the statute is not what we're trying to do in the healthcare sector. So if we can think further, a little bit further ahead, we want the FDA to please keep doing what it's doing, but realize that it is part of a broader tapestry, a broader framework, which is getting people to feel better. And the work that the FDA does can be perhaps better integrated early and more richly with other evidence generation needs and communication of those results and getting feedback from the patients whom we're trying to serve. So I love the statute. It's done a great job for the most part, but it's only part of a bigger a mission that we're all pursuing. And so your point on those patient-centered outcomes is great.
We go to Janet and then Bill, and we're going to wrap up.
Well, I don't disagree at all, but I will reiterate my point. Many of the outcomes that patients are interested in are not well captured in the current real-world evidence.
Not yet.
And so that's where we're doing the patient-centered drug development to try and integrate. Those can be captured in clinical trials as well. And the patient groups, I think, are starting to wake up and figure out that part of their role is to determine what is measured and considered benefit, and that's a good thing. But then you have to figure out how to measure it, and and then for real-world evidence, somehow the clinician has to have entered that, or somebody. Now, in the future, we will have more, as you said, patient involvement that we can use electronic whatever. Capture systems for patient-centered outcomes, but we'll have to build that in. It's not there now. It's not in claims data. It's generally not in electronic health record.
I too agree. It's a great point, and I think the problem right now is whether it's common data models, et cetera, we don't actually maybe include all the things that we might need. But of course, the challenge for all of us is understanding the science of patient input rather, and understanding what PROs and COAs might be. But it's possible that using real-world evidence will begin to at least ask hypotheses of issues that might in truth be important and true endpoints for patients, but then need to be tested. So I think that could be a great lever in order to make headway in that area.
All right, I want to thank you all for getting a lot of key issues on the table. We're going to take a 10-minute break now. When we come back, we're going to focus in on what the criteria should be for these kinds of regulatory uses of real-world evidence. Thank you.
Welcome back. We're going to go ahead and get started on our next session. So if you could please make your way to the tables. I'd like to go ahead and invite our next speakers to join me up on the stage. We'll introduce them in a moment. And we're all mic'd and sitting in the right chair. I think so. Great. So I'd like to welcome our next round of speakers. Bob Temple is Deputy Center Director for Clinical Sciences and Acting Deputy Director of the Office of Drug Evaluation at the Center for Drug Evaluation and Research. Bill Capra is Senior Director and Global Head of Oncology for Real-World Data Science at Genentech. And then Rob Metcalf is Vice President, Global Regulatory Affairs, U.S. and Global Medical Quality at Eli Lilly and Company. Building on an expert workshop that we held back in January, this session will begin to lay out a specific piece of real-world evidence regulatory use framework that we've been working on. And so you've heard this morning, we need to get specific, we need to identify the best opportunities. So these next two sessions, this one and then the following session, will begin to ask when. So this session is when do we think that real-world evidence to support regulatory decision would be most appropriate. The session after this will begin to explore how one might go about developing the evidence to fulfill a regulatory use. The specific use case that we're talking about is a new indication for a medical product that's already on the market. And so I'd like to start things off. I think we're going to go in a bit of a different order than what's in your agenda. We're going to start off with Bob. He's got some slides. Then we'll move to Bill, and then we'll round that out with Rob. So, Bob.
Okay, I'm just going to use that.
Okay.
If I can. Yeah. Well, in some ways, I have the easy task. I mean, this isn't using drugs as assigned. This is randomizing within the real-world setting. So the main question is, can you do it? What are the nuances of how to do it? Are the data any good? But the concept is, okay, you're randomizing. So I have the easy task. What are some things to think about? Well, one, and this isn't trivial, is finding potential participants. That's not a small problem these days. A second issue that I'll address briefly is how you get consent under these circumstances. A third is, do the data you have on identifying patients tell you everything you need to know, or do you really have to get into it and have the investigator confirm it all, which is more complicated than just using the data? Can we reliably know that the disease is even present from the data that's collected? And then there's all kinds of enrichment patterns that people use in trials. Can you learn those from the data? This is all about whether you've picked the right patients. You got to consider whether you need to have blinding, and whether there's going to be placebo, whether that can be done in these settings. And then are the electronic health record outcomes going to be enough for you, or do you really want to know much more? They don't collect symptoms very well. So even if you're doing an outcome study in, say, heart failure, you sometimes also want the Minnesota Living with Heart Failure scale. Well, you're not going to get that from the electronic health records. And there's probably a million other issues. So what strikes me as unbelievably simple, straightforward, and very virtuous is helping you find patients. We hear all the time that companies take years to find the patients for their trials. The most obvious possible use of electronic health records is to find the candidates for a trial. I think this should be accompanied by a system-wide urging of patients to join trials because it's good for everybody, but that's a different question. This should be relatively easy for a marketed drug. It has known properties, and you ought to be able to screen in the electronic records for whether the patient has the disease. You ought to know what the current treatment is. You might be able to take a good guess at severity, and you could probably pick up certain kinds of enrichment features like prior events, laboratory findings, at least if they're standard, not if they're exotic, and the duration of illness, all of which might help you decide whether this is a good candidate. Excuse me. I was thinking about where you might want to do this, where it would be useful, and I'm just thinking of candidates. How long should you give bisphosphonates to people to protect their bones? Well, there were large outcome studies with many thousands of people that were done. Wouldn't that be a very good thing to do within a healthcare system? That is, find out people who are on them, and then see if you can randomize to how long to take them. There's a gain. They have side effects. They're not benign. People might like to know how long it's worth taking them. So that's a possibility. Another example, just that I sort of mentioned, is the recent ticagrelor study called Pegasus. Very large study, compared 2 doses and placebo, both added to aspirin to see how long to give an antiplatelet drug. Again, you really need to know the answer, because a benefit could be reduced heart attacks and strokes, but the downside is there's more bleeding if you stay on it. So people have an interest in knowing whether to stay on them or not. And so I think you could conceivably do such a study in a randomized environment of people who are, say, on the drug for a year and want to see how long to take them. Another good candidate. Another possibility is, and this has been done in randomized withdrawal studies, how long should you use an adjuvant chemotherapy? There are consequences to being on adjuvant chemotherapy. You want to know if it lasts after 3 years, lasts after 4 years, and there have been trials like this. Those all seem like great candidates to do in this sort of environment. Now then there's a question of doing the trial. You still need to get consent. There's no way not to do that, which means there has to be, not necessarily, there probably has to be an interaction with an investigator to do a reasonable consent, someone to ask their questions, but it's not out of the question that you could do this online, at least initially online, probably with a follow-up, and that's sort of been tried. Probably, if what you're testing is whether to maintain therapy, you really need investigators to see the patients periodically. So you have to, within this system of real-world evidence, you have to have actual investigators who've agreed to be part of the study. So you still have to get them and enroll them and do all that stuff. Doesn't save you that. Whether the system can collect the endpoints for you, or you need to have patients come in and visit and fill out forms and all that stuff, is going to depend. These systems are not very good, actually, at collecting death. Sweden is, we know, because they have registries and you can use them, but it's not so easy in the US. I'm not sure what to do about that. They probably collect whether a person had a heart attack, but we don't know whether they make the diagnosis correctly or not. And everybody's working very hard on getting good diagnostic criteria for a heart attack. It's not so simple. I don't know whether if you do it on the basis of the evidence collected, it's as good as or as precise as having the investigator and then a judgment committee fill it out. If you're interested in a heart failure treatment, yes, you can get hospitalization for cardiovascular illness. You can probably get that from the system, but you won't get New York Heart Classification, probably. You certainly won't get the Minnesota score. You won't get an exercise test. Maybe you want all that stuff. So even if you use these systems to identify patients, there may be a lot of other stuff you have to collect, and that's going to have to be decided. And there's a whole range of other stuff, depression scores, ADAS-Cog, that is not going to be collected automatically. So you're going to need to have investigators, and people are going to have to visit. For a recognized approved drug, it's often possible to greatly reduce the amount of safety data that needs to be collected. We have a guidance on this, informally referred to as Phase 3-4 Light. But it basically says if you know the drug, the only things we want to know are adverse reactions that kill you or impair you or make you drop out of the study. And you don't have to collect the routine other stuff. Most of the time, you're going to want to blind the treatments. So you have to get into the question of how you do a placebo-controlled trial under these circumstances. There might be some cases where you wouldn't. So the limitations of all this for randomization within such a system, I think used to find people for enrollment, that's great. That's easy. The use of collected data has a lot of issues that need to be worked on. I don't know what that says, impression hides. Anyway, oh, no, it's inaccuracy hides. To the extent that the data that are collected are not precise and not good, they make it harder to show an effect. Everybody knows that. Noise obscures. So if the effect is large and overcomes that, the conclusion is perfectly reasonable. It would be very hard to imagine a persuasive non-inferiority study in this setting because you don't have any prior historical experience with this environment. You couldn't really usually set a non-inferiority margin. The exception to that is where the effect is just enormous. So, it's interesting. The effect of Coumadin and other drugs in reducing thromboembolic stroke in people with atrial fibrillation is something like 70%. So I'm not even sure you couldn't do a non-inferiority study with an effect that large in a setting like this. It would be interesting to, do it. And then a lot depends on how good the registry is. We've all had the recent example out of Sweden of the TASTE trial, thrombus aspiration and myocardial infarction. And it was possible because it was only one treatment. It was just 1 day of treatment. You didn't have to follow. You didn't have to maintain treatment. All-cause mortality in Sweden is easy to ascertain. You could do it. And then they had other registries that collected additional secondary endpoints in a way that I'm not sure we have. But if you have all that stuff, it's pretty plausible. And it's a fascinating example. Maybe our registries will get better. Or maybe where there is a registry already of some particular thing, you can use those data. That's not quite real-world evidence, but it sort of has plausibility to it. Okay, end.
Great, thank you.
Oops, sorry.
Good morning. I'll be putting forth an example which may be the ideal setting for assessing real-world data for regulatory label expansion. First, I want to highlight what we mean by real-world data, as some of the differences in the types of RWD impact how we can potentially use these data for regulatory approval. The strength of the insurance claims databases are the large sample sizes, but as we know, these were collected for and built for insurance purposes, not for clinical research. So the challenge then is to link the claims with outcomes, potentially through a chart review. Health provider claims databases have data completeness as they are closed systems, but the sample sizes are often much smaller. Registries are generally either disease-specific or product-specific, but often the amount of data collected per patient is limited. Electronic medical records link up all records from the general practitioner and specialty clinics, but often the key endpoints are buried within unstructured data, such as physician notes or radiology reports. I mean, there are trends in advancing this. There are trends on improving the data quality and completeness, such as, through technology, such as the ability to link up the insurance claims with the EMR and with molecular information. Now, randomized controlled trials have been the best tool for measuring the efficacy and safety of a medication, and the differences between RCTs and real-world data have been well documented. But in highlighting these differences, we can see an opportunity for real-world data in rare diseases, where the large number of treatment centers provides the ability to identify patients. Now, this will require new tools. The tools around clinical trials have been around for decades, such as case report forms. But many of the real-world data sources are relatively new, so it's going to require new tools for data collection, even for endpoint definition. So in rare diseases, because of the small patient numbers, an RCT is generally not feasible. And even a single-arm clinical trial can take years to enroll because of the limited number and the fixed number of study centers. So rare diseases may be the ideal setting to assess the use of a real-world data for label expansion. So an example where this could be applied is a rare cancer based on both anatomy and biomarker alteration. The setting here is a small population with high mortality or high morbidity and no effective treatment option. The experimental medication would have been previously approved in a larger cancer population so that the safety and benefit risk are well established. Now, it's essential that there's a biological basis for the activity in the rare cancer. For example, an agent that targets a specific biomarker pathway. And there may be existing phase 1 clinical trial data showing safety and efficacy on a very limited number of patients with a rare cancer. And that would provide the basis for a larger confirmatory cohort obtained in a real-world setting. The primary endpoint for this rare cancer example is treatment response in a real-world setting. Now, to overcome the challenge of the unstructured data in an EMR, patient vignettes could be prepared using redacted physician notes and radiology reports. The small sample size in a rare disease population enables a patient-by-patient review to assess the treatment effect. This can lead to an opportunity to define a standard definition of response in a real-world setting, and then that could be used for future applications in the same disease area, extending this beyond just one product. Many of the common issues with real-world data can be addressed in this rare cancer example. The data quality and completeness concern can be overcome through obtaining redacted physician notes and radiology reports. Patient vignettes from a limited sample size remove the need to pull data. In a setting where a single agent results in tumor shrinkage, the need for randomization is alleviated, and patient privacy can be maintained through use of a third parties to de-identify data. In summary, real-world data in a rare disease setting is an opportunity to demonstrate efficacy and safety in a population where a randomized controlled trial is not feasible. The manageable study size enables an in-depth review to confirm clinical activity. And then this could be extended to larger populations where clinical activity may be limited to extending stable disease. So in the oncology setting, overall survival can be obtained from real-world data sources, and then this would suggest a randomized design. In other broad populations, there would be a need to create data standards with well-defined endpoints in the specific diseases. A pragmatic design would then be an option here if there are not available agents with a similar mechanism of action for crossover. Thank you.
Great. Thanks, Bill. Rob?
Thank you, Greg. I think both Bob and Bill have given nice specific examples of where RWE can potentially be used in regulatory context, regulatory decision-making. I'm going to go a bit broader in terms of some of the contextual elements of what we might consider and some of the criteria from that regard. First, starting with a comment that many of the speakers this morning made, that substantial amounts of electronic health data are being generated every day. Given advancements in the quality and the documented validity of these data, along with an improved understanding of appropriate study designs and analytical methods, we are now in a position to consider these new opportunities to incorporate such real-world evidence into a range of regulatory decisions. This use has the potential, I think, to significantly improve the quality and efficiency of evidence development and ultimately drug development. These will help ensure the safe and effective therapies reach patients with the best information available in labeling to guide healthcare professional and patient decision-making at the point of care. The use of real-world evidence is growing albeit in a fragmented and somewhat disconnected manner. Post-marketing safety evaluation of pharmaceutical products, as we heard this morning, in the real-world practice has been performed for many years, becoming a mainstay of regulatory safety assessment with the creation of FDA's Sentinel system. By embracing application of real-world evidence through potential application and evaluation of efficacy, we now have the opportunity to move forward in a more effective and efficient means of drug development. These additional uses have only been considered more recently, but I see multiple potential uses in benefit assessment that are emerging, and each of these uses need clarity as to how they fit into the existing regulatory framework. A focus of our discussion today is on the use of randomized designs in the clinical setting, in other words, pragmatic study approaches, to support approval of new indications for already marketed medical products. While this is a logical area of focus, other potential uses for real-world evidence have been alluded to and could include expanding or restricting approved patient populations, refining dosing and product labeling, confirming efficacy as well as safety in the post-approval setting, and establishing, as we've heard repeatedly, historical controls for clinical trials. Evidence can be garnered from real-world data sources using a multitude of study designs, ranging from pragmatic clinical trials to prospective and retrospective observational studies to case series to case report studies. The possibilities for use of RWE to either augment or in some cases potentially replace traditional data sources are exciting and potentially quite extensive. Experience to date with the use of real-world evidence for benefit assessment has largely been on a case-by-case basis. In a number of these instances, the criteria seem to include the following characteristics: situations where there are limited or no adequate alternative therapeutic options, where the disease state is serious and life-threatening, rare diseases with small patient numbers, and when the drug effect is large and measurable. These early experiences provide a great foundation for the application of real-world evidence approaches in the regulatory context, and we have the opportunity to broaden and make more consistent the situations where real-world evidence is considered as meaningful, supplementary, or alternative data sources to traditional randomized clinical trial approaches. Clarity in terms of regulatory transparency, predictability, and consistency to establish best practices around the regulatory framework for consideration of real-world evidence to inform regulatory decisions will encourage more investment in this emerging field. As a specific use case, there's a growing interest in the utilization of real-world evidence to support approval of line extensions for marketed products. This would ideally be pursued in situations where the clinical safety profile of the product is adequately characterized, the patient population is easily identified in sufficient numbers in real-world data sources, and the outcome of interest are appropriately captured in a real-world setting. Further, medical practice should guide us as to when we should be considering evaluation of these medically accepted alternative uses. Randomized pragmatic trials are increasingly offering the benefit of high external validity through ongoing advancements in data sources, methodologies, and designs, including means to minimize bias that could potentially be created by unmeasured confounding variables. This is an important consideration as we work to establish causality. Causality is certainly an important objective when considering the approval of new uses of previously approved medicines. Causality is assessed using multiple factors, and these factors have been documented to form the basis of modern epidemiological research. In the assessment of causality, one must consider the biological plausibility and temporal relationship between the intervention and the outcome, the reproducibility of the finding in multiple studies, and the strength of evidence and the strength of the effect size and dose response, among other factors. Pragmatic trial designs have the potential to offer an alternative or supplement to randomized controlled studies to assess these factors. Approaches for new indications traditionally require two adequate and well-controlled trials by both regulation and precedent, but both regulation and precedent also allows for approvals based on single trials with supportive evidence. In situations where reliance on real-world evidence is a possibility, consideration could be given as to whether one traditional clinical trial plus evidence gathered from real-world data, such as that created in pragmatic trials, may be adequate to meet the standards. This appears to be a viable and logical place to start as we build a more consistent and transparent regulatory framework for the use of real-world evidence. As I mentioned earlier and has been mentioned previously by others, there are potential multiple other regulatory applications of real-world evidence that we could consider. One of the opportunities that's most exciting to me is the use of real-world evidence for label changes, which could include expansion, restriction, and refinements within already approved indications. As many have already articulated, data from traditional clinical trials, while very important, is collected in somewhat of an artificial environment and a very limited patient population relative to the use of the product in the real world. Data from real-world experiences can help us provide better information to patients that aren't type-matched to the exact type of patients studied in traditional clinical trials. Label changes resulting from evidence collected in a real-world setting will ensure that healthcare professionals and patients are making decisions based on the best information available at the point of care. A third category of potential use that were alluded to earlier in talks would be the establishment of historical controls, which the FDA has a number of precedents around. There are multiple examples in oncology and orphan diseases where traditional approaches to control arms may be very challenging due to the small patient population or the life-threatening nature of the disease. Use of historical controls could substantially augment the power of a study in a very efficient and effective manner. Of course, appropriate attention must be given to avoid bias and confounding factors, but through diligence and matched demographic characteristics, provider skill level, modes of care, technology use, analytical methods, and other factors, one can go a long way to ensuring that results are relative and comparable. I'd like to conclude with a strong push that a coordinated effort is needed to address the potential use of real-world evidence in regulatory decision-making. In this regard, the efforts exemplified by this workshop are critically important. I've described a few, but not all of the many potential applications for real-world evidence. We have access to inordinate amounts of data in the real world and can be converted into real-world evidence. Answers to a variety of questions have the potential to be researched more efficiently in terms of cost, time, and resource utilization while still delivering high-quality, robust information. Greater reliance on RWE has the potential to significantly enhance our approach to drug development and regulatory decision-making. Considerations of pragmatic clinical trials as part of the data package submitted seeking approvals of line extensions is an excellent area of focus. Guidance from FDA is strongly needed in order to form the foundation for the regulatory expectations to encourage investment and to enhance predictability in the use of pragmatic clinical trial designs and other real-world evidence usages, as well as other types of RWE. Our early experience in 21st century science enables us to pursue the great opportunity in front of us. Thanks. I'll end there.
Great. Thanks to all of our speakers. Looking at the presentations that I saw, and Bill, your examples were, a very specific case where real-world evidence could support, a new indication for a product that's been already on the market that has a well-established safety and efficacy profile within that approved use. But your example was, I think suggestive of where you could use real-world evidence in a case where you weren't randomizing, potentially. And Rob, you brought up scenarios where potentially randomization would be important, but then other uses of real-world evidence. And I think outside of a new indication, but potentially other kinds of regulatory decisions. So to Bob and all of you, let's first tackle I think randomization versus not, and maybe narrow it to a new indication and maybe explore that a little bit further on, are there scenarios for a new indication, and what would be the, what are the qualities of those scenarios that where one might say that even randomization may not be necessary? So Bob, do you?
Yeah, it's on. This all turns on how big the effect is. I mean, the use of this is not a new idea. These are historically controlled trials of a particular kind. And nobody really believes historically controlled trials are useful if the effect size is very small. So all of the cardiovascular use claims that I think of, which have made a huge difference in extending survival and all that stuff, it's very hard for me to imagine that a non-controlled trial, non-randomized trial, is going to be credible. The effects of these drugs are like 20%. That's great, that's a lot of lives saved. But without further evidence, I find it implausible that those will be credible with with an uncontrolled trial. But one could look. We have lots of trials. I gave one, recent trial on how long to give an antiplatelet drug. There's another study, the DAPT study, that did the same thing. If someone wants to make the case that a historically controlled trial without randomization could be useful here, let them do it. Find those populations and see what the results are like. With the gain of experience, maybe we'll learn how to adjust the population and stuff like that. But without that, they are, to me, facially non-credible. Now, our experience with the novel anticoagulants is sort of interesting. We've done a lot of things to look at bleeding for various reasons that we're worried about. It's interesting. Very consistently, dabigatran has had fewer intracranial hemorrhagic strokes. And that has shown up repeatedly. It's always better. And that's at least partly because the hazard ratio is 0.5 compared to Coumadin there. It's a large effect. It's an effect of twofold. On other matters, whether there's GI bleeding, the results go all over the place. So how big the effect is has a lot to do with this. If a secondary use is very large, the examples Janet gave, on whether it shrinks the tumor, that's perfectly credible. For other things like that, I think people have to get enough data to convince us that those are plausible. And we're learning more about how to adjust people's risk. Maybe that'll make it possible. But until somebody shows it, preferably by showing that a historically controlled trial in that environment came out the same way as the randomized trial, I find it hard to imagine that that's going to be credible.
Bill?
Yeah, the example I was bringing up was one where there would be tumor shrinkage as a single agent, and I know the agency has approved clinical studies on those type of trials. I think the reason why that I think is a nice example to look at is because the challenges we talked about earlier today was about how do you pull the information out of the real-world data source. It's different than a RESIST criteria where you, in a clinical study where you have very fixed visit schedules, fixed visit assessments. In a real-world setting, it's going to be very much different. How do you do that in a very large randomized study? It's going to be difficult to interpret relative to a small, rare population where you could actually go through the small patient numbers and actually look to see did that individual patient really respond? Because it's going to be a different way of looking at it than perhaps the RESIST criteria.
And tumor shrinkage is one thing. We're usually interested in time to progression. Is that even possible?
Yeah.
I mean, I don't know
Yeah.
Until people look.
Right. And so I think in a small population and looking at that, getting our comfort around that before we can extend it into a larger population where we'd be looking at time to disease progression in a real-world setting, which I agree that's going to be a very difficult one to interpret.
I think Bob alluded to the magnitude of effect. I think the other is it well characterized from a safety standpoint? And is the data there in those real-world data sets to give us confidence around that? Are there other places, too, where you can leverage more of that in terms of new indications in various fields where you potentially look at combinations of new therapeutics that are used out there? Oncology is a prime example of this space. If the data sources are amenable to that, if the practice of medicine has been doing that, are these data sources that can supplement or complement randomized studies in terms of their use patterns and provide through pragmatic approaches or other approaches additional information that we could see sufficient for regulatory decision making? So I think there's a couple of these areas to explore.
One of the points made in the previous panel is that these things may or may not be sufficient to prove something, but they're very good at raising hypotheses. And I think that should be recognized. If one group given a drug seems to do worse, older women or something like that, you want to go looking maybe you want to go back and look at your trial, see if it was there too. But the large amounts of data make possibilities like that very good. Mm-hmm. And if it's important enough, it could suggest that further trials should be done. That's a very valuable use.
So supplemental evidence, specifically in the case where we're looking at a new indication, I'm going
I'm going to sort of steer away from that for a moment and then go back to it because we've got a lot of time on this panel. But it sounds like, at least for now, at least a demonstration example or showing the agency that the sort of conditions are right for potentially not randomizing to show an effect in a new population, as long as the data are well characterized, can be collected, the effect size is really large, perhaps randomization is either impossible or not able. Then there might be unique scenarios where that could be the case. So the departure that I want to take is maybe around some of the other regulatory decisions outside of a new indication. So, maybe a little bit less, so like label changes. So you're still looking in the same population. You're still looking at, but you're looking at potentially different dosing, different duration of use. But the population are the same essentially. And are there scenarios where does that sort of relax it a little bit in some way, where non-randomized design, maybe a well-performed prospective study that includes registries where for the necessary data elements, some of them are prospectively collected by the clinical sites, but do leverage real-world evidence from electronic medical records, lab results, et cetera. So in those scenarios where you relatively have the right population in terms of the label, can you foresee a framework where these real-world evidence designs could actually be useful outside of randomization?
Well, I can see one. It's well known that in clinical trials, compliance is probably better for a variety of reasons, the harassment from the investigator and all that, than it is in the real-world setting. So if we, for one reason or another, approved a dose of X, and you find out in the real world that nobody stays on it, everybody stops it, that could either suggest that you want to say something about that in labeling. I mean, it might be real-world evidence of whether people will tolerate the side effects. That strikes me as something that go in the labeling. Now you'd have to decide whether the lower dose worked, so you'd bring your dose response information to it. But we often approve a higher dose, even if the evidence that it's better than a lower dose is not overwhelming. I mean, you don't have enough data to know precisely whether dose 1 is different from dose 2, so you sort of look at the curve and you make your best guess. But if you discovered that it's very poorly tolerated and people don't take it, I'm not sure that couldn't go in labeling. Can't think of any examples of it, but it doesn't seem out of the question. And that's an area where real-world evidence might trump the clinical trial setting, where the tendency to encourage and stimulate and browbeat people who don't take the drug might give you a distorted idea of whether a dose is tolerable or not. So that's one possible example. I haven't seen it. But.
To me, dosing is a logical place as well. But if you want to span, Greg, what you talked about a little bit, narrow it, keep it in the same population, you might get meaningful information on subpopulations around in the elderly, or combining dosing in the elderly. Because clinical trials won't be able to cover all those scenarios at all those doses as broadly as you do in the real world. If we have data sets, if we have abilities to collect that type of information, to mine that type of information, to think about study methodologies that might pull that out, thinking about populations within populations that might respond differently could be an area of opportunity to look and say where real-world evidence might put us to better ways to use that medication in the marketplace.
Great. So turning then to what this panel's really about, and it's we're trying to focus on sort of randomization in the clinical setting for a new indication. And in our previous expert workshop, I think somebody brought this up. I'm not sure it was Mark Berger or Sean Tunis or maybe even Adrian. That sort of, if you think about a pragmatic clinical trial, it's you have randomization on day 1, and then everything after that is an observational study. So it's not a situation where you have a well-controlled traditional clinical trial and you're bringing in data elements from an electronic medical record. It's actually where you have maybe relaxed controls on sort of switching or adherence to the medication, all of these things that happen in the real world that actually can come in to play in the study after you've randomized. For those kinds of designs, the usefulness for a new indication is the question at hand for this panel. So what we heard in the earlier panel is it would be great, and I think Bill brought this up, if we had a sort of a Latin square or sort of some way to understand more of what would the conditions need to be in order for a pragmatic design to bring beneficial information that would be sufficient for a new indication. So one of the ones that we already talked about is a large effect size. So if there's a large effect size, that would be important. Also, if we were able to collect the relevant endpoints with a high degree of validity and reliability, that would be an important aspect. But what are some others? What are other sort of conditions that you all think, at least if we're the concept of starting slow, identifying some potential pilot projects or demonstration projects? And we have some examples, but I want to push a little bit more on sort of what the conditions need to be that might lend themselves to this sort of design.
Yeah, so when we think about, we've been comparing what the pragmatic design would be compared to an RCT. We've been focusing on the randomization bit. But as you brought up, the other part is the control. Are patients staying on within a control group or not? Are they able to cross over to another effective medication? And I think that's really, one of the conditions that needs to be thought about is there another medication with similar mechanism of action that potentially patients could cross over to? If there's not, and maybe that's a way to leverage the cluster randomization. So if there, in some centers, if that's not a treatment option, potentially they're randomized to those centers versus others where they could be potentially crossing over. I think that's the question.
Okay.
But it's very hard to have this conversation without specific examples. Lots of big outcome trials are pretty large and pretty simple, and they're pretty pragmatic. They don't, we've very strongly discouraged excluding people for this or that. Some of them are less pragmatic because they have enrichment features and stuff like that, which helps you win. So you want to be careful about getting rid of those. Many of the things you're talking about, crossing over, stopping it, not complying, make it very hard to show an effect. So if your goal is to show a new effect and you're allowing all kinds of noise to enter, your chance of winning goes away. It falls. So whether that's an attractive feature because it's simpler, I don't know. But if everybody after a week crosses over to a different drug, and then you don't have a placebo group anymore. So I don't know. I think these things have to be worked out. A lot of people agree that large numbers of exclusions are bad, and we've strongly discouraged those. People used to exclude everybody over 75 from trials, or over 65, and we and the ICH are very much trying to discourage that. So they're pragmatic in that sense. We always try to tell people, don't exclude people who are on other drugs, because then you won't find out whether there are drug-drug interactions. That's silly. So more and more trials, large trials, are getting to be what some people would call pragmatic. But that doesn't mean that noise doesn't obscure. So there's a tension here. And it's not clear that trials will win if they're too noisy. Now, maybe that's real world, but it doesn't help you know whether the drug is of potential value.
So the question, based on that response, I would say, why would a company ever want to do this if there's so much of a potential for this attenuation of the effect? So it's great that we have two members of the industry on the panel. Maybe help us understand, sort of from your own decision making. And Rob, you sort of brought this in a little bit in your comments, but for you all, as you're thinking about designing the right studies, if you were contemplating more pragmatic designs, what factors go into that decision?
Well, I think, in addition to what Bill and Bob have alluded to, I think as we think about it from a regulatory context, is the disease fairly well known? Is it fairly well characterized? Is there good regulatory precedent around it? If you take a well-known disease like diabetes, for example, we know a lot about the history of that and so on, versus a less well characterize disease from historical regulatory standpoint. So to me, as we look at risk around that, do we see the opportunity where we could, as I said, meet the substantive evidence standard, not maybe solely from one source or another, but by supplementing and by using, in the right circumstances, RWE with a well-controlled randomized clinical study. That's sort of the risk-benefit that we'll look at. The other thing that we'll look at is there regulatory precedent around that, and what does the guidance look like out there? So it's a risk-based assessment on a whole host of factors, both scientific, but both regulatory from what's available from the framework and what's the precedent that's out there.
No, your turn.
I was going to say, I think another consideration is also the complexity. So as Bob was talking about, there's more noise entered into a pragmatic design. More noise means larger sample sizes. That's going to make it difficult if the endpoint is very complicated, something like a time to disease progression endpoint, versus a hard overall survival, which may be a little bit easier to do than disease progression.
Bob, did you have a?
Well, there are some areas, I don't know if they're mostly interested to drug companies, but there are some areas that could be studied in a real-world environment that need a look and need better assessment. I mean, if you had to name the greatest health problem in the United States and probably the world, it's failure to use drugs that we know work. The off-therapy rates for antihypertensives are probably still 30 to 40% after a year. People are going to die because of that, We know that. Same thing for staying on your statins, maybe because it's all the noise that people use to object to them, but something like 50% are off them at the end of a year. Well, that doesn't make any sense. And large numbers of people don't use anticoagulants because they're too worried about bleeding. It would be of great interest to see if you could improve retention rates and use rates for some of these major gains. I mean, we're talking about effects that reduce mortality by 20 and 30 and 40% and do it over a long period of time. It's the major gain in the health system, these things. And they're not used. There might be interventions that could help people stay on them. Lottery tickets are my primary example, but I'm sure there are other ways that you could probably do it, and that's worth studying. I don't know if that's of interest to the industry, but it ought to be of interest to payers and other people, unless they'd rather have them stop. I don't know. I don't know what the rules are. But those are areas that have to be done in real-world environments. There's no choice but to do them there, and I think that needs a lot of thought.
Yeah, I think those are great examples for, even for groups like PCORI and the NIH Collaboratory that we talked about before, but sort of these growing opportunities to leverage real-world evidence to bring important questions about value, quality, et cetera. One of the things that we also talked about you guys weren't at the expert, at least Rob, not at the expert workshop, was around this relative cost to doing a pragmatic design. When you think about, because of the noise, the population has to be much larger. That could be a benefit because with real-world evidence, we have the opportunity to generate data and evidence from larger populations. But on the other side, it's really hard if the electronic medical records aren't routinely collecting the information, if medical records across sites aren't able to be sort of efficiently linked, and things like this. So there is some sort of barrier in terms of technical linkage, but also cost. From a company perspective, how are you all sort of viewing that challenge, and how does it play into sort of your strategy and sort of thought process for exploring more pragmatic designs?
So the electronic medical records were built primarily for physician use with patients, not for pharmaceutical research. I mean, the way we're looking at it right now is there's other ways that we can obtain some of that information through potentially third parties who are de-identifying data. But it's really, I think a lot right now is we're using that is for understanding hypothesis generating, as Bob talked about, understanding the disease space, understanding how treatments are being used today. So as we design for how to best design our actually our RCTs.
Yeah, I would say very similar.
Okay. Great. So we have a lot of time left on this panel, but a lot of people in the room, so that's a good thing. And I suspect many of you might have questions about this, particularly the panelists for our next session, who are folks that are already designing and conducting and have experience with these kinds of studies. We're going to hear about how to do that. But any questions from the group to the idea of developing at least a framework for when these designs would be useful and when we might think about approaching them? Oh, thank you. Adrian.
So I was particularly interested in Bob's kind of opening comments about under what conditions this would be important. So certainly the belief that randomization is important, and so that would be key for this. But I guess when you speak to the so-called noise in the system, that's a concern that it would be hard to detect efficacy. I mean, one thing that we often hear is that we don't necessarily know about the heterogeneity of effects or efficacy. And the only way to get to that is have larger sample sizes that would have suitable subgroups to evaluate that more. So would that be a context where, say, a new drug have an expanded indication to promote having a large study, so then and more represent the real world to evaluate different subgroups?
Well, there's no question. I mean, it depends on the area you're talking about. But if you look at labeling and you want to see subgroup analyses, once you leave the cardiovascular area, you won't see them. Maybe you'll see men and women if you're lucky, black and white, but you won't see any of the others. Disease severity and stuff. We don't show these forest plots for anything. So a large enough sample size to be able to detect factors that influence how you do would be attractive. I want to mention one other thing that maybe wasn't clear. If you're just using these systems to discover potential candidates, that's sort of a no-brainer. I mean, there's nothing to lose there. It doesn't harm the study because you're then going to do a study that will have the same endpoints you would have if you were just doing a regular old trial. A question is whether you can collect the data from the real world, from the evidence already collected, but that's a separate question. But just using it to identify patients, if it works as we all hope it would, is really a no-brainer. Everybody should do that. And one reason is to have a trial large enough to be able to look at subgroups. As you know, if you read labels, for most symptomatic conditions, it's hard to identify a whole lot of subgroup analyses. Now, which ones are important remains to be seen. We do demographics all the time, and sometimes we do disease severity, but there could be other things that influence how you do. Your income could, for example, could imply whether you stay on therapy or what kind of healthcare system you're in, or all kinds of things could be influential, and it would be nice to be able to know that. So one attractive feature of having an easier way to find patients is that you can do a trial that's a little larger. Hard to argue against that.
Any other questions from the, yes? What's your name?
Khalid Sarsorj with Genentech. I want to come back to the question of noise in the pragmatic clinical trial design, and also something that Janet touched upon earlier with respect to data capture systems and the data quality. Could we say, is the problem of data, of noise in the data and confounding and bias addressable through more complete and more accurate data infrastructure and data capture, so that we can actually mitigate the challenges associated with that?
Great.
I think it depends upon the type of confounding. I mean, if we're talking about the challenge when patients cross over on day 2 to another medication, no amount of different data capture or different analyses is going to remove that. So I think it depends upon what you were talking about. If we're talking about different data systems and how the data are captured, but patients are remaining on their control therapy, then that's a different question that we can adjust for.
I mean, if your endpoint is hospitalization for heart failure, and in the data, they can't tell whether you entered the hospital for pneumonia, heart failure, a new myocardial infarction, or a stroke, and they get it all mixed up, you're not going to, you're not going to be able to find your effect unless it's, really huge. So there needs to be some precision on this, and we see this all the time. We do these diabetes studies, and the endpoint is either MACE or MACE plus or something like that. For what it's worth, when you use MACE, which is dying or having a heart attack and stroke, which are pretty precise, your numbers are more precise than when you use MACE plus, because MACE plus includes more subjective things. So that's always a concern, how precisely the endpoint can be done. Now, there's a process right now in trying to give better definitions for essentially all cardiovascular outcomes. If those became incorporated into real-world data and stuff, I imagine you could hope to be using those outcomes as endpoints better than you do now. But most studies have a precise definition for what they mean by a heart attack. I don't know how that relates to the definition used in the doctor's office, and if that's important, you're going to lose precision and make more noise and have more difficulty showing a treatment effect.
Yeah, so just to add on to that point is that, a big thing that came up this morning, and even Janet brought it up, is one of the big challenges is the data themselves. And the ability for electronic medical records, claims, et cetera, to collect the endpoints that we really want to study. And there are a lot of challenges, as Bob mentioned, particularly in the claims. You might be able to know a little bit more from the electronic medical record that hospitalization with heart failure, but a lot of energy is being spent on identifying what those challenges are for measuring the outcomes in claims data, coming up with sort of claims-based algorithms. So you might find that just one claim with an ICD-9 code for heart failure and the place of service was the hospital is not sufficient, because you sort of pull a sample of those, go to the medical records, and you find out that most of those patients went into the hospital for something else, and maybe it was history of heart failure or something like that. So you develop more sophisticated algorithms, and folks at Sentinel and individual organizations are spending a lot of energy in developing more sophisticated algorithms to really identify the endpoint that you want. And some of these come up with positive predictive values of over 90%, which is pretty good. But the other challenge is that it's not necessarily the quality of the data, it's that the data just aren't captured. So patient-reported outcomes and things like that. And it's so I would say that it seems like as we continue to improve our ability to utilize these data, as we continue to understand the true validity and reliability of these data, and put investments into enrichment, so being able to link that data to the medical record or patient surveys, or even today, health plans are doing more and more about collecting quality of life information. Large provider systems are collecting quality of life information. So as we get better data, it seems like, combined with an example where there's a large effect size, other things seem straightforward for randomization and more pragmatic design, that these efforts would be worthwhile.
It also seems important to distinguish between what a billing record might show and what some of these others. I mean, I don't think a billing record is the same as what an HMO collects. My bet is the VA has, which is really an HMO, has vastly more data, probably of better quality than a billing record does. Or that, the Sentinel has some HMOs in it, and it has some billing records. One could probably look to see if there's a difference in the quality of data there, or what you can collect.
Rich.
So does the FDA have a regulatory interest in effectiveness? Because I'm trying to make a silk purse out of the problem that you pointed out, Bob, that if you do a pragmatic trial, you're often left with the fact that that the participants will take the meds the way they take their meds. But if we could not show a difference in that circumstance, shouldn't we care? And is there anything in the way the FDA is charged to operate, or that sponsors think about evaluating their products that should play into the regulatory decision-making?
One of my least favorite distinctions is between efficacy and effectiveness, but we'll let that go. I mean, what we're obliged to know is that the drug has the effect it's claimed to have. That's how it's described. It doesn't say efficacy, it doesn't say effectiveness, it doesn't use those terms. If there were something in the patient or in the properties of the drug that really made people not use it, we would of course want to know that. And there are plenty of drugs that make people drop out right away if you don't use them right. So we worry about titrating, you know, the alpha blockers and stuff like that. Nobody would use them if you took full dose right away. So they're all labeled to be titrated up. And so if you want to, that's sort of an effectiveness concern, because nobody will take the drug if you just go on it at full dose right away. And we do worry about that. We worry about when you see an adverse reaction, you want to know if it's dose related, if it occurs early, if it can be mitigated by tapering, by slowing the dose more, or by watching closely for this or that. So we're definitely all worried about that. Probably doesn't make you, if that's a problem, it probably doesn't make you think the drug doesn't work. But it's very important to giving good advice on how to use it. So I would say we're very interested in that. And if studies in which control was less tight, which is sort of what effectiveness studies are defined as, all of a sudden just can't show anything and someone can try to figure out what the reason is, that could be very informative. It doesn't make us think the drug's ineffective, but we would be very interested. Does that answer your question? Or did it evade it?
Adrian?
Yeah, so I have another follow-up question about the outcome ascertainment. So, Bob, you mentioned, you know, the precision was really important for kind of outcomes because of certainly to evaluate efficacy and make sure that it would be more helpful, because that'll be there. But just even in clinical trials where we do event adjudication, as it goes through quality assurance processes where events are recycled, even with the expert panels, that we don't get 100%. That's one thing. Secondly is even when studies have evaluated CEC-determined events versus site-reported, for the most part, the interpretation or inferences don't change. And so if you think about leveraging electronic health records or claims for outcome ascertainment, there probably are certain categories where there's strong incentives to be accurate because of reimbursement practices, and there's some that perhaps less so. So the contrast would be, as you noted, say hospitalization for heart failure or a procedure, that would be highly focused, as opposed to, say, a gout flare, where that may not be captured that well. Does that seem a reasonable interpretation of what you've been talking about in terms of outcome ascertainment? Because that seems a critical aspect here.
No, your comments are right on. Whether adjudication of events is useful is highly debated. I don't really have a position on that. We've accepted trials that use what the investigator said, and you're right. It usually doesn't show a major difference. I'm more worried about not having any definition at all. I mean, even in a trial where you rely on the investigator, there is some definition of what constitutes an MI. I just think you have to know how good the system is to know what it's going to do to your finding. If it's very noisy because it's billing records, it's going to make it smaller. It won't give you the wrong direction or anything like that, but it might make the effect smaller so that you have trouble detecting it. It's just got to be something on your mind. It doesn't mean you can't do it that way. Even noise, if it's just added onto it, won't obliterate an effect if the study's large enough.
Mark?
So I want to suggest where noise can be your friend, not your enemy. And this is where I have seen the biggest opportunity for pragmatic clinical trials. If you have two different therapies that have very different modes of administration and dosing schedules, there could be different preferences by patients to use it. A number of years ago, we were working on an oral medication as an asthma controller, which clearly in kids was preferable because kids didn't want to have to use their inhaler in school. And the question is, if you did a randomized pragmatic trial, could you show that there's a difference in adherence and persistence that translated into difference in outcomes? So I think noise can be your friend when you think that you're going to have a difference in persistence and adherence between you and a comparative medication. And that's a place where I think is a good example where it could be an entry point where a randomized pragmatic trial could be of value. Now, are there many examples like that? I don't know. But clearly, this is not well studied very much. And I think that, Bob, you brought up the point that, looking at persistency of effect, that is another good place that you might want to. You could use a study like this. So I think there are entry points, and it doesn't necessarily have to be that noise is always the enemy. What you want to know is, how is it going to play in the real world? So you could look at patients who are on, have many comorbidities and are taking 6 or 8 medications versus those that are taking only 1 or 2 medications. We know that persistency and adherence changes depending on comorbidities and how many medications people have to take. So I think there is a there, but I think we need to look at noise as our friend, not as our enemy.
By noise, I meant imprecision. You're defining bona fide differences in how people are willing to stay on, maybe because of side effects. That's worth finding out. That's the value of real-world studies where you don't harass people to take the drug and you let them decide. You can find out things about intolerability and things like that that would be very important to know. I think that's good.
Great. Sean?
Yeah, hi, Sean Tunis from the Center for Medical Technology Policy. So I just want to make sure I'm not misunderstanding what I think the converging view of this panel about the potential use of pragmatic clinical trials in kind of a healthcare system setting like collaboratory or PCARNet to support approval of a new indication for an already approved drug, right? So it sounds to me like, and doing it in a way that's maybe 10 to 20% cheaper and faster than would otherwise be required for getting a new indication, because otherwise, why would we care, right? The whole idea here is cheaper, faster, and it still gets you a new indication. And it sounds to me like the answer is that's probably not going to happen. So I probably misunderstood something, but I'm just wondering if, before we go to a break, if the panelists could sort of say, yeah, I don't see a way that's going to happen, or yes, I can see that happening, cheaper, faster, using collaboratory PCARNet or something, and we can get a new indication, or, and under what circumstances is that even feasible or, imaginable in the next 5 years?
Let's be sure we have the definition right. Pragmatic clinical trials to me doesn't mean uncontrolled or observational or historical. It means you're not excluding. I mean, I have that thing where there are 9 factors, but that still includes randomized trials. So my view is that a pragmatic trial that has fewer exclusions and doesn't see the patient as often and all that stuff is a perfectly fine way to get a new claim if you win. I don't think there's any impediment to that at all. And it has some attractivenesses because it's a little closer to how it's going to work in real life. I can understand why people would like that. So am I misunderstanding your question? I mean, pragmatic doesn't mean uncontrolled.
No, I'm thinking of pragmatic as a randomized trial. And as you pointed out, probably it couldn't be pragmatic in the sense of allowing lots of crossovers and all the ways in which your effect would probably be eliminated. So those wouldn't be the domains of the précis wheel or whatever that would be more pragmatic. But I'm still trying to figure out, well, what what would be more pragmatic that would be simpler, cheaper, and still adequate? And, you know, what's an example of that? Because I get it, how it's theoretically attractive, but it just seems like there's so many complications and barriers and hesitancy to use it for regulatory purposes that it's just hard to see when it would work.
Yeah, I'm not sure why you perceive hesitancy about those. We generally have a bias toward including a full range of patients. Don't exclude because of age, don't exclude because of other therapy, don't do those things. I'm not sure there's a bias against pragmatic trials that are still well-controlled trials. I don't have one.
So just, you know, I think what we're hearing from the agency, especially, you know, and this is consistent with what Janet was saying earlier, is that there are potentially good examples where more pragmatic designs that leverage real-world evidence would bring valuable information. I think, Bob, you outlined a few examples that would be sort of ripe for some of these early demonstration cases. I think the barriers, Sean, that you're hearing from the group is that, well, maybe those first couple of demonstration cases may not be 20% cheaper. We're going to hear some examples in the next panel and then throughout the day that, you know, because of the, and this is not a regulatory uncertainty or regulatory barrier, but just simply because of the barriers that we're seeing in the technical ability to bring electronic medical records all together to measure the right thing and to make sure that we're sort of valid and reliable in what we're measuring, having the right linkages with registries and claims, et cetera. There's a whole infrastructure that's not quite, maybe it's not quite prime yet, maybe it is, but the infrastructure to be able to do these studies routinely, efficiently, that's not necessarily there. And we're going to spend some time even tomorrow talking about, okay, if we have some pieces of that infrastructure, but what would it take to get more investment, to get more participation, and to really, as a group, improve this infrastructure? And the reason for doing that, there's many, but one of them is that it would enable more efficient, and it would enable folks to see 20% lower cost and being able to do these studies, but the data are there and the data are of such high quality that we can identify, maybe not all studies, but certain parts of regulatory questions where the data are sufficient to answer these questions. So I think you're right that it seems like this is all theoretical, and right today, maybe the business case isn't there to do this, but this has been, there are examples where this is being done. And in certain scenarios that the business case might be there, but we do need to put a lot more energy into the data infrastructure.
I would have thought that even being able to identify patients to inquire about being in the trial in a more efficient way than we now have would be worth a vast amount of time and money. Am I wrong about that?
Mark?
Bob, we're doing that already. So we are mining data sets, both claims data and EMR data, to look at what are the scenarios if you manage your inclusion-exclusion criteria, what does that do to the target population? Yeah. And there are tools available through Optum, actually, where you can then identify physicians that have large clusters of patients. Okay. And by doing that, we've been able to, on a number of occasions, accelerate the enrollment in clinical trials. We're not the only ones who are doing that. Everybody's doing that. So that's the current state of affairs now.
Well, it seems very attractive, very important, and there's sort of no risk to it. I mean, it doesn't damage the data in any way. Why not?
I think from a company standpoint, there are examples out there where this has been done, where real-world evidence has been used in a regulatory context to help support even initial indications, let alone follow-on indications. I think that the challenge is, how do we work towards a system that makes that more predictable? And Greg, you mentioned a lot of those things. I'd say the other thing is, how do we do some of these test pilots that we talked about a little bit this morning so that we can get a better regulatory understanding, through position papers or through guidance on where these opportunities are seen routinely. Where do we start with that? And we have talked about some criteria around that: large effect size, well-characterized disease states, information databases around endpoints that are very clear. How do we make those more routine? I think it's a multifactorial element of how do we make the data more robust, how do the methodologies continue to evolve. And we see this every day within our company I know you do as well. And then how do we make sure we've got the external onus to be able to put a system in place that makes it more routine? We'll still do this one-off. Trust me, when we see an opportunity, you see an opportunity, we'll do this one-off. I think the goal is how do we make this part of the system of drug development and information delivery to prescribers and to patients, in addition to payers and other constituents. I think that's really what we're trying to accomplish.
Jennifer, I think you.
Jennifer Graff with National Pharmaceutical Council. So one thing that I didn't hear previously was the word adaptive. So where you have an RCT and then you adapt into a real-world evidence type, more pragmatic approach. And one of the elements that comes into mind is infectious disease. You know, we saw this week the HPV vaccine where they looked at longer-term outcomes. You know, you have a certainty of whether or not the exposure occurred, you have randomization. But I'd like to hear, you know, is that a test area to do some of these demonstration projects in some of these short-term diseases like infectious disease, vaccines, et cetera? And how would that play in if it was an adaptive, more pragmatic feature?
So is your question, so the examples where it's already on the market,
It's
right?
already on the market. There's already been that could be one. Would you ever allow more of that real-world evidence to look at those longer-term outcomes? Or for new vaccines that are coming to the market and that might be coming out, would you consider an extension to the primary phase 3 to allow for that more adaptive studies, come back with a label enhancement that when those studies are completed?
Well, I can't quite answer because I'm not sure I understand what it is. So you're saying you start with a randomized trial of a vaccine versus no vaccine?
Could be, yes.
Okay, and then what's the adaptive part? You follow those patients
Follow
in
those
their,
patients out.
in
So
their-
you'd have an interim point that would be your end of phase 3. You would allow that to go out beyond and use real-world evidence data capture for those longer-term
Yeah,
endpoints.
well, not quite like that, but some of the NIH studies of lipid-lowering drugs have had 14, 15, 16-year follow-ups long after the trial was closed. Now, where they got the data, I can't tell you. I think they had the names of the people, but they've done 12-year, 15-year, 20-year follow-ups on some of those, which is not so different from what you're talking about. And, you know, it's still the randomized population. You don't have them all, but it's better than nothing. And I think people have tried to do that.
I guess to build on that, would the agency open to a design where you have a randomized clinical trial, and then based upon a surrogate endpoint, getting accelerated approval, and then the long-term follow-up that's required for that, for the conversion, would be obtained in a real-world setting rather than in another RCT?
But do you mean the same patients?
Potentially the same patients, yeah.
Well, if it was the same patients followed later, you'd worry about retention and all that, but that doesn't seem crazy. That's the randomized trial. That seems pretty good. I guess another question is, if you had, well, I mean, this is a question we always ask. If you have your surrogate marker, would you now accept, if the effect was very large, would you now accept essentially historically controlled data? The answer is, as Janet said, if the effect is huge and you know, you have great data on response and you have, now you're looking for survival, and the effect is very large, that's credible. We've used stuff like that.
Encouraging.
But again, it has to be pretty large.
Great. That takes us to the end of the session. I know we have a couple more questions, but we're going to be tackling these topics throughout the rest of the day. I'd like to thank all of our panelists. We do have lunch provided for you. It will be in the lobby. We'll be eating lunch in here, and then we'll begin back up at exactly 1 o'clock. Thank you.
Your call has been forwarded to an automated voice messaging system. 321948882.
Hello?
Can you hear me?
I can hear you.
All right, sounds good.
Yeah.
All right, cool.
Should I count?
Huh?
Should I count?
Yeah, you can
That
count.
sounds good. 1, 2, 3, 4, 5,
You're good.
6,
You're good.
7, 8. All right.
Yeah, everything's good. Thanks.
I think I didn't change the names. If you want to trash the one you got.
Okay, which one is that? This one?
Master deck, yeah. And then just the new master.
Oops. And are there any videos or audio on this?
Nope.
Cool.
Just make sure that I got the right slides added in. Yep, okay.
So where are we now?
Should be good to go. And we are right here.
Now, is there a master whole slide, like something that?
Yeah, it's this.
It's this.
So, and we put it in between each
Gotcha.
presentation, so hopefully that helps.
It does.
We had some issues with the aspect ratio.
Yeah, I
earlier,
heard.
but
yeah,
I think that should be.
Yeah,
all
he fixed that.
right?
Yeah, because everything.
Yeah,
Yeah, this
'cause
room is set up for. It's all
the
HD.
widescreen,
Yeah,
yeah.
16:9. So.
But other than that, we should be good.
I'll put this up, and then they'll continue from there, right?
Yep.
Cool.
All right. Thanks.
No problem.
says here. I lost my mouse. And Adrian's. And then a hold. And then anything here. Everything look great?
Yeah. There's only about 14 of them, aren't there? So there's a couple got animation on, so I just want to make sure they work.
Oh, yeah.
Slow? It's very slow. Sure it wasn't that slow on my machine, but it'll do. Even slower. Hopefully that's going to work a bit better on the real thing. Well, we'll find out, won't we? Kind of does what it does.
Sometimes, yeah. Does everything else look okay?
It ends up where it's supposed to,
Okay.
so that's all right. Yeah, that's fine.
All right.
Great stuff.
Just reset here.
Make sure it's in the right place.
Yeah. And then you should be all set, and then Mario, the guy that we were just talking to, will be the guy that gets you all mic'd up in a few minutes. So.
All right, good afternoon, everyone. Good afternoon. We're going to start our afternoon session in just a minute, so I'd like to ask everyone to head back to their seats, get whatever you need to get, and we're going to get started.
I don't like these public.
All right, once again, welcome back for our afternoon session. We're going to continue our discussion on regulatory applications of real-world evidence with this focus on randomization, regulatory use cases that involve randomization. You may remember before lunch we talked about some example cases and some of the issues that come up or that would come up in these examples. We talked about issues like the clarity of the intervention being defined, the nature of the outcomes, the ease of verifying the data, and other issues that affect the feasibility of randomization and the value of the evidence emerging from such real-world studies. In this session, we're going to switch gears to focus more on the how, so study methods and data development considerations for the efficient implementation of real-world evidence randomized studies for regulatory purposes. And I'd like to just briefly introduce our speakers. We're going to start with some opening comments from three distinguished speakers, and then we're going to proceed to discussion, similar in format to the sessions this morning. First, we'll be hearing from Alyssa Lavange, the Director of the Office of Biostatistics in the Office of Translational Science at the Center for Drug Evaluation and Research at FDA. Then Adrian Hernandez, the Faculty Associate Director and Director of Health Services and Outcomes Research at the Duke Clinical Research Institute, and Martin Gibson, who's the Chief Executive Officer of Northwest eHealth. Martin, thanks for coming over here from England to join us. Adrian, Alyssa is going to help present some of the issues for this session. Adrian and Martin, I think, are going to talk about some specific examples, again, focusing on the how-to. So moving from the when and the settings and the issues to determine whether randomized real-world evidence development is appropriate to designing these studies in ways that make them effective for regulatory use. Obviously, the studies need to be designed to be sufficient for answering the questions at hand. And again, we're focusing on approvals for new indications. At the same time, be feasible, minimizing the burden for sponsors, researchers, and patients. So we're going to focus on design considerations that fall into four broad categories: study design, patient identification and selection, data collection and quality, and outcomes and endpoints of interest. We're going to draw on some worked examples that illustrate the potential promise and challenges that these studies have in the current clinical study environment and how to overcome those challenges. So, Alyssa, I'd like to turn this discussion over to you.
Okay, thank you. Good afternoon. Thanks for the introduction. I can see my slides, but you can't. So, there we go. Great. My task is to talk about some study design considerations that come up if you're trying to figure out what you can do with real-world evidence, and I'm just going to skip across the top of a few topics that I think are related and try and build on what you've heard already from this morning. So the first was just to make a plug for the use of electronic health records or information in randomized clinical trials, what I call the low-hanging fruit. We've talked about this in similar meetings before, but there have been in use for a while now trials that look like your standard randomized trial, but supplement the data collection with electronic health records. So some things are skipped in the case report form and pulled directly from the electronic record. This might be medical history, could be demographics, concomitant meds, and so forth. And this can be more efficient than extracting and retyping or re-entering the data onto a CRF, and it also can reduce the source data monitoring because the electronic health record is the source in this case. But there are problems in implementing this, and the biggest is that, at least in the US, we use different systems and different data formats, and I just, as a disclaimer, I successfully did this in one healthcare system, but had trouble rolling it out for a patient registry in another area. Although I will say that the system I used has now adopted something more standard, so I think it could be easier. And then we always have to worry about data quality, and you heard a lot this morning about the quality issues that people are concerned with electronic health records. But that being said, I think that this is something that is happening now and will probably happen more in the future. And you'll hear more about this when Martin gives his talk, which is an actual example. I'm talking in the abstract. All right, then the other thing we talked about today is the importance of randomization. So as a statistician, I always find it helpful to remind people why we care about randomization, because we seem to want to hold on to it so strongly. And the fundamental issue with randomization is if I do have a good, sturdy, randomized trial, high quality, the only difference between the groups should be due to the treatment. And that lets me attribute the observed effects back to the treatment itself. Now, there are problems with what can go wrong. First of all, it's easier to do some of these things when you're masked to treatment assignment, and that's not always possible. The study does need to be well controlled, and you have to understand your control group. You have to worry about the delivery of the intervention, and you really have to worry about follow-up. And we know that as soon as you have a problem with missing data or differential dropout, then that attributability, is gone because your comparability is gone. But that being said, it's easier if you can start with randomization than if you can't. One of the criticisms in randomized trials in the traditional sense is that their inference is limited to the clinical setting or the population that's being studied. And while the randomization lets you make inference about treatment effects through comparative analyses, you have to be concerned possibly about how you can extrapolate that to a larger population. So that's where the pragmatic clinical trials come in. Now, before I say too much about pragmatic trials, I want to just talk about a word that I think gets misused some, and that's representativeness. We just had a meeting last week at FDA about representing minority subgroups in our clinical trials and how to make inference about treatment responses in different segments of the population. And I think people use representative to mean several things, so I want to just take a moment to think about this. So we are interested in drawing inference to the population who would take a drug after the drug is approved. That's what we care about at the FDA, right, is the safety and effectiveness. We want good, safe drugs to be out there for people that need them. But representative means something very particular statistically. So if you are in a sample survey setting where you have drawn a probability sample from a target population, then every member of that population has a known non-zero chance of being selected. That's what makes the sample a probability sample. That's the definition. And that means that if you estimate something from the sample, you have a good, solid estimate for that characteristic in the population. So the percent who would be compliant, the percent with a hospitalization, the percent who died or whatever. And the makeup of the sample mimics or mirrors the population. That's what you get. If you do probability sampling in the right way, then your sample looks like your population. So you have the same proportion or relative proportion of your demographic groups, for example. Observational studies are usually thought to be more representative of the population we are interested in, those who would take the treatment. But they may not be fully representative, and that's because we don't draw probability samples for them. So the randomized trial you could think of as probably the least representative in terms of going back to a broader population. Observational studies, especially driven by healthcare records, get you a little bit closer, but you're probably not you may not be as representative as you think. So, you know, just something to consider when you're trying to weigh the pragmatic and randomized trials. Okay, so then back to the pragmatic trials. They are randomized, and so we've got that. They're often not, I think they're most often not masked to treatment assignment, so you worry about that. I think they're thought to generally have a more meaningful comparator, more actual use. In a randomized trial, pre-market in particular, you often choose the actual comparator as something to help show superiority, so it may not be the same, you know, it may be, we worry about that a little bit. And then a pragmatic trial is going to be more diverse. Is it all the way to representative? You know, maybe not, but it would be more diverse in terms of the types of patients you get. However, the diversity, especially in the choice of clinical centers, could impact the study conduct and the data quality. And then the outcomes are usually broader. There's interest in quality of life and cost effectiveness. That's because there's more stakeholders behind these trials. And the only concern there is just to make sure the assessment of what we're worried about in terms of approving for another indication or assessing the risk once the drug's used by a broader population, that those assessments aren't impacted by the other assessments that have been added to the trial you wouldn't have found in a more traditional randomized trial. And then the interventions in a pragmatic trial, you know, they're often adapted to the clinical setting. That's what gives you the real worldness in some sense. Whereas a randomized controlled trial, you're going to be highly standardized. You could have, we talked about this in the last session, there could be some loss of power because of that variability. You could think of it as inducing a little bit of noise. So that's something to think about. The results of pragmatic trials, because you've got this adaptation in some sense, are useful to decision makers, and it is helpful if you collect information about any variability in the delivery that might happen. And then I think, you know, when this happens, it's hard, it may be hard to determine how to attribute the effectiveness that you might observe or the safety risks that you might observe if the delivery of the intervention is changing that much. And by intervention here, just as we talked in the last, it may be more than just the treatment. It may be the treatment plus the compliance plus other treatments or rescue meds, depending on how you define the things that you're comparing. Now, this, to me, a natural follow-on to pragmatic trials is something that I think is getting more and more interest today, and these are sequential multiple assignment randomized trials. These are really thought of as another type of randomized controlled trial, not necessarily pragmatic, but they allow multiple randomization points, multiple clinical decision points about the next treatment. And they're very real world in the sense that the outcome of one treatment assignment decides what the randomization choices are for the next treatment assignment, and there can be multiple of these. So it's like the whole personalized medicine taken into action. I think they have some of the positive aspects of pragmatic trials and more mimicking the real world treatment that really happens. And the nice thing about these is they're very tightly controlled. The decision points and the randomization arms are all specified in advance, so you've got to do a lot of agreement up front. But the statistical analysis is complicated, but thanks to a lot of very smart people who've been working on this for a few years, with the techniques of machine learning, reinforcement learning, Q-learning is one example of that, the analysis is possible. The quandary for us as regulators is, if I get to the end of the trial and I've had multiple treatments, how do I know which treatment to attribute the good outcome to? Because I need to know that if I'm trying to approve a drug. But certainly in a post-market setting, these things make a lot of sense, and that's where we're seeing them now. And then I know this is the topic of the next session, but I just moving along my sequence of study designs, I want to say just a couple of things about external trials. So we do allow for the use of historical controls. Dr. Temple can cite the wording from the rule and regulation. He does this for me quite often. It's usually in the case where randomization to a concurrent control group isn't feasible or isn't ethical, and single-arm studies may be the only choice, such as in an ultra-rare disease or some oncology settings. So I'll make a plug here for the use of the actual historical or external control data. Sometimes we call a trial externally controlled, but we're really just, you know, taking a response rate and treating it like it's the truth. But in fact, if you have patient-level data on an actual control group, then you can adjust for confounding factors if the two groups, you know, don't look comparable. And this is necessary if you really don't know with so much certainty what the response rate would have been if you hadn't treated these patients. Well-controlled, of course, implies a certain level of rigor, and for us that means pre-specification. Simple things like don't pick your control patients after you know what the outcomes were in the treated group. The more you can do up front, the better. And so, continuing with that, the importance of planning can't be overemphasized. It's hard for us to sometimes be able to interpret results when the external controls were selected post hoc. And then the availability of the actual control patients is important, so you can check for comparability to the new patients and adjust for confounding, as I've already said, but also to assess the variability in the estimate of the response rate, and you may realize you don't know it as well as you thought you did. And there are some, Rich Simon is one who's published this, that if you don't have these data, you really have no business doing an external control trial. Now, there's some recent work that's been published recently about using historical or external control to supplement a concurrent control if you're in a setting where the population is limited, and that you can do this to increase your power, avoid randomizing too many patients if you're in a rare disease setting, and possibly even do an interim analysis to check the comparability and adjust afterwards. So that's an exciting opportunity, I think, for leveraging. So I just copied a few selected references on pragmatic trials that you're familiar with, I'm sure, and historical controls going all the way back to the 70s, and mentioning that the CDRH, the devices group, has a good discussion of historical control data in their pivotal trial guidance. And then the SMART trials, if you're interested, these are two of the methodology papers. So just a few design thoughts, and the two examples you're about to see will bring all this together, I think, and show you that a lot of what I think is important is actually possible with real-world data. Thank you.
Okay, so actually hopefully my voice makes it through this. I've actually, over the last few days, wanted to be part of a randomized trial because I'd go to the flu and cold symptom counter looking at all the choices that are there for relieving my sore throat and runny nose and cough, and yet when I look at the label, I actually can't tell which one's better. And so I certainly would want to be part of a randomized trial so the next person could have a faster recovery. So I'm going to actually talk about two examples that will hopefully frame our discussion. But the kind of main context here that we're all saying is that every day there are millions of patients who are walking through the doors of the health systems, and there should be a rationale to learn from those experiences and make sure the next patient is better. And if we could only insert randomization, we'd have even stronger evidence for that. And what I'll highlight is actually examples where we're actually going to do that. Bob nicely formulated kind of key questions that we have to address, and some that are easier and some that are hard. And so one notion is that certainly people are using EHR to facilitate the recruitment, but actually having piles and piles of data for which you know their patients are eligible for trials is potentially necessary, but it's not sufficient. There actually has to be other parts of this in terms of promoting engagement and actually wanting to participate. And so for the example in cardiovascular trials, we easily can see that there are hundreds of thousands, if not millions of patients through our healthcare systems that would be eligible for trials. And so it's not just actually being able to understand that they are there, they're actually there, but can we make things more efficient? The next kind of question would be, well, for just describing who the population is, so the table 1 of understanding where is that population and how is it compared to what we expect in a treating population, can the EHR reliably provide such baseline characteristics? And I think probably a lot of people would argue that that would be yes, that's the case. But there still probably needs to be some validation of that scenario to actually compare what the electronic health record has versus what is manually entered. And actually, what is better quality? What can be done in a more systematic fashion? And does direct extraction from the medical record, is that better than, say, human entry? The last question, which is probably one of the most important ones, is actually the outcomes. Can we have valid outcomes collected and endpoints, and are they reliable? And so really, can the EHR or claims, if you will, find events in a systematic fashion? So right now, what we have is a world where patients come in to a site visit, perhaps every 3 months, perhaps in some other longer-term studies every 6 months, and then we expect a study coordinator in a 10 or 15-minute interview ask if you've had any events, and then also to kind of peruse the electronic health record, which now has become so complicated to see if there are any other events. So is that a good enough system as opposed to doing something that systematically queries electronic health systems and claims to ensure that we're asking the question in the same way, in a highly automated way? And then getting down to the bottom of this is that, ultimately we do need to ensure that results are of high quality. And so I think we have to go back to guiding principles on this. And as I illustrate the case examples, hopefully people can think about it in these principles. Are we enrolling the right patient? Is it representative of the patient population that we'd ultimately treat? Do we actually employ the right intervention? Do they have the right exposure? Did we have the right outcomes? Are they defined correctly? And are we systematically collecting them with completeness? And similarly for important safety outcomes, and then also, are we doing the right thing for the studies? And through all of this, I think one thing that should be recognized is that we have built a system for clinical trials that collects a lot of data that actually is never used. So we wind up collecting data that often is for risk mitigation, but in terms of what's the top-line results that actually becomes the deciding factor of whether a drug or device gets an indication, the top-line results and the efficacy is what is a primary focus as well, and then secondly, the importance of the safety outcomes. So just to go through the couple of examples here. So one is the adaptable trial, which is the first, Procornet's first pragmatic trial. And the context here is that there are 2 doses of aspirin, and there's wide variation in practice for using those 2 doses. And so the goal is actually to look at the 2 doses of aspirin, 81 versus 325, among patients with heart disease, to look at MACE, essentially mortality, MI, and stroke. And then also importantly, look at differences for bleeding. The way the study is going to be conducted is identifying patients with cardiovascular history using the electronic health records by computable phenotype. Those patients will be contacted electronically and then have electronic consent. And then they'll be randomized to 81 versus 325, and then we'll follow them up electronically over about a 2-year period. And the outcomes are similar to other cardiovascular outcomes in terms of what we've looked at before. This just illustrates that one can define a computable phenotype that can leverage electronic health records to identify a generalizable population of people would say this, yes, is patients with heart disease, plus an enrichment factor. And the informed consent meets all the regulations and will ensure that patients understand what participation means, and will have the ability to also use by providing even more context in terms of illustrative purposes such as video of what it will mean for being part of the study. And this is just a mock-up of the screens that patients would go through for doing that. So it's actually a bit different than a 14-page consent that people often get for consent. So, and at the basics, this illustrates how we would e-screen, electronically enroll, and electronically follow up, but also have a systematic fashion so that we collect outcome data not only through the PCORNet common data model, but also systematically through claims, and then importantly collect patient-reported outcome data in terms of their health status and other issues that may be important for taking aspirin, such as bleeding or GI problems. And then finally, because we want to ensure we have complete outcome data, we'll also have a backup system where we can call patients as well. And when you compare it, everything that's being done for a traditional trial is actually being done, but with a different method and working in this universe. So that's an unblinded trial, but I just want to highlight it is possible to do this with a blinded trial. And so this is a study that's being funded by NIH, that's being led by these investigators who have willingly wanted to share this study concept with the community, and it's testing 2 doses of influenza vaccine. So one can imagine that there's a lot of uncertainty around that, and that's true, and that influenza actually has important public health outcomes, and knowing the right dose in terms of cardiovascular endpoints would be very important. And so in this study, it's similar to adaptable, where patients who have acute coronary syndrome or heart failure are randomized to high dose versus standard dose influenza vaccine in a blinded fashion, and then patients would be followed up in 6 to 9 months for primary outcomes of death or cardiopulmonary hospitalization. Importantly, patients will be identified through electronic health records, but most importantly, actually, is we'll use the system to capture outcomes. And the outcomes of cardiopulmonary hospitalizations is pretty well defined in terms of having to have hospitalization and for the cardiac and pulmonary illnesses that are associated in this population. So it can be done in the same manner, and it can also be validated. And similar, you can see that it can use the same systems to do a pragmatic trial, but it's simply changing the intervention and the type of outcomes, but using the same systems. So just in conclusion here, I think there are these opportunities, and it's up to us to define where they are and what are the boundaries for doing these pragmatic trials with EMR, and also making sure we understand the purpose. So it fits in some scenarios, but may not fit in all, and there are a lot of ways to define that. So thanks, and I'll transition over to Martin. Okay. Water.
Good afternoon. I want to explain to start with why these slides are just a dreadful color. And this pink color is actually the corporate color of the city of Salford. And all of the vans and everything that go around the city are this color so that you can spot them. And if you wanted to know where Salford is, there is a map of the UK, and you can see approximately where it is. We're actually running a late phase pragmatic trial in Salford and the surrounding areas, which includes around about 7,200 people, and they're monitored in pretty close to real time for both safety and outcomes using the linked electronic record system. And the results of the first study will be coming out later this year. Actually, 2 studies. There's one in COPD, which is the first one, and then that's a 2,800 patient study. And then there's a 4,400 patient study, which is the asthma study, which is still ongoing. And to put this in context, we're doing this, this is a GSK-sponsored trial, and I think probably most people in this room are aware of this, but do RCTs represent the real world? And the answer is, of course, no. If you look at the left-hand side of this bar chart, you can see around about 100% of people that would be eventually prescribed a drug, in this case for COPD. And then you can look at the bars decreasing in size as you take out the things that are most common in COPD trials. And you end up in a normal kind of phase 3 COPD trial with around about 1.7% of people that would ultimately be prescribed the drug actually getting it in the trial situation. So another aspect is, what about adherence to medication in these kinds of respiratory studies? On the left-hand side of this chart, you can see what happens in a normal randomized controlled trial. We basically really strongly get people to take their medication, and in fact, there's usually an enrollment phase where if you don't have 80% of your medication in that phase, you're not going to get into the study at all. So on the left-hand side, you can see the 3 which are set at 80%. Then there's 2 more after that. One seems to have managed over 120%, which is interesting. And then that contrasts with what happens in a real-world environment, where you can see in the studies that have been reported, people basically don't take their medication. You've got to take that into account if you want to do an effectiveness study, which is what we're trying to do. So the ambition of GSK and ourselves in doing this Salford lung study was to make it as near real world as we could. But we were doing that with a pre-licensed medication at the time. So it also had to be acceptable from a regulatory perspective. So we wanted to embrace the heterogeneity of the population, normalize the patient experience as much as possible, and provide usual care in each arm, with relevant endpoints being collected. But intentionally, we wanted to maintain scientific rigor. So it is interventional, it is randomized, it is controlled. And so on the left-hand side of this slide, down the left-hand panel, you've got what the challenges were to doing this, and down the right-hand side, you've got some of our solutions to that. How do we recruit patients? So we need all comers in this trial, for all the reasons that Lisa was talking about, as many as possible. Broad inclusion criteria. We want the results to be applicable to the whole population. Pragmatic diagnostic criteria. So if your GP says you have COPD, you have COPD. And very few exclusions. So the solution to that over on the right side is to recruit patients through their primary care provider. So every GP in our area has to be the principal investigator for their site. And you'll see how many sites that is in a moment. The next bullet point down is, how do you ensure normal care for patients during the study with minimal study procedures, normal prescribing, and normal dispensing? And I should point out that normal prescribing in this situation is your GP prescribes your asthma or COPD medication, and you pick it up from your local retail pharmacy just down the road. So the solution to that was that we had to get the study drug to be accessed through the local high street pharmacy. And you'll see how many of those we had to get on board in a moment. No additional review, and no change to care as usual. And the final point on this slide is, how do you monitor patients to regulatory standards without carrying out frequent reviews? How do you minimize the Hawthorne effect? How do you ensure patient safety, which is vital? And how do you ensure that we collect the endpoints we need? And as everybody knows, because that's what we're here for, we do that through the integrated electronic patient record. And that means that we actually capture things in real time. And you'll see some of the benefits of that in a minute. So here's a study outline for the COPD trial. The asthma one is not that dissimilar. 2,800 patients, you can see in the left-hand little box. And those are very open criteria for a COPD trial. You'll notice also that you're randomized, and either to new treatment or existing treatment. And then we have, I know this is a little bit confusing because there are really only 2 visits, and they are labeled visit 2 and visit 6. But take it from me, there really are only 2 visits. The first one is actually consent, then you have visit 2, where we collect some information, study specific, and the same at the end, which is visit 6. The 3, 4, and 5 for information are, if the patient hasn't seen anyone in the NHS, the National Health Service, during the 12-month period of the trial, we give them a phone call, and that was the regulator asked us to do that. And that means that all of the rest of it, all of the outcomes and all of the safety is done by the electronic systems. You may want to just scribble this down. This is the simplified schema showing the linkage between the different data systems that we have. And you won't be able to read it, but basically the ones down the left-hand side are national systems that tell us whether people have changed their name, moved, died, whatever. The next ones across are actually local systems where we get data from primary care records, out-of-hours services. Along the top, you have more study-specific pathology data. Down the right-hand side, the two different hospitals that are now involved in the study. And those data flows are happening pretty much in real time. This is a slightly more understandable version of what you've just seen, which is that all of the data flow in from the left in electronic form. They're put into a linked database system in the middle, and then there are outflows from that for different things. So for safety monitoring, for regulatory monitoring, for the actual study analysis, and for the management purposes. Just to give you a bit of scale, I mentioned this before, so there are 88 primary care sites in the study, so that's 88 principal investigators. Most of those people were completely naive to doing a late phase 3 trial, which is what this is. So we had to train them all. That means that over 3,000 people in the end were trained to GCP standards. 128 community pharmacies participate. They're all in competition with each other normally, so we had to bring them together. We had to learn how to extract the data from their diverse electronic systems and bring them together into the study. And the rest is as it says. This is a map of the Manchester area, and this is just to show you that it's quite big. That's about 30 miles across that map that you're looking at. And you'll see there are little dots on it. Those are all the different pharmacies. So we have to go out and we have to recruit them all. We have to get all the data from them. They're all in different chains. One thing that we are particularly pleased with in this study, and this is, I think, important to how we take this kind of technology forward, is the safety monitoring. I do a lot of industry-sponsored trials. And as an example, I might see a patient on a visit today, and they might tell me about something that happened 3 weeks ago. Maybe they were admitted to hospital with a myocardial infarction. They're fine, but that's the first I know about it. I then have to report that with the usual, within the usual timeframes. But the event was 3 weeks ago. With this, we get the events in almost real time. So a study nurse will, if you take it from the top left, a study nurse will tag the patient in the MR, say that patient is admitted to through the emergency room. There'll be a pretty much instant note through to the independent safety monitoring team, who can then track that patient in real time through the hospital and see exactly how that event is evolving. They can do an initial unlocked SAE submission to the sponsor. We can then process all of the data over that next period of time and send a link through to the PI, who can then establish causality and severity, and then that gets sent through to the sponsor and then subsequently the regulator. This is very, very good. In fact, we can talk about this later, it's almost too good. So how has this influenced our thinking? So here's a kind of slide that you'll have seen a lot of, which is on the top I've put some arbitrary numbers about no years down to out to 16 years from original concept of a drug through to getting it used in regular care. And then at the top, I've put your health technology agencies. We have in the UK something called NICE, which you'll be familiar with probably. And on the bottom, the regulatory agencies, and you can see the different phases there. So on the left-hand side, you have efficacy, on the right side, effectiveness. And interestingly, the Salford lung studies sit right on the middle bit of that. So we've now got experience, which is really fortuitous, because that means we've got experience of a late phase 3 trial, but also how you use it for effectiveness. And I think there was some talk about that this morning. And we now have confidence that we can build this kind of technology earlier. So you could use this from phase 2 onwards. But you need some additional bits to be able to do that. And so we have also designed to work alongside this a series of other tools which allow us to do rapid protocol design and feasibility. Those tools also enable recruitment. And then subsequently, after you've done the trial, because of what we've built with the lung study, we can do long-term pharmacovigilance and safety monitoring and look at cost effectiveness. So if that's now, what does the future look like? And this is a real-world presentation. So I don't think this animation is going to work very well, having seen the last one. But we'll just see. It should all sort of sort itself out so that in the future, maybe, if anything's going to move on this slide, we actually start to overlap efficacy and effectiveness, and ideally we can then shrink the whole timeline, which is, I think, what everybody would like us to do. And that gives us the cost saving and the benefits, and patients get the drugs sooner. So what have we learned from all this? The Salford Lung Study, or Salford Lung Studies, are the first of their type. They maintain scientific rigor, they're randomized, there's an active control, and there's a robust primary endpoint. It's actually a hybrid of an RCT and real world, and I think that's important to bear in mind because I think that's a really good way of looking at these things. We initially built a whole ECRF with this. It does have an ECRF, but you can flex it. You can flex the safety monitoring as well. That means that we've got a really flexible trial design system based on the stage of a medicine's development. So you can have something that looks a little bit like a standard RCT, and you could run it with this, right through to something that's much more like an observational study. And I think one thing it has taught us is that we're going to get an awful lot of information on how to conduct real-world studies, and probably a whole load of information on how not to conduct real-world studies. Thanks for listening.
All right, thanks very much for all the presentations. Thank you, Martin. I'd like to ask our panelists to come up on stage, and we're going to turn to the discussion segment of this session. And certainly a lot to discuss as we move from the right circumstances in which to do real-world evidence studies, particularly randomized studies, to the mechanics of how to actually do it. And I think in both of these examples that we walked through, Lissa, they paid a lot of attention to the issues that you had thought were important in study design. I might start with, remember we talked at the beginning of this session about a number of key issues like study design, patient identification, data collection, and outcomes tracking. Let me start with randomization. How hard was this in the applications that you all have set up? And how important did you think about any other ways of any other levels of randomization, cluster, other, or is that just too central to the mechanics of answering the question at hand?
Right. So these studies are in the process of launching, and so there's been important aspects towards ensuring that we randomize appropriately. And actually, in the context here, we actually did think about cluster randomization. So both for the aspirin study or the influenza study, you could certainly think about how you could randomize practices, for instance. But because of different aspects, people really thought that patient-level randomization was necessary for the questions we're addressing. Other examples, though, the collaboratories where we do a lot of cluster randomization at practice level or hospital levels.
And I'm slightly glossing over the fact that we did quite a lot of work before we started on this study. And one of the things we looked at was in what you might call a real-world data setting. So we actually took all of the data for patients with COPD exacerbations for several years before the start of the study, and we modeled what it would look like and how many numbers we would need, what power we would need. And that actually would quite quickly rule out a cluster-based randomization for a study like this. So we wanted to do a patient-level randomization, but there were certain other things we needed too. We needed to make sure that pretty much everybody could get into this trial for all the reasons that Lisa was talking about earlier. So we wanted to make sure that everybody had the possibility of doing that. So cluster randomization would have given us a problem because practices are different sizes and have different capabilities to run the trial. They might have several nurses working for them, no nurses working for them, and so on. So we wanted to make it so that everybody could get in, everybody could have access. So we went for a patient-level randomization based on power that we knew from previous exacerbations over the previous few years.
And in these examples, at least, it sounds like, I guess, Adrian, you're still in process, but it sounds like the randomization mechanics are working out okay in terms of identifying the patients, getting them assigned, getting the informed consent done, and getting them assigned to treatment.
Right, and I'd say the example of the influenza vaccine, that would certainly lend itself to doing cluster randomization, because you can imagine different practices getting randomized to one dose or another. There are other aspects of that study that people wanted to do patient-level randomization, but that had to do with understanding more the immunology. But that's a perfect example where that you could do a cluster randomization, collect the same outcomes, and the fidelity of the exposure would be very high in that setting. And whereas the aspirin study, the concern is about the fidelity of the exposure.
Before we move on to some of the other issues, Lissa, any other comments that you've got about study design concerns here? Do you feel optimistic about where we're headed based on these examples?
I do. I mean, I think the examples are impressive. And, just one comment about the cluster randomization. I think the Center for Biologics, my counterpart, Estelle Russett-Cohen, is in the audience there, says that they do, I don't know how often, but they have taken cluster randomization. And the other setting we've seen we've seen recently is emergency room settings where there's logistical advantages to having everyone coming into this care setting receive the test treatment, and everyone coming into that receive the other, and then doing a randomization. And if you can have a large enough sample of practice sites, then you can control, there will be differences, but there'll be differences regardless in terms of practice sites. And in some ways, randomizing that out can help you. The power loss, it depends on the outcome, because you would still be treating the patient, not treating the site, and you would be measuring the patient and analyzing at the patient level, but you'll have a cluster effect. So your effective sample size will be something between the number of patients and the number of sites, but it won't be so severe as to just be the number of sites. And I think we've talked about this in the setting of antibiotic trials where people won't, the patient groups tell us they don't want to be randomized. They're nervous about if they have a terrible or bacterial infection to agree to be randomized. But if you've randomized the sites, then you come in and consent them and tell them if you agree to be in this study, you will get X, because you'll know that site has the new treatment or doesn't, and that there's some idea that might help, because I think it's been used with antivirals in some cases, and it helps. So it's something to consider, and it doesn't have to be a big power hit, but there needs to be a reason for it. And in the Salford case, I don't think there was a reason for it. You didn't need to go that route.
Now, both of you emphasize in your slides sort of the funnel equivalence of typical, carefully controlled randomized, traditional randomized studies having narrower patient populations as opposed to something that's more representative, as Lissa noted in her opening remarks, of the population that was likely to be treated. I guess that's good. I guess the flip side of that is keeps coming up in a lot of the work that PCORI's doing, is that, the point of having a broader population is not to get a more precise estimate of an average effect in that whole population, but to be able to look at potential differences across subpopulations. And how does that play? How has that been playing out in your experiences? Are the data rich enough and enough data there to support real subgroup analyses?
Yeah, so I mean, just using, say, adaptable as an example. So we can immediately understand, how well our sample will be relative to the overall population, because we'll have information from all the health systems for that. And so just among 5 of these networks that represent about 25 health systems, there's close to 1 million people who are eligible for adaptable. And so you can have not only kind of good representation for demographics, but actually racial and ethnic minorities. You can also look at special areas of interest in terms of kidney disease, et cetera, diabetes, and the severity of that. And so it allows, hopefully, a broader base population with the comorbidities that we encounter in healthcare routinely.
And you're seeing high enough study participation to enable those subgroups to be.
Well, that's always the goal, so that would be the goal. So.
This is a difficult one for me to answer because the actual analysis is ongoing at the moment, so I don't want to query the pitch too much. But we, as you can see from the last slide I left up, there's a lot of data here, and there will be plenty of opportunity to do secondary analyses. But I mean, I think the question from our perspective is how generalizable will the findings be? Because we've had a really good response rate. 50–60% of people that have the condition are actually in the trial. But then that still means there's quite a big percentage that are not. This is the only study I know where we'll actually know what happens to them as well, because we collect that information. So we'll be able to see whether participants in the study do as well as people who didn't wish to be in the study. We will know that. We're also doing a separate study with CPRD in the UK, which is the Clinical Practice Research Data Link, which will actually compare the effect in our site compared with lots of other sites across the UK. So we'll get a good idea of whether actually putting this study into Salford has changed behavior of practitioners or not.
So you're going to have an estimate of the famous study effect.
Yes.
I mean, one thing I will note is, the contrast between adaptable and say invested is that for adaptable, we're aiming for 95% enrollment electronically. So patients who get contacted electronically and enroll electronically. And I'm sure there are going to be differences between that method compared to what people would do in terms of, say, a direct study coordinator, for instance. But we'll be able to understand on that whether there are differences or not. But if you gain efficiencies that way by being able to do larger studies, more efficiently or cheaper, then you could answer many more questions. So there's always going to be that trade-off.
And we've talked some about identification of patients and representativeness, talked some about study design issues. Wanted to turn to data collection. In both of these examples, using a combination of electronic records and maybe administrative claims and other data sources. I can't reproduce your whole chart, Martin, but a lot of information coming together there. Any broad comments about how big challenges in using this data? Are they sufficient for capturing the variables that you were interested in? Any other challenges in selective data loss that you're concerned about?
I think the two things, at least that are important in the U.S., is that we do have health systems that are not fully integrated, and so understanding the complete outcome is important. So at least the way we've approached it is actually kind of a tiered approach so that we can ensure that we collect the encounters that do hit the health systems that are part of PCORNet, but not just solely relying on that, because the absence of an event doesn't mean it didn't happen elsewhere. And so being able to link to other data such as Medicare claims, and then also contacting the patients directly, electronically or by phone, to ensure we haven't missed any outcomes. So we have 3 layers that we go through to have complete outcome data. And then on the definition side, again, for certain things it can be very good, but say if our outcome was acute flare for gout, that may or may not be able to be as good as, say, hospitalization for heart failure and hospitalization for MI.
My answer is a little bit anecdotal. When we first started the study, we thought we might need a whole ECRF to run it. And we soon realized that was a really bad idea, because that meant a huge army of nurses transcribing information that was actually already in the electronic patient record. So we pretty quickly stopped doing that, or did a lot less of it anyway. There's still some ECRF-related information. But it gave us a little bit of an opportunity to look at how much information were we getting from an ECRF and how much information were we getting from source data. And what we discovered was we were getting far more information from the source data than we were from the ECRF. So I think we can be confident that source is better.
And sorry.
No, go ahead.
I'm not the moderator.
No, but I was going to actually turn to you next. I wanted, Adrian, you had a similar comment about data that you were collecting from source versus what do you call it, the 15-minute case interviews.
I mean, so I actually didn't show the picture here, but if you take a picture of a study coordinator now looking at, say, the electronic health record that has all sorts of notes there, and especially if a patient's been in and out of the healthcare system, it's very easy to miss things, and then also recall can also be a problem. And so we're hopeful that a more systematic way of capturing and actually using the data system to query it will actually be more complete.
So after the experience, what do you do to try and come up with some assessment of the quality? I mean, you've got CRF data, you've got EHR data, you've got people looking at both because you determined that you didn't have to transcribe everything. It was better just to take the E. So then what does that reduce the monitoring to? Because I know with my limited experience, we had trouble coming up with anything to monitor since we were collecting the source.
I can't remember the numbers offhand, but we started off with an awful lot of CRAs going out and doing a lot of checking, and we've got a lot less of them now. So,
a kind of a qualitative
yeah.
assessment.
I mean, I think our approach is that for the outcomes, we're going to be validating them. So again, like the definitions and the algorithms that we use to collect those endpoints. Do some validation work in terms of abstracting the medical record or having actually a human look through and seeing does this actually fit. So it's not too different in a way than what one would do for event adjudication, except again it's using sampling to identify any quality issues or reassure that they're quality. And if there is, and we're actually going through that just as kind of the identification process, using computable phenotypes, and then say for the first 100, making sure that people understand, like, yes, this person does have the eligibility criteria intended to approach.
So just to comment that you mentioned the word validation because we've spent an awful lot of money doing validation on all of this so that we know that if we take data from a particular source and move it across here and amalgamate it with this, that is exactly what we've done. We've done a huge amount of testing on that. And similarly, the safety algorithms and triggering systems that we use, again, we've validated every single one of those. And that's probably cost more in terms of the development work than any other single bit of the work.
In your source data, did you mostly pick up fields that were kind of discrete, or did you do any text mining or any, with all the notes that are available, did you get into any of that kind of data extraction? Either one of you.
So no, we haven't done text mining. We've mainly used coded data from the system and actually what I might call hard data, numbers from pathology results and so on. However, when it comes to the safety monitoring, we can access all of that information if we wish to, which is an added bonus.
So our plans there is that we're really focused on the key primary outcomes and key safety outcomes. And so in that context, as part of the validation work, we'll be looking at it, records manually for a sampling, but not doing it in any way for the scale, because it's just not feasible. And again, for the purpose here, everyone should recognize whether it's a clinical trial done traditionally or not, there's confidence intervals between all the information we collect.
Now, Alyssa, you did comment in your opening statements about the importance of data completeness and selective missing data, and how FDA has a lot more experience with the sort of the case report approaches to clinical trial data reporting as opposed to this. What's your sense about have you seen many studies come in like these, and what's your sense about the validity of what you are seeing and what you're hearing about in these practical trials?
So my sense, this is conjecture, really.
Yeah.
But my sense is that with the larger pragmatic trials, it might actually be easier because you've got the mechanism set up to go after simple outcomes, even if you've lost a patient, like vital status or something, because you're in the system, you've got access to the health records, and you may, maybe it's easier with you because you've got all kinds of data on people you don't even study, whereas we don't usually have that, but we could in a healthcare system. I mean, the missing data problem is everywhere, and it can, break all of the wonderful things that we get out of randomization and the other trial design aspects that we rely on. But if you've got a system set up where you can access people, periodically, and I don't know, with phone calls or EHR scanning or something, get any kind of information about their status, then that helps, certainly. You may not, and of course, you have fewer visits anyway, visit 2 and 6. I love the idea of calling your second visit number 6. So, there's less things to be non-compliant with in a rudimentary sense. But it's still a problem.
Well, hopefully one that's getting better as the experience accumulates and the systems for collecting the data become more robust, which actually brings me to the last question I want to ask before opening this up to some broader discussion, and that's about sustainability. I don't know if either of you all are in a position where you could summarize what sort of the cost of implementing the study has been, each of your studies has been. One would expect that to be a somewhat large number given all of the initial work that you had to do. There haven't been, at least for some of these systems, in your case, Adrian, these health systems haven't participated in a study quite like this. And Martin, I think you described your work as somewhere between, was it truly real world and a controlled trial, and that you kind of this augmented structure on top of it. The hope, I guess, would be that between some significant costs related to validation, as Martin was describing, and some significant costs related to just getting this core infrastructure in place to be able to put data together, enable investigators and systems to be put in place that could conduct these analyses, that the cost is going to come down significantly. What's your sense about how far we're getting to sustainability of these kinds of models?
In the setting up the CoreNet, there were significant funds to establish an infrastructure, and so now that's being transitioned to actually implementation, execution of the studies. And so, after that, the incremental costs for adaptables estimate $850 a patient. So that's in contrast to, say,
that's pretty good.
Yeah, so in contrast to, say, another example, NIH trial that was coordinated at DCRI called PROMISE, which is about $3,500 a patient. That was 10,000 patients and considered very pragmatic. And compared to another type of trial that was less pragmatic, that's $13,000 a patient. So really are trying to change the scale of efficiency, but it does rely on kind of the systems that have been the investment of getting electronic health data mapped to a common data model, the engagement of patients, clinicians, and health systems together.
So, I don't know how much this has cost, and I don't work for GSK. But I'm reliably informed by the people that do work for GSK that this has cost no more than it would for a phase 3 trial of this type. However, we've done some stuff that we only need to do once. So the validation, the actual algorithms and everything else that we've built, the data linkage systems, which actually would be applicable across the whole of the UK NHS because they're the same, means that we can scale it. And indeed, we have scaled it already because we started with just Salford, we've doubled the catchment area, brought in new hospitals and new practices. So this particular example, I guess not that cheap, but most of that development work doesn't need to be done again. So it will be cheaper and it will be scalable.
Yeah, no, I would consider it the capital investment, if you will, that as more and more studies are done, then the return of investment comes through. And the final concept is that right now, we have things that are just, we do a study, we break it down, and we do another trial, even if it could be the same population and same outcomes. So the ability for the network, for instance, to implement, say, the next trial like Invested should be easier based on the experience from Adaptable. And so having systems that can be reusable and tools that are reusable are key.
Now, you've got 235 million rows of data, so if you look at the cost per data element, it might be a bargain.
Right.
Yeah, that's
Probably
actually out of date
very
now.
favorable number, a good one.
That's a good way to think about it.
Yeah. You know, I'd like to open this discussion up to the rest of you who are participating in the conference today. So if you do have any questions or comments, please raise your hand and we'll get to you. Yes, please go ahead.
I have a question for Adrian about the vaccine study, which was blinded. How did you actually capture vaccine information in the EHR since it was blinded?
Yeah, so this study is being launched, and so this is a hybrid, if you will. So like there's some elements that we have to retain, say, from a traditional trial, and again, because of the interest in keeping the vaccine blinded, so there's not any, there may be some concerns about patients wanting the high dose, for instance, as opposed to the standard dose, that was felt to maintain the blind. And so for that part, the drug allocation, that'll be done similar to what a traditional trial is, and that information will be available for in the actual trial database that augments the records.
Okay. Other questions? Yeah, Sally.
Oh, thank you. Good presentations. I really enjoyed them. I'm Sally from Patients Like Me. I'm truly interested in thinking a little bit more about the real-time notion. Right now, the real-time capture, for example, I think in the adaptable study is when someone ends up in a system environment that becomes real-time understanding of something that may have occurred. I'm wondering whether you have a tolerance for or interest in expanding real-time to be what the patient's experiencing outside of your gaze, so that they're still part of the study, but they're gathering or contributing some information on their own to also give you insights into things that may not end them up in the system, but may still be of importance, of interest.
Yeah, yes, definitely. So we have a two-pronged approach. So while I was emphasizing the encounters that happen in the health system through the adaptable patient portal, we'll actually be reaching out to the patients periodically to capture their experiences. Likewise, if they have something, they can go into the portal to report their experiences. But in there, we're actually randomizing the follow-up in the portal. So 3 versus 6 months, because we actually don't know what's the right touch points to optimize patient engagement or participant engagement and collection information. Some people feel like if I get pinged every month, that's too much, and then if you get pinged every 6 months, that's not enough. And so we're actually going to randomize.
And yes is the short answer. We really want to do that. And from our point of view, that is another data feed that we want to include. We already do that in some of our direct care provisions. So, for example, all of our renal patients are part of a renal view system that allows them to contribute to their information, see all of the data themselves. Similarly, we're rolling that out for diabetes. But we are very, very, very interested in getting that information directly into the source data. It will become source data.
As you move forward on efforts like that, what kind of concerns do you have about the selectively missing data that Alyssa was talking about earlier when applied to now data that's perhaps selectively coming in from patients?
Yeah, so we were actually wrestling with this because originally some people thought, well, why not just get patient information and that be the gold standard in terms of as they report it through the portal. But I think because of the concerns about the validity and the completeness and any kind of recall issues, that would be problematic. And so we have a tiered approach in terms of, if we see it, say, in the health system, that's very clear, it counts. If we have it reported through the portal, we also want to ensure we validate it by looking at the health system, or if it wasn't at the health system that was part of PCORNet, to find that information.
So nothing coming from the portal was prompted. It's just...
No, we'll prompt,
are
we'll
prompting.
ask, because we are also looking
the
to
different
make
time.
sure that we can capture out-of-system events.
Right,
And
right.
so, just like a study coordinator asked, did you get hospitalized any time in the last 3 months? We'll be doing the same thing, but through the portal or through the call center. And if someone reports something, we'll validate it, that we capture that in the coordinate common data model. If we don't have it there, then we'll go and say, ensure where was it, and ensure that we have that record from that health system.
Sally, do you have a follow-up?
Just one quick follow-up. Thank you. I appreciate that. And one last thing, I was also wondering whether you have any mechanism for giving information back to the participants with some regularity, and what that looks like when you do.
Yeah, so that kind of community, creating the adaptable community is really key. And so we actually have, they name themselves called the adapters. So these are patient representatives that have basically informed everything that we've done, including the logo and so forth. And also we'll be able to send information back to that community about what's the latest about heart disease and what have you. And then at the end of the day, once we know the answer, then we'll be able to transmit that answer to the participants, as well as potentially eligible patients who were similar to the adaptable population. So that is very, very much on our mind.
And that is a really interesting question, because it depends on what you're trying to find out. What we're trying to find out with this particular series of studies is whether people would adhere with their medication more or less, depending on whether it's an easy-to-use medication or not. So we deliberately do not want them to know they're in a trial. So we spend no time whatsoever reminding them that they're in a study or asking them to contribute to it. However, the system is set up so that you could run a study exactly like that, and you could have far more interaction. It really depends on what you're trying to do.
And do you have those same kind of concerns, Adrian?
For us, because we want to we need to have engagement of the participants to ensure that we have complete follow-up and adherence to the therapy. That's or adherence to the 325 or 81. Like, that's an important aspect, and so we want to have that engagement. So it's a different setting or different context that we're aiming for, and so from that lens, that's where our adapters group has defined how we do certain things, and actually even the web portal and the consent process was designed with potentially eligible patients, and we modified. We actually thought we were pretty smart. We totally were wrong, and so we went through a number of modifications based on the responses from the adapters.
Now, aside from getting it sounds like some really good, practical, useful advice on how to design the reporting, the web, and the portal, and so forth, you're not going to have results to tell them for a while. So,
Right.
What are you telling them now?
Yeah, so I mean, we will periodically tell them information or new information about cardiovascular health, and so again, it'll be a learning community, and so while we won't know the specific answer for the question, we still want them to understand that if there's something that new comes on, we should make sure they know about it.
Jonathan, did you have a?
Sure. I have a comment and a question. So the comment, today we're focused on the FDA regulatory use, I assume, or EMA of this, but there obviously are many other uses, including payers will use this information. And I'm thinking about the SMART trial design that Alyssa was talking about. One of the areas that we really neglect is clinical practice guidelines. And due to the vagaries of when a drug comes onto the market, particularly in oncology sphere, you may have 7 drugs that are approved for metastatic renal cell carcinoma. Some of them were approved for first-line therapy, some were approved for second-line therapy, some were approved with no differentiation, but it was based largely on the trial that was performed for practical considerations of who they could recruit, both ethically and feasibly. And what we really need is a blueprint, because it's being used out, all these drugs are being used out there in varying orders, as patients fail on one drug going to the next drug. And so these SMART trials would be very helpful, this SMART design to do sequentially, even though you couldn't say for certain which drug had the benefit from a purely regulatory, are we going to approve the drug based on that? It does give you a lot of information that would be useful for the medical community. So my question is more rhetorical and maybe a little bit devil's advocate. So going to Professor Gibson's slide about the COPD and the 100% and the 1.7% making it into the randomized controlled trials. So for many drugs that we approve based on that 1.7%, we give a broad indication. We extrapolate and say that that 1.7% is reflective. We don't know of any reason not to approve it for all of COPD and not list every exclusion that was in there. Sometimes we do, but oftentimes we don't. And so a drug will get on the market for treatment of COPD, even though it was just that 1.7% that had all these restrictions and who could enter the trial due to exclusions. And we kind of know why they do those exclusions, at least in theory. It's to decrease the noise and have a better chance at having a positive outcome, whatever they're comparing it to, and the amount of patients that they're willing to pay for, both practical and monetarily, to enroll in a trial. So now we go into the era where we're doing a real-world evidence thing. Whether it's, let's ignore for the moment randomization, and just say whether it's observation of all 300 million Americans being treated with a drug, and we're making a comparison between something. And we have everybody. We don't have any exclusions. But we see it only works, and this is going to make her cringe, without pre-specifying it in that 1.7% that didn't have all those things. So we didn't pre-specify it, or even if we did, let's say. So would we just approve the drug? So this is somewhat rhetorical, but would we say this is only approved in people that have exclusion 1, 2, 3, 4, 5, and not in everybody else? And then, of course, would it be used that way?
So yeah, we'll be able to tell you whether you were right or not to approve it, is the first answer. Be nice to be able to say you were wrong, or really you should have approved it.
Or partially right.
Or partially right. But then seriously, I mean, you know, in most COPD studies, you know, people with heart disease are excluded. They're all smokers, you know, what's the sense in that?
I mean, I would just say on a kind of a clinical practice guideline or from professional societies, I mean, you know, while there's certainly a lot of interest to kind of look at the totality of evidence, which is kind of what you're bringing up, it's pretty hard to say this is what everyone should be doing as kind of recommendations based on studies that are not pre-specified or observational nature. It is hard to do that because there have been a number of instances, at least in the cardiovascular field, where something made biological sense. We had good observational data to support it, and then went and did a trial, and then got surprised. And so after you've been burned with some of those scenarios, you know, and it comes up often. So we may see very extreme results that look like reincarnation. And so then you got to question, is that really valid? But then the results that are more modest, that may be true, but you start getting nervous about, you know, is it, you still have residual confounding that you haven't understood. So it becomes a challenge. So.
Yet the level of evidence backing many of the guidelines still remains
right, or like
limited.
actually even so the ordering of medications. I mean, we wind up saying background therapy is because of historical nature, but if someone had just chosen to study a drug earlier that was in the development, then that would have been the background therapy, and actually would have been maybe it had a stronger effect, but we just never saw because of its later stage in development.
I don't think you were directing your question to me, but
Well, go
it's
ahead,
an
though.
opportunity
That's
to make a couple of comments. So, you know, we're trying to decide if the drug works. I mean, we talk about this a lot internally. And so, you know, we may be in the 1.6%, and we want the clean, and we exclude the people with heart disease from a COPD trial, which doesn't sound like it makes any sense. But if you're trying to see if the drug works in a clinical setting, you want to get down to this somewhat artificial environment where you can get the cleanest signal that you can. And that's a different question than asking, okay, I know the drug works, now I want to figure out who it works for. And the statistics may be very different for those two. Mm-hmm. We have a lot of conversations in FDA about subgroups because it's like missing data in subgroups. That's our job security in statistics. But, and I can't count how many times people say, is this subgroup finding for real or is it due to chance? And I just go, well, but I mean, and you can compute. We actually can compute. We have a working group, Estelle was on it, subgroup working group. We published a paper in December. We actually put in there probabilities that if you have a trend overall and a significant finding in the other direction in a subgroup, how likely is that versus a trend in the other direction? It's more likely than you think it is when there's nothing going on. And for that reason, statisticians are very cautious about subgroups, especially post hoc subgroups. And there are methods that borrow information to say, okay, the subgroup looks different, but maybe it needs to be shrunk a little bit with a shrinkage estimator back to the overall mean, but still allow that it looks different. But the way I see this playing out with these types of trials is pre-market we decide if the drug works. Post-market is a great opportunity to determine who it works for. And pragmatic trials will let you do that if you get a more heterogeneous population. But I think the statistical test might not be what you're used to.
Or what about expanding indications? So, the two examples I gave, certainly, they're not being intended to have a label change, but one could imagine that, influenza vaccine is...
Well, why couldn't they support a label change?
It's actually just, right now it's on the market for influenza, but the goal of the trial is actually to see if it can improve cardiovascular outcomes. And so that's seemingly an important outcome. And it's already in the market. Similarly for aspirin, that if there was a difference between the two doses, then that may be important.
I'm sure Bob has something to say.
Well, and it would get in, aspirin isn't exactly labeled, but if you knew that one dose was better than the other, that would change everything. It's not as though people haven't been worried about that for a long time.
But how would you feel about that evidence coming from sort of a real-world trial, whatever you want to call it?
Well, as I said before, I don't think real-world is the issue. I think if it's randomized and if you collected the data and if
pre-specified,
and if you actually, the worry about all these data is that they're noisier. But if you still show something, this goes to whether you'd believe non-inferiority and equivalence in such a trial, and that's harder. If you actually saw a difference, that's perfectly credible. Noise doesn't have a direction.
In that context, so if you saw something, because you're worried about noisy data, that it's more believable.
Right. I think in this environment where we're not that experienced, failure to see a difference is less certain. You'd be hard put to know. You do have reasonable estimates of what aspirin does, so you could try to write a non-inferiority margin, but my nervousness would be in this environment, I don't know what it would really be. But you'd have to talk about that. But if you actually saw a difference, that would be pretty credible.
But we have to be careful about that criteria of seeing a big difference, because you could also have a big confounding effect.
But they've randomized.
Beauty's in the eye of the beholder. So if you see, if you're randomized, then you...
Yeah.
We're randomized.
If you're randomized,
that's what he did.
But if you don't have differential follow-up, if this, if that, there's a lot of things that have to happen here. And certainly in an observational setting, if you see a big difference, we like to say we believe it more, but it's really that we believe that the confounding effect wouldn't be as big as the difference that we saw. And you have to rationalize that. And a small difference might be fine if there's no confounding whatsoever. So,
So if we randomize and we actually had 100% complete follow-up, and even if we had showed a small effect, but it was significant, that would translate in terms of a larger population health.
Well, and you had the subgroup specified beforehand and so forth. It seems like you could potentially run through a lot of questions that
in one big trial like the ones that you all have described. A lot of questions about that are relevant for label modification, also frankly be relevant for clinicians, patients, and payers in looking at some of these subgroup effects.
Yeah, I mean, in fact, we put out the protocol publicly for comments and revise it publicly, and it's, you know, as we do things, we'll have it there. So it's not just pre-specified, it's out there for everyone.
Bob, did you have a further?
I forgot. Is the aspirin study blinded?
It's not blinded.
Well, actually, let's talk
Microphone?
If people have some view about what aspirin does or doesn't do, and they stop more in one group than the other, that would make you nervous. So blinding would be better, but probably not as easy to do.
I mean, here the outcome is death and cardiovascular hospitalizations.
Well, no, but there's still the question of, well, for example, if you're on the 300, you can't take ticagrelor. Okay, well, ticagrelor works pretty well. It adds a lot to aspirin. So could that lead to differential treatment of the two groups in an important way? Maybe. I mean, if a lot more people on the 300 didn't get ticagrelor and the other people did, that could affect the results of the study.
Well, so actually for that specific one, we actually asked the community about whether we should include patients on ticagrelor, would there be willingness to randomize. And it was 51% versus 49%.
And that's significant.
And it was, and so, but because of the concerns about its current label, so we exclude patients with ticagrelor. So we won't actually
You could include them and include that as a subgroup. It would go the opposite. Even if 300 was better in general and went the other way, that's exactly what I'd predict.
Yeah. It's, I mean, it is, the sample sizes that you're talking about do permit potentially a lot of subgroup analyses like this. I guess the challenge comes into how many of those can you really anticipate well ahead of time, and where do you really want to put your, you know, put the bang for your buck, which subgroups.
I actually had a question slightly.
Okay, and it's got to be a quick question.
All right, it's slightly off. Has anybody in a systematic way tried to find examples of, or an enumeration of the places where differences have been found between, you know, the study that led to approval and the real world data that showed that they're frequently different? I mean, everybody sort of assumes they might be, but has anybody sort of collected all this to see when that happens, or how many examples of it there are?
Well,
we'll have an example for you later this year.
I mean yet.
I mean, we've done comparative effectiveness studies using observational methods, and we do see differences periodically. I'd say where we see it is for devices, because of selection of devices in the patients, we tend to see results that seem to be more effective than what the trial results showed for efficacy. And there I think it is because of the selection of patients and for the device or the procedure. For medical therapies, we often have found fairly similar results to on efficacy.
Well, I mean, there is this obsession with effectiveness versus efficacy. I'm just wondering, I'm sure poor compliance in the real world could make a big difference. We know that one.
And
are there other things that also we're not smart enough to have figured out?
Yeah,
Or
so in the scenarios where we haven't seen the effectiveness translate in practice, when we look further, is because there is very low adherence or persistence to the therapy, and so that became the explanation.
Well, that one you could sort of predict. If they don't take the drug, it's not going to work.
Yeah, still an important one in practice.
That's right.
For clinical impact. Well, I want to thank our panelists for a great discussion, and we're going to take a break now and then build on this in the next session. Thank you all very much.
Okay, we're going to go ahead and get started on our next session. If I could ask everyone out in the lobby to begin making your way back to your tables. Is this on? Okay, thank you. We're going to go ahead and start our next session. The focus of this session is to begin looking at the potential applications of purely observational data in the regulatory context. I'd like to go ahead and introduce our first speaker for the panel, who's Jonathan Jarrow. Jonathan is in the office of the director at the Center for Drug Evaluation and Research at FDA. Jonathan's going to give a presentation, and then I'm going to introduce the rest of the panelists who will join me up here on the stage. So, Jonathan?
Thank you, Greg. Oopsie. Good afternoon, everyone. We're now at the most exciting portion of today's content, observational trials. To make it even more exciting, to paraphrase Bob Temple, not over my dead body. So I mean, where to begin? Even though we've done it, let's just start with some ground rules. So what is evidence from clinical experience? So really, most of today, we've been talking about randomized controlled trials. Yes, within the healthcare system, using different forms of data collection, et cetera. But for the FDA, as you've heard Bob say, this is really a no-brainer. Randomized controlled trials is something we've accepted for decades, and this is not a big deal. There's a lot of things we need to learn about how to do it. What settings it can work, and what kind of endpoints we can collect, and the validity of those. But by and large, that's not the big challenge. The big challenge is this holy grail that no one will admit to thinking of, which is if I'm a guy or person with a great idea, can I take an existing database, punch a button, and compare one drug to another therapy or a device to another therapy, whether it be no therapy or an alternative therapy, show evidence of efficacy, and get labeling of a marketable product in the US? And that's really the question. I mean, it's really the question is about efficacy. So the definition would be evidence obtained from observational studies or clinical experience, and this can be from registries, electronic health records, claims data, social media, et cetera. And the uses from a regulatory perspective is safety or efficacy, and we shouldn't spend a lot of time talking about safety. I will go over some of this today, but basically all of our safety collection to date has been observational. We do not pre-specify the collection of safety data in most of our drug development programs, with the exceptions of the big safety outcome trials that we do for certain drugs looking for cardiovascular risk. But it also could be used by healthcare, economic information for payers, research for academia, or for hypothesis generation for drug development. So there are many potential uses outside of the regulatory sphere. So let's spend a little bit moment going over the statute. And I heard people talk about the statute early this morning, kind of implying that the statute requires people to use arcane, non-informative, non-relevant, clinically relevant endpoints in clinical trials. There's nothing further from the truth. The statute basically says for drugs and biologics, one or more adequate, well-controlled studies that demonstrate that the product has the effect purported to have under the specific conditions of use. It doesn't say you have to use an arcane endpoint of a surrogate that maybe doesn't mean anything to a patient, like blood pressure or lipid levels or FEV1. We use those. FDA collaborates with industry to use those because they're very practical. And it's something that can be measured. It's something that people have confidence in that does predict a clinical benefit, but maybe it doesn't. And that it is an opportunity to win. In other words, to demonstrate superiority over another drug, or in other cases, non-inferiority over an already approved, established therapy. What's interesting is on the device side, CDRH, they have this pilot program called parallel review. And this is a parallel review by both FDA and CMS, so a payer, for a medical device that's coming to market, and they have input on the development program. And one of the participants in this parallel review was doing studying a device that was for the treatment of hypertension. And FDA said, blood pressure is fine for your primary endpoint. Just show that you've reduced blood pressure. And CMS said, no, that's a surrogate. You need to show that you've actually saved lives. And was not happy with that surrogate, even though we've been accepting blood pressure as a surrogate for regulatory action on drugs for decades. So you can see how this develops and transpires. And the other thing, just to get one more subtle point from that pilot, is that they also wanted to make sure that there were a lot of people over age 65, because CMS only covers patients over 65, except of course Medicaid. But they wanted to make sure that that was in the study, because otherwise the study wouldn't be applicable to their patient population. So back to Martin's 1.7%. The other aspect of the statute doesn't say you have to have substantial evidence of safety. So we've never required substantial evidence of safety, again, with the exception that I was talking about a moment ago. The other big difference is drugs versus devices, and I assume that today we're really focused on drugs and not devices, but the statutory standard for devices is reasonable assurance of safety and efficacy. It's not substantial evidence. And the guidances that come out of CDRH say that valid scientific evidence for reasonable assurance can include randomized controlled trials, but also include observational studies, registries, and case series, and large patient experience. So all of those are included as potential valid scientific evidence to establish reasonable assurance of safety and efficacy. So really, again, back to our Holy Grail. The Holy Grail is an observational data showing efficacy of a drug or biologic, is what people are really about. So as you know, unfortunately, going to the end of the day, everything that I was planning to say has actually been said multiple times already. But just to quickly go over the spectrum of evidence, and this is analogous. Someone was talking about our clinical practice guidelines being based on fairly weak evidence in general, mostly expert opinion. But there's a spectrum of evidence out there that's analogous to the levels of evidence that we rate for clinical practice guidelines. And randomized controlled trials and pragmatic trials, as long as you define them as randomized trials in the in the healthcare space, these have the highest level of evidence. Are they perfect? No. There's all sorts of problems with these. And we get applications with randomized controlled trials that are purported to show an effect, and we review them and say, no, you didn't have substantial evidence of that effect. And Lissa alluded to some of the problems. Some of it's missing data, some of it's crossover, some of it's picking a post hoc subgroup to look at because they failed on the ITT population. Some of it is just in the way if you look at progression-free survival in a cancer trial and have a bias in the way that the studies are performed in terms of the timing, not being in the windows of, that were predetermined, there are all sorts of issues that could have informative censoring of patients and the Kaplan-Meier curves. So there are a lot of issues in randomized trials, so they're not perfect. And then we get to the observational trials. And obviously, we prefer prospective observational trials rather than that retrospective hitting a button after all the data has been collected and see what you find. So this is where you pre-specify what the endpoint's going to be, you pre-specify what the treatment's going to be, you pre-specify your groups, and you have some control, at least over the safety data that's collected. You can specify what specific side effects to look for. Registries are single arm by definition, and these are typically prospective, and you can pre-specify, but not all registries are equal. And then you have case series or case reports. And this would probably be considered, at least in our opinion, the lowest level of evidence. So history. What has FDA done with observational data in the past? We've looked at safety for new molecular entities. We use Sentinel for post-marketing safety assessment. For efficacy, rare disease. We keep using this term rare disease. Lung cancer has become a rare disease, at least the way FDA defines orphan designation. So you can have rare diseases that are tens of thousands of patients in the US, and you can have a rare disease that's 20 patients in the US. And there's a big difference between those two, as you'll see in a moment. We also use observational data at CDRH for devices. Can we use it for labeling changes or labeling updates? So let's go over some of the case examples. So here's a list, and Janet referred to some of these this morning, of rare diseases where the efficacy findings for that drug was based essentially on case series or case reports. So FDA has already done this, despite our statutory requirement that we have substantial evidence defined as one or more well-conducted, adequate and well-conducted trials. We have, for instance, Lumizyme for Pompe disease, survival data from an international registry. Carbaglu, data on plasma ammonia level reductions in a case series. Cholbam, data on growth, survival, and reduction in laboratory parameters of cholestasis in a case series. Glucoropidase, for methotrexate toxicity. This was based essentially on a treatment protocol, expanded access use through the NIH. Meterleptin, another one that was from a expanded access or compassionate use treatment protocol, and most recently a treatment for 5-FU overdose was also based on animal data and a series of cases in expanded access used in the US. So there are times in which we fully understand the disease very well, we understand the pathophysiology, we understand what the treatment is doing, and there is a large effect size. So we're not worried about confounders. And some of these, almost every patient in the US was treated with this drug, and so we actually had the entire population. We had Martin's 100%. What about labeling changes? We've heard a little bit about the influenza vaccine being studied, but there was actually an observational trial that was done retrospective based on Medicare claims. So it had almost 1 million people on the high dose and 1.6 million on the standard dose, and was able to demonstrate what FDA interpreted as a real effect of the high dose being superior from the standard dose. Rabies vaccine is kind of an interesting case example. This information came out of a drug shortage. So the standard dosing of 5 doses, there wasn't enough supply for everyone that needed it to get the 5 doses. So people were routinely using a reduced dosing regimen, and they showed an effect that was convincing enough for CDC to make that the now recommended treatment with this is only 4 doses. For hypothesis generating, the FDA, in collaboration with the NIH and CDC and the World Health Organization, has this project for neglected tropical diseases, looking at the world experience of repurposing drugs for treatment of neglected tropical diseases with a website and a mobile app and a global reporting tool to report cases. So will there be an approval based on this? Not over Bob Temple's dead body. But, but there may be. Wait and see. So I'm going to talk just a moment about methodology. I am not a statistician. I'm a urologist by background. I am a statistician's worst nightmare. I have a stat program on my computer, and I love punching the buttons and seeing what kind of p-values I can get without knowing whether it's the right test or anything like that. But let's talk a moment about it. So one of the big concerns with observational data that we don't have with randomization, assuming that there's not a high dropout rate and crossovers and the like, is that we don't have to worry about known confounders and the unknown confounders. So now I'm sounding like Rumsfeld. So there are the factors that patients have that we know will influence their prognosis and potentially their response to a therapy, predictive and prognostic. But there are attributes of patients that we have no idea about. So before we knew about BRAF, did we know, or EGFR, did we know what patients would respond to one drug versus another drug? No. And so the question is, in observational studies, when we have errors, whether it's errors missing data or errors of diagnosis or errors of outcomes, is it random? Which, Bob keeps saying, well, if you win and it's just random errors, noise, it should be equal in both arms of your observational study, comparative study, and therefore you can trust the results because if you win in the presence of noise, it must be even a bigger effect. But what if it's systematic? And do we have tools to detect whether something is random or systematic? And that's really a question to you and something that's going to have to be established as we start looking at using this type of evidence to make regulatory decisions. The other thing is analysis and review. I referred earlier to we still have problems even with randomized controlled trials in differentiating whether there's still a bias introduced post-randomization. And this is one that really struck me. When I started out in FDA in the reproductive urology and bone metabolism division. And so I'm very familiar with this. In 2000, we've been using bisphosphonates to treat postmenopausal osteoporosis. There are a number of drugs approved, been on the market for a long time, and there are constantly new safety signals coming out with this, like atypical hip fractures. And in 2010, there were 2 studies published in the literature, one in JAMA and one in BMJ, both looking at the association of bisphosphonates and esophageal cancer. And I was struck by the fact that both observational studies were performed in the same database over the same time periods. They had the same people that had esophageal cancer in both of these studies. And one methodology, and I'm, again, not a statistician, so I won't make a comment about which one's right or wrong, but one came out with no evidence of an association with a relative risk of 0.96 and the confidence interval, as you see. The other one found a confidence interval that excluded 1 and said there's a 30% increased risk of esophageal cancer for women taking bisphosphonates versus never having taken bisphosphonates. And then when they studied duration of bisphosphonate use, it was even higher relative risk. So which way do we go? Which study do we believe? If they were 2 different populations, I would see might be understandable. But this kind of makes me very uneasy with observational data when you can have 2 studies of the same populations, just using different observational statistical techniques, get different results. So what do we need? As discussed over and over again, we need some demonstration projects. We need to be assured that we can do this. We can link the data between claims and EHR, that we can measure outcomes, that we can find out who died using this thing, because death is not, as was mentioned earlier, is not typically in each EHR or claims data. So we need demonstration projects to determine both methods, problems, and then which outcomes this will work for. We've always seemed to talk about MACE, myocardial infarction, stroke, and death as an outcome measure. That's the low-hanging fruit. How are we going to do depression drugs? How are we going to do overactive bladder drugs? How are we going to do a variety of drugs? I mean, many of our drugs are for symptomatic diseases. Are we going to leave those out from all this? As mentioned by Janet, there's some regulatory issues that we have to deal with, and this is for FDA to do. You know, is this going to be, how are we going to apply Part 11, which is, you know, the electronic data, and what needs to be ordered, what's going to be considered source data, and then, of course, Part 50. If you're doing your randomized pragmatic trial, you're collecting consents at the outset of the trial. If you're doing retrospective, you run into a problem. So let's say we have a device, is the typical example. We asked the company to do a post-marketing study to look at some outcomes, whether it be safety or efficacy with their device after approval for marketing, and they can't accrue, or they have trouble accruing, and they come back and say, can we use this registry? There's a big registry that was created in Europe. This device has been on the market there for 5 years. Can we use the registry? And FDA says, sure, that's great. But now, can they use the registry? FDA requires, US law requires that they get consent if it's a trial being done for regulatory purposes. And so even though the common rule would exclude that as part of standard care, low risk, FDA's current regulations do not. So those issues have to be dealt with. And as you'll hear tomorrow from Melissa, we're working on developing a federal evidence generation system. So we need a lot more academic research. Bob keeps on making pleas for people to look at it. When has a pragmatic trial shown a different effectiveness than the randomized control trial? When we have a randomized control trial, can you duplicate those results in doing observational studies after the fact? So these are all things that need to be done and hopefully will be done soon. Thank you.
Thank you, Jonathan. I'd like to go ahead and invite the other panelists for this session up. Richard Platt is professor and chair of the Harvard Medical School Department of Population Medicine at the Harvard Pilgrim Healthcare Institute. Mark Berger is vice president of real-world data and analytics at Pfizer. And then Marcus Wilson is president of HealthCore, subsidiary of Anthem. Welcome all, and we'll go ahead and begin our discussion. So Rich, I think you have some opening comments?
Right, I'm next. So I ought to begin by saying I'm here as a poser because it's above my pay grade to answer the question, what would it take for purely observational data to be suitable for regulatory needs? If Jonathan's talk convinced you of nothing else, it's that I'm not in a position to advise FDA on what it should make, how it should make regulatory decisions. But I am prepared to talk about what it would take to be able to use observational data to drive, to be high quality evidence to drive clinical practice, for instance. And as background for it, I do want to comment on some of the remarks earlier today about the quality of observational data. Say claims data has lots of problems. I'd say at this stage of our lives, EHR data has many more problems. That is, we've got decades of experience working with claims data, and we understand a lot about what it can and can't be used for. I think we're still in the Wild West days for most EHR data. I mean, I applaud the many initiatives that are going on to work towards standardization of EHR data. But for the time being, we live in an environment where customization of electronic health records so that they are mutually incompatible in terms of what they contain is a feature and not a bug. And there's a substantial chance that that part of the harmonization efforts will get to a superficial level of harmonization, while the actual coding of what the clinician intends to the EHR may still be quite different. So I have high hopes for EHRs as sources of high quality data, but we still have an enormous amount to learn about that. I think the other thing to say about observational data is we should explicitly include prospectively acquired observational data by reaching out to clinicians and patients about their experience. So to Jonathan's point about the fact that many of the things that we care about is information that really can only be obtained from the individuals themselves. I think that's part of what we should consider to be the observational data set. That is baked right into the way PCORNet operates, for instance, the notion that patients are really active participants in the development of evidence. And I think that's going to be an increasingly important piece of our thinking about how to do these things. With regard to what kinds of evidence should we be attentive to, I think we have to be prepared to be opportunists. In the absence of randomization, there are certain circumstances in which I think we can be more confident about accepting observational data as evidence than others. So, for instance, the influenza vaccine study that Jonathan mentioned is a very good example, I think. If you haven't had a chance to look at it, I recommend it to you. It has several appealing features, but one of them is that people who received high-dose vaccine had a better outcome than people who received regular dose vaccine. Think if the result had been flipped. We said high-dose vaccine, people who received regular dose vaccine had a better outcome than people with who received high-dose vaccine. It would suggest to us that there was important unmeasured confounding, because the expectation is that if there are biases, that we would expect them to operate in the direction of the sicker people getting higher dose vaccine. So I'd say that's a situation in which if I tell you that the effect estimate is 20% different, I'd say it starts off seeming like better evidence if the advantage is for the high dose vaccine than the low dose vaccine. And I think that basic approach holds true for a number of situations. Secondly, that study was remarkably well designed. As you read the methods section, what was it, 2 million people who were studied approximately in that. They were drawn from a much larger population of Medicare beneficiaries for whom CMS had information about the vaccine they'd received. But they restricted the population in a way that makes it seem more plausible that the population who received the 2 doses were likely to be comparable. One way an important way that they did that was to say, we will only study people who received their vaccine at a pharmacy. And the reason for that was to say, it gives you some sense that people were ambulatory, that there was some self-efficacy. And so we're talking about a population that is likely to be somewhat more homogeneous just by the fact that they went to a pharmacy. And then the next thing they did was to say, they matched within pharmacies. That is, they only accepted people who received their vaccine at a pharmacy that had distributed both kinds of vaccine within a 2-week period. Those are really very clever design features. I haven't read about them in any textbook. That is, this is a design that arises from deep knowledge of the not so much of the vaccine, but of the population that's immunized and the way healthcare is delivered. So I'd say it's always good to make sure that you don't just push the button and ask for an answer to come out, because you can have more confidence if you know how the trial was designed. I'll retreat though. My final point is, so I think that study is extremely high quality evidence, partly because of the way the result came out. I think in some ways that kind of result would be credible partly because of the direction that it went, but that we should also We should also be true to our intrinsic natures as Bayesians. That is, all of us approach decisions based on what we think about the world anyway. And so I'd say the place for observational studies should at least start in environments where we think we have a pretty good idea what the answer is, and we're trying to extend that to a new situation. So if we have good evidence about how a vaccine works in the under 65 population, then observational study seems to me that gives a plausible result is pretty credible if we say we're going to extend the age range or we're going to extend it to a population that hasn't been well studied. We're not going to do randomized, you usually won't do randomized trials in that environment. And the question is, are we better off using the observational data there to inform what we think we know about effectiveness than we would be if we said we know nothing. And so I'd say we start with a presumption that we know a fair amount about how in this case, the vaccine works in a particular setting for a particular population, and extending it to others or to other conditions is probably among the safest ways we could use observational data to support the development of good clinical practice, evidence-based guidelines. So where does that leave us? Unlike the situation of randomization, where we should be able to trust randomization to deal with so many of the problems that we care about, I think for the foreseeable future, we're going to have to say observational studies can only be useful for certain kinds of questions right at the outset. We should start off saying it will be unhelpful to do an observational study in certain settings, and then be prepared to only accept certain kinds of answers from observational studies. Back to you.
Great. Thanks, Rich. Mark?
So it's a pleasure to be here. First, I'll start with a disclaimer that my remarks are my personal remarks, do not represent the views of Pfizer, Inc. And I'll also start by saying that there's no doubt that the randomized controlled clinical trial was the single most important methodologic development in clinical research in the last 100 years. There's no question about that. We're not getting rid of the RCTs, but we are entering a digital era of data, and this digital era of data is going to power our learning healthcare system, and people are using it to make decisions today. Now, whether it's good enough for regulatory purposes, that's the question today. But payers and providers, and I've worked for pharma companies, I've worked for payers, I've been a provider, they use it all the time. They use it to say which patients might be better off getting pre-screened for a disease, which patients deserve to have case management or care management. How do I design my benefit design to make sure I'm getting the best population outcomes? It's being used today. And I imagine that if any of you walked into a surgeon's office today and you needed some surgery, you'd like to know, of the last 1,000 patients that surgeon operated on that looked like you, same age, gender, comorbidities, what was his outcomes with that surgery? Don't tell me anyone here wouldn't want to know that information. So evidence-based medicine says you use the best available information at the time to inform your decision. So that's one thing at the bedside. It's another thing at a treatment guideline level, and it's a very different thing at a regulatory level. So we have to, I grant that. And so you've already heard, I'm not going to go into it, the FDA is already using observational data. And the question is, are we reaching a tipping point? Can there be things that the FDA can do today or in the near future to more use observational data to inform regulatory decisions? And I'm mostly interested in labeling, because that's the holy grail. If it's in the label, you can talk about it. If it's not in the label, you can't talk about it. So we've heard there's challenges. Data quality, it's all over the place. However, that means that observational data does not equal observational data does not equal observational data. The data from the Salford study is probably better than most observational data that's out there. And even if the data is not that clean and has some missing data, it doesn't mean that the conclusions you're going to get from that are incorrect. Because how you analyze the data to deal with bias and confounding varies dramatically in analytic and design rigor. So not all studies are equal. So how can the FDA decide whether the data is good enough and the design and analysis was good enough to support a decision? Well, I've been really privileged over the last 10 years of my career to be involved with a bunch of different initiatives where we've been trying to set forth best practices about how to analyze observational data. And in 2009, I was part of an ISPOR task force that put out good research practices for retrospective database studies that was published in Value and Health. In 2010, the National Pharmaceutical Council sponsored the GRACE principles, which was principled in, published in the American Journal of Managed Care. And then there have been other things, and most recently, there's been a collaborative between ISPOR, the National Pharmaceutical Council, and the Academy of Managed Care Pharmacy, the pharmacists who do all the P&T reviews to decide whether drugs go on formulary, to do a CER collaborative initiative. And one of the things we developed in there was a questionnaire to help people who are trying to evaluate individual pieces of observational data say, what is the relevance and the credibility of this data? It's not a 1-0. This is not a binary function. You have to make a judgment about how good the data is. So in this questionnaire, we divided it into two big domains. One is relevance, and you've heard about it out today. Is the population the right population to which you want to make a decision about? Are all the interventions that you're interested in, were they included in that study? Were the outcomes, both primary and secondary, the ones you're interested in? And did you measure them over the right period of time? What was the context? This is like, basic blocking and tackling. But under the credibility side, we go through specific things about design. And in design, we make recommendations. So there's nothing wrong with using observational data for hypothesis generation. Great. But if you want to use it to make a recommendation for a health policy or make a recommendation more generally to a population of patients, you should have an a priori hypothesis. You should have a formal protocol. This is where we went with RCTs. We need to go to the same place with observational data. And we even argue that the way to know that is that those studies should be pre-registered. Now, we went through the pre-registration conundrum with RCTs in the last 15 years. Why? Because there's publication bias. Not every journal wants to publish a study with a negative result. But you need to know what the negative results were and the positive results. And you need to make sure that what people are reporting on is actually what they did. Well, we should do the same thing if we want to elevate observational studies to make them more credible for the FDA. Data quality, there were many things we go through there that talk about how you do data quality. I'm not going to go into that. Analytics. Rich has mentioned one thing. If the result of the study comes out and actually flies in the face of an expected bias like channeling bias, that should give you greater confidence. Things that we talked about in that questionnaire was, if you want to build on an RCT, and we think that all observational data is built off, generally speaking, most of it's built off of RCT data, do an analysis that shows that if you restrict the population to look like the population that was in the published RCT, and you can reproduce the result you got in the RCT, then when you look at the more general population, you should have greater confidence that what you're finding there is not hooey. It should make sense. And maybe you can draw conclusions about that and make some regulatory decisions about it. More recently, a lot of work has been done. David Madigan has led this effort to show that in order to deal with systematic bias and bias in the data sets, you need to include negative and positive controls. And we talked about this in the questionnaire about using outcomes that are completely unrelated to the intervention. It allows you to look to see if there are temporal or other systematic biases built within the study. And we've done studies where we see that there are general biases built into the databases, but even with that, when you use positive negative controls, you can still find important associations that are real and powerful. I think you could make regulatory decisions off of that. The FDA recognizes this is coming. So we heard a preview, and there was the announcement about repurposing Sentinel, which my good friend is a very important part of, to start looking at effectiveness and efficacy over time. And this day is going to come because patients are going to be making decisions. They're taking more control over that. They want to get a hold of their own data. And they're going to want to know what patients like them, what happens to them. And so the label, which changes from time to time, will fall out of step. And the question is, how do you keep it more in step so that it actually is relevant to the practice of medicine? So what is the place to start? And the first place to start is in the relevance place. So if, as Rich has just mentioned, if you're not going to do an RCT in a population that wasn't included in the primary RCTs, but you have good observational data, and it makes biological sense and it's plausible, why wouldn't you want to put information about that into a drug's label? Somewhere. The credibility issue is much more important, and this is where I think that we need to call and inspire the FDA to get actively engaged in shaping the criteria for understanding what good credible evidence is. As long as we look at all observational data and tar it with a broad brush, we're not going to get anywhere. So what are things that you might do, that you might advocate for, that might move the people who are doing observational studies to go to best practices? Well, one of the things we do with RCTs is we say, one RCT is generally no good. You need to have two. Well, with observational studies, shouldn't you have multiple observational studies that show the same result? And shouldn't you require that it uses multiple analytic approaches to deal with the problem that Jonathan was talking about? Yes. So if you have multiple observational studies that are using different analytic approaches and they all point to the same result, I got to tell you, that's about as good as it gets in evidence-based medicine. We have to have pre-specification of hypotheses, we have to have pre-registration of results, and we have to look at where we think the known confounding and biases will push it and see if the results are going in that direction or against that direction. So I think there is opportunity today, and I realize that there are some regulatory hurdles that need to be talked about, but evidence-based medicine would say, we are using this kind of information today to make the best available recommendation to patients going forward how to take care of themselves for their individual problem, for who they are individually. And I think there is an opportunity for the FDA to start looking at populations that are not included in randomized controlled trial, looking at dosing, as you already have, looking at adherence and compliance. I think that there is enormous differences in adherence and compliance, and whether or not that is due to the particular therapy, but the way the therapy is administered or the way it's used or the fact that it's, the patient's on 7 other medications, isn't that interesting information that a patient would like to know, that a treatment regimen A in most patients has more adherence and compliance than approach number B? That would be useful information. And I think down the line, as we begin to do that, I do think there's an opportunity for new indications, particularly off-label indications, where you're not going to want to conduct an RCT. And I do think that, despite my good friend Bob's concern, and I agree with you, all relative effectiveness studies have relatively small effect size. I think that if you do multiple well-designed, well-executed, non-randomized observational studies, and you use multiple different analytic techniques, and they all go in the same direction, I think you can infer causality. And there are a number of statisticians who believe that as well, particularly if you put it in a Bayesian context. If you have a baseline RCT that says, we know in some population it does work, it's plausible to work in this other population, I think we can get there. And I think we will be there, and I think the world's going to pull us in that direction. And so my plea is, rather than saying, let's hold back the dawn, I'd rather have the FDA be actively engaged and putting out incentives saying, look, you ever hope to get an observational study to get a labeling claim? Don't come to me with one observational study. Come with me with multiple studies using multiple different analytic approaches have your study pre-registered, and et cetera. If you don't pre-register it, and you don't have a pre-hypothesis, then listen, that's a great study, that's a hypothesis. Now go do that in some other database. Go do that with multiple analytic approaches. Show me you get the same result. I think we can elevate the criteria and begin to decrease the separation between the gold standard of the RCT and what you think is, I think they're not as far apart as you think they are. Anyway, I've been proselytizing that for a long time. Bob knows that.
Thanks, Mark. Marcus?
So how do you follow
that?
How do you follow that? I don't know. So I've got Jonathan's problem. Everything you want to say it gets said as you go down the line. So I'll try to be. Can't be as provocative as Mark was, but I actually really agree with the last point you made on this. And I think really what we're trying to accomplish, and what I think is really the primary objective of our efforts leading up to today, and then even our efforts after this, is to begin to develop a blueprint. Because if we reframe the problem, really, when I came into this discussion, it really wasn't me trying to find a way that we can include pragmatic designs or observational designs or real-world evidence development in regulatory decision-making. It was much more about how do we address a very big issue that we're facing in our system, and that is that when a product receives regulatory approval, then those of us who have to make decisions post that approval or have woefully inadequate levels of evidence to make the decisions, whether that's policy-related decisions and payment decisions or prescriber-level decisions. We really don't have enough evidence to make an informed decision at that point. So think about it in the context of, it has been mentioned several times, but I'll give a little stats around it, and that's the quality of the evidence that goes into therapeutic guidelines, which, whereas as payers, we use a lot of the guidelines to sort of to frame out our quality measures. As providers, we use a lot of the guidelines to help guide therapy and choices we make. And so they're really, in many ways, the gold standard of care for a lot of organizations and provider types. And so if we think about the quality that goes into it, think about cardiovascular guidelines. When you look at a very systematic review of the evidence that goes into cardiovascular guidelines, depending on the guideline that you look at, the worst case scenario is there's 0% good evidence, where almost all of it is expert opinion. The best case scenario, which is actually congestive heart failure, is 25% of the time we know what we're doing. So that means 75% of the cases we really don't know what we're doing. For oncology, it's much worse. It goes from 0 to 18% with an average of 6%. For infectious disease, it's a little better, but it's only 15 to 30%. Meaning that most of the time when we're providing care, making decisions of any type, we really don't know if it's the right decision, and we don't have a systematic way of following up to see if it really worked out right. I think we were at a conference, maybe it was a CMTP conference, and maybe it was Sean who said this in a sidebar conversation we were having, and we talked about variability and care, and it's one of those things that everyone always thinks, well, if we just get everyone to follow the guidelines, we're going to achieve a high-quality, affordable healthcare system. And I just gave you evidence that that's probably not the case, and that the variability in care is probably justified to a large extent. And Sean, one of the things that Sean said that I was really getting to on this is that it's really not the variability that occurs. It's not being able to study that variability that's the real tragedy in all of this. And Sean, I don't know if I'm attributing it to you or not, but it's actually a really good comment. But from my perspective, I think that's really our problem we're looking to solve. So my question is, if it's not using real-world evidence, then what are we going to do to solve that problem? And so I'd really like to look at it a little differently and say not why we can't do something or where we can, is really how are we going to solve these issues? How are we going to solve these problems? So if I think about it sort of in a mathematical way, if I think about the improvements we're making in randomized clinical trials and the increasing investment in those, including using real-world data to help facilitate the conduct of those studies, perhaps we can get to in time where RCTs, defined as traditional RCTs, may get us a third of the way there. I actually think pragmatic type designs, and I know that there's not necessarily a clear distinction between those, but I think pragmatic designs may get us another third of the way. But there's still going to be a pretty large gap left after that, that something, whether that's observational data or some other form, has to begin to address that gap because it's still pretty darn big. especially if those decisions, if you're fitting in that last third of that gap in your decisions are made about you or your child or your parent or your siblings or your wife or your husband. So I think from my perspective, we still have a pretty big gap. Now the challenge I think for all of us that we're trying to get to, and this is a little bit of going toward what we're looking at I think tomorrow in the discussions I hope, is that if we look at pragmatic designs and observational designs, and we base our judgment on how well they fit into regulatory decisions or other decisions based upon the real-world data that goes into those designs today, then we're probably accurate when we really put a lot of qualifiers on the value of those data. And so what I think is important is that we think about what are the things that would amplify the utility of the data that exists today. And I think there's really three major amplifiers within that we really need to focus on. One is integration of data. Now mind you, there's a difference between linking data to enrich and integrating data. And I think it's beginning to resolve some of those issues that were referred to earlier around really understanding how good the quality is, but linking data and integrating are actually different in my mind. So the more we can integrate to give a more robust view and enrich that view of that individual, the better off I think we're going to be. So that's a major amplifier we've talked about. We've talked about another one only briefly in a couple of remarks that came across, and that is the longitudinality of the data. Right? So right now we get snapshots of data, and when someone within our Anthem health plan leaves and goes somewhere else, we lose the follow-up with them. Or when they're coming to us from someone else, we don't have that historical perspective. Now we're working on ways to try to solve that. Starting in California, we have an initiative called CalIndex, or California Integrated Data Exchange, where we're going in with other payers to try to build that longitudinal view, sort of putting data, if you would, in a pre-competitive space and saying, look, what's more important to all of us than anything is if we create that integrated longitudinal view of the population, and then we'll compete after that. But let's do that because the most important thing is that's going to power a lot of decisions that we can't make today because we don't have that integrated view. And I think that getting providers involved in that as well is really our big aim in trying to create a longitudinal, deep and longitudinal view. But there's also a third piece of this, and I think it's just as important as the first two, and that is that as we build that view, that integrated view, we, especially today, and Rich said it extremely well, is that we really don't know how to use these data. Claims data, we do have a lot more experience with it. We do have a lot better sense of what we don't. Well, we know what we know what we don't know, and what we don't know what we don't know is a very small slither with claims data. With electronic health record data, it's a big, we don't know what we don't know. That piece is very huge. So from my perspective, that's a big part of this. But that third piece of this, the third amplifier, is maintaining provenance, a robust provenance. We need to make sure that we stay close to the systems that generated the data so we understand the circumstances under which it was generated. Because if we don't, we can't look at things like non-apparent confounders, non-apparent sources of bias, that we may find reasons that we find things in the data that we can't explain. Maybe we can trace back in the systems and sometimes uncover really what that was and adjust for that. Or in some cases, what we found working with some of our provider groups is that when we're looking at data that goes into the electronic health record system and we're pulling it out and we're realizing there's 30, they want to try to clean it up too, especially if there's a reason for them to do it. We did a project just outside of New York City with a very large group practice there, and we pulled all their EMR data out and started integrating it with our claims data, with our Empire Blue Cross Blue Shield plan there. We realized that all the height and weight data were really bad, really bad. I mean, it was almost unusable. And so we went back to that practice and said, here's what we found when we started trying to look at your height and weight, simple entries into the chart. They went back in and systematically corrected that. They didn't realize how bad they were, the fact that they were including some of it in a note and some of it in a structured field, and they weren't really paying attention to the units of measure. They realized that there was a consequence to that, so they started changing how they entered that. And I think that's part of the feedback and part of that blueprint we have to get to is saying, look, it's not where it needs to be. What do we need to do to get it there? And what are the things we need to inform to make sure we get it to the right spot? So I'll end with that, really with a plea for all of us is not really think about why justifications for using real-world evidence in regulatory decision-making, but how are we going to solve that big problem that exists today post-approval of a product and getting evidence that we really need to make a continuation of good decisions that post-approval. So, thank you.
Great. Thanks, Marcus. So, just building upon the last two comments, I mean, it sounds like the onus is really on the researcher and the people who are putting this data together. So Marcus, you walk through steps to make the data better, steps to, and we heard a lot about that this morning. The challenge tends to be in the quality of the data. Is it measuring, is it showing what we think is it measuring what we think it's supposed to be measuring, and how can we use that? And so Marcus, you walk through how to improve the usability of that data. And Mark, you also sort of focused on what researchers need to do to elevate the credibility of the actual research, rather than what we didn't hear is a lot of sort of what FDA needs to do, and what it's more on producing better research and demonstrating that we have better research. So Mark, you mentioned that there are a lot of sort of best practices, ISPOR, NPC. A lot of groups have gone through and put together best practices in an attempt to say, if researchers did this, then that would be elevating the credibility of the results. But I suspect as those groups were putting the best practices together, they probably weren't thinking of, for regulatory uses. Traditionally, the data is thinking for payers to make a decision, or for providers to make a decision. Are the best practices sufficient? So it's a run-on, but I've got a question in here. Is the problem that typically researchers are just not following the best practices that are already out there? Or do we need to improve upon the best practices to really make sure that these studies are at the high level of credibility that you're talking about?
Listen, this is a new field. I mean, health services research has been around for, what, 30 years? 35? Epidemiology has been around for maybe 50. Outcomes research, less. Editors don't really understand how to really evaluate the quality of the information. The peer review process is not as robust as it is for RCTs. And so the quality is all over the place. At the same time, methods have advanced dramatically. And so in terms of dealing with confounding, one of the big advents during the late 90s was the advent of propensity score analysis to match your populations. It's not perfect, but it was a lot better than what it was before. And so the methods are improving as well. Researchers respond to incentives. So when the International Consortium of Medical Journal Editors put out some criteria saying, you want to get your study published, you have to do X, Y, and Z, guess what? Researchers do X, Y, and Z. And this is where I think the FDA can have a role. If the FDA wants to engage, it could say, you want to have any chance of getting an observational study to get you a change in a label having not to do with safety? Then I want you to do at least X, Y, and Z. FDA has not engaged with that as well. That would be a very powerful incentive. Around the world, this data is being used. It's being used by regulators. It's being used by HTA authorities. It's clearly being used by payers and by providers. And yes, all of these best practice things have been really focused on the concepts of evidence-based medicine. So evidence-based medicine has a different mandate. And the mandate of evidence-based medicine is to use the best available evidence to make the decision at the patient's bedside, which means you're always operating at the penumbra of what you know and you don't know, because you can't know everything for a particular patient. Having said that, you can use those kinds of best practices and those recommendations to think about how you would apply it if you want to make a population recommendation, whether it's a treatment guideline coming out of a medical society, or whether it's a labeling decision made by the FDA. I think we can apply those same kinds of standards. The question is, how certain do you have to be, and what's the regret for making a wrong decision? Well, if you're starting from the point of view, you're going to root it in, I have baseline RCT that tells me that this therapy works in this population, I've proven it, okay? And if the results are biologically plausible, and if you do multiple analyses multiple ways, and the results go against what you think the prejudice should be or the bias should be, I got to tell you, as a good Bayesian, most clinicians would say, I believe that. And I would think that there is an opportunity for the FDA to believe that too.
Morgan?
So you asked what I think the FDA could do, and to build on those comments, is really...
No, I didn't ask that.
No.
I think Mark asked that, just to be...
Okay, so what did the FDA...
Mark interprets that as...
Okay, so I wasn't sure of the run-on there.
Yeah,
So the... I got some good stories about Greg. Greg used to work for us. So I could just,
Marcus actually gave me an award. I don't think you remember this, about putting together a learning module for how to do propensity score matching in your company. But
yeah, I know.
Okay. But that's an old method, according to...
It's an old method now. It's outdated.
It's obsolete.
So can we have the reward back? I think the one thing I would say and is that I really do believe that we have to not just think of real-world designs, if you would, broader type definition of this, in the post-approval space. I think we really need to contemplate the using of them. And I would love to hear the FDA think that way. You don't have longevity in your genes, right, Bob? Oh, geez. Okay, so because you said not when your lifetime. I really do think it's essential that we think about that, because I think we're trying to close a gap sooner. We're trying to get to relevance much faster than we can today, and to wait to a post-approval time period, we're just adding on to that. We're really not accelerating it to the great. It's not transformative. It's evolutionary, but not revolutionary kind of thinking about it. And if there's a way, and what I would like to see from the FDA, by the way, is to say, okay, look, Marcus, if that's going to be the case, then here's what has to happen. Here's where we really need. Here are the basic principles that have to be met in order for something like that to occur. I think that's when you begin to get everyone shaping their data development efforts in the right direction. That's when you start getting folks thinking about designs differently than they have today. They start thinking about methods. They start thinking about their drug development process perhaps differently. And I think it's that signaling aspect that says, look, we have to fix a problem. If we're going to do it in a much more sustainable, rapid way, then here's the roadmap we need to get there. So that's what that's my opinion.
I can respond to Marcus. So I have a lot of empathy for you, because our job is difficult to some degree, but your job is an order of magnitude more difficult. So to get a drug into the marketplace, you just have to show efficacy somewhere. And then, not because of, well, for whatever reason, we wash our hands of it and say, we don't regulate the practice of medicine. And so once it's approved for COPD, if a doctor wants to start prescribing it for an off-label use, whether it's a different dose regimen, a completely new indication, that is their prerogative. Mark, working for Pfizer, wants us to put it in the label so that they can communicate about it. But your job is much more difficult, because we deal with whether it works, whether the risk-benefit profile. There's a great deal of uncertainty about that at times, sometimes more, sometimes less. So we don't have 100% certainty about a drug when we put it on the market. But you've got to make a value decision. You have to determine the clinical value. We don't say, as a statutory requirement, that a new antihypertensive drug has to beat every other drug on the market in order to get approved. It technically only has to beat placebo. It could be worse than any other drug that's currently on the market, but we think it provides another option for those patients who potentially can't tolerate the other drugs. And so we don't require them to show that they're better than everything else, except for the accelerated approval pathway, where you do have to be better than available care. And so then you're stuck, because now this drug's out there, and everybody wants to prescribe it. And it costs a lot more, potentially, than some generic drugs. So what's the value? Is there value to it? And so the decisions that you have to make as a payer are, I think, an order of magnitude more difficult than what we have to do to determine does the benefit override the risks of the drug, and is it efficacious? Having said that, when a drug is approved, even after approval, there remains thousands, and Janet said this earlier, thousands of questions about that drug that we would love, in the ideal world, to have a randomized controlled trial showing in every subgroup, every dose. Most of our drugs that we approve are very poor development program in figuring out the right dose. We would love to have a plethora of randomized controlled trials to answer all of those questions, but we're not going to get that. It's not feasible, it's not practical, and so many of these questions can be dealt with by real-world evidence, observational data, and we do for the most part. Basically all the safety, we have this like the esophageal cancer thing. I mean, they reviewed multiple studies and epidemiologic databases to come to the opinion not to make a labeling change for the class of bisphosphonates and add esophageal cancer as a new risk. And so we do it all the time. Have we put out a guidance on how we do it? We have not, and I think it would be worthwhile for us to talk about this and get some general principles because I agree with you. If the pharmaceutical industry saw a direct benefit to doing observational data and submitting it to FDA as a pathway for a new indication or expanding the population or contracting the population. That's indicated, or finding better doses, or find anything about their drug, I think that there would be more money invested in this space and therefore potentially greater development and greater confidence. I guess I'm not a Bayesian. You said all physicians are Bayesian. I guess I'm not a Bayesian because I'm a little concerned when you say, when someone makes a statement, if it shows a result we expect, I'm going to believe it. Yeah. And it's just, that worries me. I had an old professor teach me that research is a little bit like skydiving. It's best done with your chute open. And so it's best to have an open mind about what can come out. And what we expect to be the result is typically not the result when you do pure bench side research. So,
having said that, I meant when it's built upon an RCT. Not just...
No, it wasn't you that said it, actually. It was Rich that said it.
Well, I would say we are all Bayesians.
Well, and physicians generally treat the patient in front of them based on their experience with their last patient and not with all of their experience. And so, we prefer evidence-based medicine, but quite frankly, most of medicine's not evidence-based. There's still a lot of problems with physicians prescribing, for instance, anticoagulants for people with atrial fibrillation. And there may be some variance in patterns of care, but that's not one situation where you would really want that to be a varied practice. So, but again, getting back serious about the task at hand, I think there is a role for observational data. Maybe not everyone within FDA sees it that way. I think that we already use it, as everyone keeps on saying, we already use it. I understand that there's interest in us using it more, and we need to get more information about our drugs and how best to use them and which ones are better than others and what sequence is the best sequence and all that. And so, we're not going to be able to do randomized controlled trials for all of this. And so we're going to have to use the best available evidence. And what crosses the line to make it into labeling? You had it happen with a vaccine. It didn't make it into the labeling. They didn't believe
The
it.
Medicare study did not show up in the label. The company came in with a separate randomized study that ultimately wound up in the label. But the effect size was in the same direction. It was the high dose was better than the regular
well,
dose.
there you have it. So even that we didn't put in the labeling.
I went.
So where the channeling, where it went the opposite of what we expected. So, I think we have a ways to go. And again, it is an issue that needs to be worked on, and I can't go any further than that.
Sure. Okay, I think we have a minute or two. I'd like to see if there's any questions in the audience for the group. Bob?
All right. Not a question, but look, we already can, under our rules, use historically controlled data, and that's what this is. That's what all of these are. That's what they are. So we can use them if we're persuaded. We're usually not persuaded when the effect is modest and it's not very visible. But the suggestions Mark made, which we never see, mind you, are very good at helping people do it. One is doing it twice, showing the results the same. Having a protocol for one of these studies, having a protocol, never. This is not a new thought. I wrote an editorial in JAMA in 2000 or thereabouts saying you should always replicate your historically controlled trial. But it's not commonly done. So The other thing is, we take into account what we know already. I mean, if you know our evidence document, it says, when you're looking for a second claim that's very similar to the previous ones, we accept one study. What's more Bayesian than that? We have a law that says we can rely on a single study plus confirmatory evidence. They didn't write it as Bayesian, but that's sort of what it is. You've got priors, you've got beliefs that they all fit. And the idea of modifying a claim slightly when you already, as Rich said, have controlled trials showing that it works, it's intuitively obvious that you don't need quite as much data. But there's no experience with this. I'd be very interested in seeing all the things that people want to do to refine treatment of heart failure and stuff like that, and what kind of studies might expand current labeling to help you do that. I mean, just for one thing, all of our labeling on heart failure drugs assumes that you're on all the other drugs, because that's how all the studies were done. I'm sure in the real world, lots of people haven't been on a diuretic, lots of people haven't been on the other things. Maybe there's a way to look into that. That would be very informative to us. It would help modify our labeling. But some real world examples where people do this and then replicate it, that just seems like a great idea. For what it's worth, studies don't always come out the same. Rich knows more about it than I do, but the OMOP analysis of the data in Sentinel was very distressing because the results came out different in every site all the time. So is that a problem, or is that overcomable by doing it right, or I don't know. But there's a lot to learn before we're going to do this much. But we already can use a persuasive historical control.
So we are running out of time, but any quick reaction to?
I'd say one of the very illuminating things about this conversation is the fact that we've been talking about the fact that there are degrees of evidence, and yet regulation tends to force us into a black and white classification of it. And so as long as we're clear that, that there's a gradient and we need to know where we are on the gradient, then the hard job as FDA is to decide where on the gradient you can decide that white has changed to black.
Marcus?
So I'll go back to, there's a couple of points been made today about no distinction between efficacy and effectiveness, and no real distinction between randomized clinical trials and pragmatic clinical trials. Are sort of, especially with randomization being present. And so here's a consequence of us not drawing that line of distinction, and the FDA not drawing that distinction, is that if any of you have ever gone back into a pharmaceutical company, starting out with a pragmatic idea, and then take that back into the development process within that pharmaceutical company and try to keep it pragmatic or real world in any way, you'll realize that the gravitational pull back to a randomized traditional clinical trial is extraordinary. You never survive those. You'll have the greatest concept, and immediately, not long after, it gets pulled right back into a traditional design because of their concern with the FDA. And so the point is that if you really are as open to that, then be clear about that. Write better language about it. Draw some distinction to send that message that these are a little different, and we're good with that being different, and specifically when that might be, because it creates that black and white scenario where inside of these organizations, because of the fear of the FDA, and I think that's what we've got to try to overcome in part with the messaging on behind it.
Can I say, if pragmatic means randomized trials with fewer exclusions and things like that, what used to be called the large simple trial, we don't have any reservations about that. We never have. So I'm not sure
why.
what everybody's worried about.
Is quite different.
Yeah,
So
yeah.
our perspective from the FDA side, and we meet with companies at end of phase 2 to discuss the phase 3 development trial, is we are encouraging no exclusion criteria. And they want those because they're fearful, again, back to most drugs that end up being approved have a small incremental effect size. If you had a drug, so let's go back to what was being asked a moment ago. If there was a drug for metastatic melanoma that had 100% cure rate at 1 year, we would approve. We would be committing grievous harm by not getting that on the market as quickly as possible. So again, it's understanding. Is there a confounder to death? No. Is there a, if people don't die, if historically everyone was dead by between 6 months and 12 months, and now everyone's alive past 1 year, and no evidence of disease, and you have you have the historical control. I mean, we would just, if it was a single arm, totally observational from clinical practice, we would have a hard time not approving that. The problem is that you take a BRAF inhibitor that's approved for melanoma, let's say, and you move it into a new tumor type. You do the ASCO study, the TAPER trial, where they have evidence from clinical experience in a group of oncologists using it off-label for a different tumor, and it results in a 2-month greater survival or progression-free survival, if you want, either one, than what you would have expected. What do we do with that? We have no idea what to make of that. And in a randomized controlled trial, that may be all it does, but we have a randomized controlled trial, and we're confident in that result. So again, it's the effect size and understanding that it's almost a never can be event. Now, is there something in between that? Obviously, yes. But that's where the difficulty is figuring out where that something in between is.
Okay, great discussion. We're going to continue many of these tomorrow. I know Mark is going to take us home with a final conclusion. Thank you. Thank you, panelists.
Yeah, we are just finishing up now. I want to thank the panel for, as Greg said, a great discussion, and thank all of our panelists today and all of you for your contributions to this meeting on enhancing the application of real-world evidence and regulatory decision-making. We heard a general agreement that the increasing availability of real-world data and methods to turn it into evidence presents some opportunities, important opportunities, for improving regulatory decision-making and patient care. But we also noted some challenges around data collection and causal inference and this wide range of methods that can be applied to real-world data settings, from retrospective observational studies all the way through pretty sophisticated randomized and controlled designs, and those methods are becoming clearer. Suggests that there are a set of identifiable and potentially addressable issues in using real-world evidence to address regulatory needs to help promote better evidence for regulatory decision-making, as well as better evidence for doctors and patients to use in care. Tomorrow, we're going to focus more on some of these policy implications and on how to accelerate the development of a better infrastructure for decision-making and evidence development from real-world settings. So we hope you'll join us for that. It's same place, same time. I do want to say that we're scheduled to have a half-day meeting, and the weather is scheduled to include some snow and other complications. We are keeping an eye on it, and it's always in Washington, D.C., you never want to say these storms aren't going to amount to much, but the forecast at this point doesn't look like it will be very disruptive for activities like our conference. If that changes, we'll notify everyone about any adjustment in our start time or anything else. But at this point, we're definitely planning to go ahead as scheduled tomorrow morning right here, 9 o'clock. Given that we are webcasting and we can have people join remotely if necessary, we don't think we're going to need to make any changes, but we hope that all of you will be able to join us as scheduled in the morning. The schedule tomorrow includes sessions on building a 21st century evidence development infrastructure, what it should look like, how it can address some of the challenges and build on the progress that we've heard about today. We'll talk about incentives and policy options to support this, and you heard just on the last panel a lot of agreement about potentially more clarity about how this evidence can be used in regulatory decision-making, contributing to that. And we'll talk about engaging providers and patients as partners in this effort as well. So that's all coming tomorrow. Before we finish today, I want to quickly thank our speakers for their contributions, especially those who were members of our planning group for this set of real-world evidence activities. And that planning group, which has contributed a lot of expertise and time over the last few months, includes Janet Woodcock, Jonathan Jarrow, Alyssa Lavange, Melissa Robb, Sally Oken, Rich Platt, Adrian Hernandez, Sean Tunis, Marcus Wilson, Mark Berger, and Mark Overhage. I also want to thank our staff from the Duke Margolis Center who contributed to the planning and execution of these events, including Greg Daniel, who you just saw, Morgan Romine, Adam Atten, Pranav Arora, Wasim Nozair, Jonathan Bryan, Joanna Klatzman, Christina Flores, and Brittany Wise Goddard. And most of all, I'd like to thank all of you for attending and for the thoughtful comments and contributions throughout the day. All the event material from today will be available on our website. There will be some further steps from this conference. We'll talk more about those tomorrow. In the meantime, have a great evening, and we hope to see you all in the morning. Thank you very much.